बने रहो। पूरा एनसीईआरटी कंप्लीट आज करने जा रहे हैं। तो इसमें पहले यूनिट 1 लिविंग वर्ड करेंगे। तो सबसे पहले लिविंग वर्ड, इसमें 4 जितने भी चैप्टर हैं पूरे करेंगे। उसके बाद स्ट्रक्चर ऑर्गेनाइजेशन इन प्लांट एंड एनिमल, इसमें 5th मॉर्फोलॉजी इन फ्लावरिंग प्लांट, एनाटॉमी ऑफ फ्लावरिंग प्लांट, इसमें देखो सेकेंडरी ग्रोथ नहीं करेंगे। और स्ट्रक्चरल ऑर्गेनाइजेशन इन एनिमल्स करेंगे। और उसके बाद आएगा सेल बायोलॉजी, इसमें सेल यूनिट ऑफ लाइफ, बायोमोलिक्यूल्स, सेल साइकिल एंड सेल डिवीजन करेंगे। उसके बाद करेंगे प्लांट फिजियो में ट्रांसपोर्ट इन प्लांट, मिनरल न्यूट्रिशन दोनों नहीं करना है। फोटोसिंथेसिस इन हायर प्लांट, रिस्पायरेशन इन प्लांट, प्लांट ग्रोथ एंड डेवलपमेंट करेंगे। उसके बाद यूनिट 5 में ह्यूमन फिजियोलॉजी में डाइजेशन नहीं करना है क्योंकि सिलेबस में नहीं है। ब्रीथिंग एंड एक्सचेंज ऑफ गैसेस करेंगे। बॉडी फ्लूइड एंड सर्कुलेशन करेंगे। एक्सक्रीटरी प्रोडक्ट एंड देयर एलिमिनेशन। चैप्टर 20 लोकोमोशन एंड मूवमेंट। चैप्टर 21 न्यूरल कंट्रोल एंड कोऑर्डिनेशन। उसके बाद केमिकल कोऑर्डिनेशन एंड इंटीग्रेशन। ये चारों जितने भी यूनिट्स हैं पूरे चैप्टर एक वीडियो में करने वाले हैं। तो देखते हैं कितना टाइम लगता है भाई। अभी देख लूँ साउंड कैसा आ रहा, चेक करके बताता हूँ भाई। चलो साउंड वगैरह सही चल रहा है तो भाई स्टार्ट करते हैं महा मैराथन। 1 मिनट 1 मिनट कुछ। एक मिनट भाई करते हैं स्टार्ट थोड़ा। सो तो भाई रेडी हो, सीट बेल्ट बांध लो सब अपनी, करते हैं स्टार्ट चैप्टर को। इस चैप्टर में होल एनसीईआरटी कवर होने वाला भाई। सो लेट्स गो ब्रो। So first unit diversity in living world. Chapter on living world. Biology is the science of life form and living processes. The living world comprises of amazing diversity of living organisms. Early man could easily perceive difference between inanimate matter and living organism. Every man defied some inanimate matter, wind, sea, fire, etc. And some among animals and plants. A common feature of all such forms of inanimate and animate object was the sense of awe or fear that they evoked. The description of living organisms, including human beings, began much later in life human history. Societies which indulged in anthropocentric view of biology could register limited progress in biological knowledge. Systematic and monumental description of life form brought in out of necessity, detailed system identification, nomenclature and classification. The biggest spin of such study was recognition and sharing of similarities among living organisms, both horizontally and vertically, that all present-day living organisms are related to each other and also to all organisms that ever lived on this earth. Was a revelation which humbled man and led cultural movement and conservation of biodiversity. In the following chapter of this unit, you will get description including classification of animals and plant from taxonomics perspective. Ernest Mayr, 1904 to 2004. Born on 5 July 1904, Campton, Wait 1 minute, bro. Germany and Ernest Mayr, the Harvard University evolutionary biologist who has been called the Darwin of 20th century, was one of the 100 greatest scientists of all time. Mayr joined Harvard Faculty of Arts and Science in 1954 and retired in 1975, assuming the title Alexander Agassiz Professor of Zoology Emeritus. Throughout his early 18-year career, his research spanned ornithology, taxonomy, geography, evolution, systematics, and the history of philosophy of biology. He almost single-handedly made origin of species diversity the central question of evolutionary biology that it is today. He also pioneered the currently accepted definition of biological species. Mayr was awarded 3 prizes widely regarded as Triple Crown of Biology: Bazon Prize 1983, the International Prize for Biology in 1994, and Crawford Prize in 1999. Mayr died at the age of 100 years in 2004. So, Baljan in 1983, International Prize for Biology 1994, and Crawford Prize in 1999. So let's start chapter 1, the living world. So लिविंग वर्ड में अपन क्या पढ़ेंगे आज? लिविंग वर्ड, डाइवर्सिटी इन लिविंग वर्ड, टैक्सोनॉमिक कैटेगरी, टैक्सोनॉमिक एड्स. तो इसमें तो हर्बेरियम वगैरह हट गया, लेकिन वो भी करेंगे, कर लेंगे भाई, छोड़ेंगे नहीं उसको. हाउ वंडरफुल इज लिविंग वर्ड. The wide range of living type is amazing. The extraordinary habitats in which we find living organisms, be it old mountains, deciduous forest, ocean, freshwater lakes, desert, or hot spring, leaves us speechless. The beauty of galloping horse of the migrating birds, the oily of flower or attacking shark evokes awe and deep sense of wonder. The ecological conflict of cooperation among members of population and among population of the community, or even molecular traffic inside a cell, make us deeply reflect on what indeed is life. This question has two implicit questions within it. The first is technical, 1 and 6 answer to that what is living and as opposed to the non-living. And the second is philosophical, 1 and 6 answers to what purpose of life is. As scientists, we shall not attempt answering the second question. We will try to reflect on what is living. 1.1 What is living? When we try to define living, we conventionally look for distinctive characteristics exhibited by living organism: growth, reproduction, ability to sense environment and mount the suitable response come to our mind immediately as a unique feature of living organism. One can add a few more features like metabolism, ability to self-replicate, self-organize, interact, and emergence to this list. Let us try to understand each of these. A living organism grow. Increase in mass and increase in number of individuals are twin characteristics of growth. A multicellular organism grows by its cell division. In plants, this growth by cell division occurs continuously throughout their life span. In animals, this growth is seen only up to certain age. However, cell division occurs in certain tissue to replace lost cell. Unicellular organisms grow by cell division. One can easily observe this through culture by simply counting the number of cells under microscope. In majority of higher animals and plants, growth and reproduction are mutually exclusive events. One must remember that increase in body mass is considered as growth. Non-living objects also grow if we take increase in body mass as criterion for growth. Mountains, boulders, sand molds do grow. However, this kind of growth exhibited by non-living object is by accumulation of material on the surface. The living organism growth is from inside. Growth therefore cannot be taken as defining property of all living organism. Conditions under which it can be observed in all living organisms have to be explained, and then we understand what that it is characteristic of living system. A dead organism does not grow. Okay. Reproduction likewise is a characteristic of living organism. In multicellular organism, reproduction refers to production of progeny possessing features more or less similar to those of parents. Invariably and implicitly, we refer to sexual reproduction. Organisms reproduce by asexual means also. Fungi multiply and spread easily due to millions of asexual spores they produce. In lower organisms like yeast and hydra, we observe budding. In planaria flatworm, we observe through regeneration of fragmented organism regenerate the lost part of the body and become a new organism. The living in fungi, the filamentous algae or the protein mosses all easily multiply by fragmentation. When it comes to unicellular organisms like bacteria, unicellular algae or amoeba, reproduction is synonymous with growth. Increase in number of cells. We have already defined growth as equivalent to increase in cell number or mass. Hence, we notice that in single-celled organism, we are not very clear about the usage of these two terms, growth and reproduction. Further, there are many organisms which do not reproduce. Example, males, estradiol, worker, infertile couple, etc. Hence, in reproduction also cannot be all inclusive defining characteristic of living organism. Of course, no non-living object is capable of reproducing or replicating itself. Another characteristic of living metabolism. All the living organisms are made of chemical and the chemicals small and big belonging to the various classes, size and function, etc. are constantly being made and changed into some other biomolecules. These conversions are chemical reaction or metabolic reaction. There are thousands of metabolic reactions occurring simultaneously inside all living organisms, be the unicellular or multicellular. All plants, animals, fungi and microbes exhibit metabolism. The sum of total of all chemical reactions occurring in our body is metabolism. No non-living object exhibits metabolism. Metabolic reaction can be demonstrated outside the body in cell-free system. An isolated metabolic reaction outside the body of an organism performed in a test tube, neither living nor non-living. Hence, while metabolism is a defining feature of all living organisms without exception, isolated metabolic reaction in vitro are non-living things but surely living reaction. Okay. Hence, cellular organism body is defining feature of life form. Cellular organization is defining feature of life form. Perhaps the most obvious, technical and complicated feature of all living organisms is the ability to sense their environment, surrounding and environment, and respond to these environment stimuli, which could be physical, chemical or biological. We sense our environment through our sense organs. Plants respond to external factors like light, water, temperature and other organism, pollutant, etc. All organisms from the prokaryote to most complex eukaryote can sense and respond to environmental cues. Cues means sign. Photoperiod affect reproduction in seasonal breeders, both plants and animals. All organisms handle chemicals entering their body. All organisms therefore are aware of their surrounding. Human being is the only organism who is aware of himself and has self-consciousness. Consciousness therefore becomes the defining property of living organism. When it comes to human being, it is all more difficult to define living state. We observe patients in a coma in hospital, virtually supported by machine replaced by heart and lung. The patient is otherwise brain dead. The patient has no self-consciousness. Are such patients who never come back to normal living or non-living. In higher classes, you will come to know that all living phenomena are due to underlying interaction. Property of tissue are not present in constituent cell but arises as a result of interaction among the constituent cells. Similarly, property of cellular organelles are not present in molecular constituent of organelle but arises as a result of interaction among the molecular component comprises the organelle. These interactions result in emergence of property higher level of organization. This phenomenon is true in the hierarchy of organizational complexity at all levels. Therefore, we can say that living organisms are self-replicating, evolving, and self-regulating interactive systems capable of responding to external stimuli. Biology is the story of life on Earth. Biology is the story of evolution of living organisms on Earth. All living organisms, present, past, and future, are linked to the one another by sharing common genetic material but varying degree. Diversity in living world. If you look around, you will see a large variety of living organisms, be it potted plant, insect, bird, pet, or other animal plants. There are also several organisms that you cannot see with your naked eye, but they are all around you. If you were to increase the area that you make observation in, the range of and variety of organisms that you see would increase. Obviously, if you were to visit dense forest, you would probably see a much greater number and kind of living organism in it. Each different kind of plant and animal organism that you represent a specific number of species known to describe a wide range between 1.7 to 1.8 million. It represents biodiversity or number and type of organism present on Earth. We should remember here that as we explore new areas and even old ones, new organisms are continuously being identified. As stated earlier, there are millions of plant and animal in the world. We know plant and animal in our own area by their local names. These local names would vary from place to place. Even within a country, probably you would recognize the confusion that would create if we did not find ways to talk to each other to refer the two organisms we are talking about. Hence, there is a need of standardized naming of living organisms such that particular organism is known by the same name all over the world. This phenomenon is called nomenclature. Obviously, nomenclature or naming is only possible when organism described correctly, and we know that organism name is attached to this is known as identification. In order to facilitate the study, a number of scientists have established procedure to assign the scientific name to each known organism. This is acceptable to biologists all over the world. For plant, scientific names are based on agreed principles and criteria which are provided in International Code of Botanical Nomenclature (ICBN). You may ask, how are animals named? Animal taxonomists have evolved International Code of Zoological Nomenclature (ICZN). These scientific names ensure that each organism has only one name. Description of any organism should be enabled people in any part of the world arrive to the same name. They also ensure that such name has not been used for any other known organism. Biologists follow universally accepted principle to provide scientific name to no organism. Each name has two components, the generic name and a specific epithet. This system of providing a name with two components is called binomial nomenclature. This naming system given vicarious lineage is being practiced by biologists all over the world. This naming system using two-word format was found convenient. Let us take example of mango to understand the way providing scientific names better. The scientific name of mango written as Mangifera indica. Let us see how it binomial name. In this name, Mangifera represents genus, while indica is particular species of specific epithet. Other universal rules of nomenclature are as follows. First, biological name given in Latin and written in italics. They are Latinized and derived from Latin irrespective of their origin. Second, the first word of biological name represents genus, while the second component denotes a specific epithet. Third, both the word in biological name when handwritten are separately underlined and or printed in italics to indicate their Latin origin. 4. The first word denoting the genus start with capital letter, while the specific epithet start with small letter. It can be illustrated from example Mangifera indica. Name and author appear Name of the author appear after the specific epithet that is at the end of biological name and written in abbreviated form, like that is Mangifera indica Lin. They indicate that this species was first described by Linnaeus. Since it is nearly impossible to study all living organisms, it is necessary to devise some means to make this possible. This process is classification. Classification is the process by which anything is grouped into convenience category based on some easily observable characters. For example, we easily recognize groups such as plants or animals, or dog, cats, or insects. The moment we use any of these terms, we associate certain characters with organism in that group. What image do you see when you think of a dog? Obviously, each one of we see dogs and not cat. Now we were think Alsatians. We know that we are talking about. Similarly, suppose we are to say mammals, you would of course think of animals with external ears and body hairs. Likewise, in plants, if you try to talk of wheat, the picture in our mind will be wheat plants, not rice or any other plants. Hence, all these dogs, cats, mammals, wheat, rice, plants, animals, etc. are convenience category we use to study organism. The scientific term of this category is taxa. Here you must recognize that taxa can indicate very different level. Plants also form taxa. Wheat is also taxa. Similarly, animals, mammal, dog are all taxa, but you know that dog is mammal are animals. Therefore, animals, mammals, and dogs represent taxa at different level. Hence, based on characteristics, all living organisms can be defined into different taxa. This process of classification is taxonomy. External and internal structure, along with the structure of cell development process and ecological information of organism, are essential and form the basic modern taxonomic study. Hence, characterization, identification, classification, and nomenclature are the process that are basic of taxonomy. Taxonomy is not something new. Human beings have always been interested in knowing more and more about the various kind of organisms, particularly with reference to their own use. In early days, human beings needed to find sources for their basic need of food, clothing, and shelter. Hence, the earliest classifications were based on use of various organisms. Human beings were since long not only interested in knowing more about different kind of organisms and their diversity, but also relationship among them. This branch of study was referred to as systematics. The word systematic is derived from the Latin word systema, which means systematic arrangement of organisms. Linnaeus used Systema Naturae as the title of his publication. The scope of systematics was later enlarged to include identification, nomenclature, and classification. Systematics take place into account of evolutionary relationship between organisms. 1.3 Taxonomic categories. Classification is not single step process but involve hierarchy step in which each step represent a rank or category. Since the category is a part of overall taxonomic arrangement, it is called taxonomic category, and all categories together constitute taxonomic hierarchy. Each category referred to as unit of classification, in fact, represent a rank, and commonly termed taxon, plural taxa. Taxonomic category and hierarchy can be illustrated by an example. Insects represent a group of organisms sharing common feature like 3 pairs of jointed legs. It means insects are recognizable concrete object which can be classified, and thus we given a rank and categories. Can you name other such group of organism? Remember, group represents category further denotes rank. Each rank or taxon in fact represent a unit of classification. This taxonomic group categorize are distinct biological entities, not merely morphological and aggregates. Taxonomic study of all known organisms has led to the development of common categories such as kingdom, phylum, or division. For plants, class, order, family, genus, and species. All organisms, including those in plants and animal kingdom, have species as the lowest category. Now the question you may ask is how to place an organism in various category. The basic requirement is knowledge of character of an individual or group of organism. This helps in identifying similarities and dissimilarities among individuals of the same kind as well as other kind of organism. 1.3.1 Species. Taxonomic study considers as a group of individual organisms with fundamental similarities as a species. One should be able to distinguish one species from other closely related species based on the distinct morphological differences. Let us consider Mangifera indica, Solanum, tuberosum, potato, Panthera, leo, lion. All the three named indica, tuberosum, and leo represent a specific epithet, while the first one, Mangifera, Solanum, Panthera, are just genera and represent another higher level of taxon or category. Each genus may have one or more than one specific epithet representing different organisms but having morphological similarity. For example, Panthera has another specific epithet called tigris, and Solanum include nigrum and melogena. Human being belong to species sapiens, which is grouped in genus Homo. The scientific name thus of human beings is written as Homo sapiens. 1.3.2 Genus. Genus comprises a group of related species which has more characters in common in comparison to species and other genera. We can say that genera are aggregates of closely related species. For example, potato and brinjal are two different species but both belong to genus Solanum. Lion, Panthera leo, leopard, Panthera pardus, and tiger, Panthera tigris, with several common features, are all species of genus Panthera. This genus differs from another genus Felis, which includes cat. 1.3.3 Family. The next category, family, has a group of related genera with still less number of similarities compared to genus and species. Families are characteristic on the basis of both vegetative and reproductive feature of plants species. Among plants, for example, three different genera, Solanum, Petunia, Datura, are placed in family Solanaceae. Among animals, for example, genus Panthera comprises of lion, tiger, leopard is put along with a genus Felis. Cat is in the family Felidae. Similar, if you observe the feature of cat and dog, you will find some similarity and some difference as well. They are separated into two different families, Felidae and Canidae respectively. 1.3.4 Order. You have seen earlier category like species, genus, and families based on a number of similar characters. Generally, order and other higher taxonomic category and identify based on the aggregate of characters. Order being higher category is the assemblage of families which exhibit a similar characters. These similar characters are less in number as compared to different genera included in a family. Plant families like Convolvulaceae, Solanaceae are included in the order Polymoniales, mainly based on the floral characters. The animals order Carnivora include family like Felidae and Canidae. 1.3.5 Class. This category includes related order. For example, order Primata comprises of monkey, gorilla, and gibbon is placed in the class Mammalia along with the order Carnivora that include animals like tiger, cat, and dog. Class Mammalia has other order too. Okay. 1.3.6 Phylum. Class comprises of animals like fishes, amphibians, reptiles, birds, along with the mammals constitute the next higher level category called phylum. All these based on the common feature like presence of notochord and dorsal hollow neural system are included in phylum Chordata. In case of plant, classes with similar character are assigned to higher categories called division. 1.3.7 Kingdom. All animals belonging to various phylum are assigned to the highest category called kingdom. Kingdom Animalia in the classification system of animals. The kingdom Plantae, on the other hand, is distinct and comprises all plants from various divisions. Henceforth, we will refer to these two kinds groups of animals and plant kingdom. The taxonomic category from species to kingdom have been shown in ascending order, starting from with species in 11 figure 1.1. These are broad categories. However, taxonomists have also developed subcategories in this hierarchy to facilitate more sound and scientific placement of various taxa. Look at the hierarchy in figure 1.1. You can recall the basis of arrangement. Say, for example, as you go higher from species to kingdom, the number of common characters goes on decreasing. Lower the taxa, more the characteristic that the member within the taxon share. Higher the category, greater the difficulty of determining the relationship to the taxa at the same level. Hence, the problem classification becomes more complex. Taxonomic category showing hierarchical arrangement in ascending order: kingdom, phylum, class, order, family, genus, species. Mnemonics: Kohli, play cricket on flat grounds. Table 1.1 indicates the taxonomic category to which some common organisms like housefly, man and mango and wheat belongs. Table 1.1 organism with their taxonomic categories. Common name, biological name, genus, family, order, class, phylum or division. So man, biological name Homo sapiens. Genus Homo, family Hominidae, order Primata, class Mammalia, phylum Chordata. Housefly, biological name Musca domestica, genus Musca, family Musidae, order Diptera, class Insecta, phylum or division Arthropoda. Mango, Mangifera indica, genus Mangifera, family Anacardiaceae, order Sapnidales, class Dicotyledon, phylum or division Angiosperm. Wheat, biological name Tritica estium, genus Triticum, family Poaceae, order Poace, class Monocotyledon, phylum Angiosperm. 1.4 Taxonomic aids. Taxonomic study of various species of plant and animals and other organisms are useful in agriculture, forestry, industry and general in knowing our biological bioresources and their diversity. These studies would require correct classification and identification of organism. Identification of organism require intensive laboratory and field studies. The collection of actual specimen of plant and animal species is essential and is the prime source of taxonomic studies. These are also found fundamental to these and essential for training in systematics. It is used to classification of an organism, and information gathered in is also stored along with a specimen. In some cases, specimens are preserved for future studies. Biologists have established certain procedures and techniques to store and preserve information as well as specimens. Some of these are explained to help you understand uses of these aids. 1.4.1 Herbarium. Herbarium is a storehouse of collected plant species that are dried, pressed and preserved on seed. Further, these seeds are classified according to universally accepted system of classification. These specimens, along with their description on herbarium seed, become a storehouse or repository for future use. The herbarium seed also carry a label providing information about date and place of collection, English and local botanical name, family, collector name, etc. Herbaria also serve as quick reference system in taxonomical studies. Herbarium showing stored specimen. 1.4.2 Botanical Garden. These specialized garden have collection of living plants for reference. Plant species in these gardens are grown and for identification purposes, and each plant is labeled indicating its botanical or scientific name and its family. The famous botanical garden are at Kew, England, Indian Botanical Garden, Howrah, and National Botanical Research Institute, Lucknow, India. 1.4.3 Museum. Biological museums are generally set up in educational institutes such as school and college. Museums have a collection of preserved plants and animal specimens for study and reference. Specimens are preserved in the container or jar in preservative solution. Plants and animal specimens may also be preserved and ripe specimens. Insects are preserved in insect boxes after collecting, killing, and pinning. Large animals like birds, mammals are usually stored and preserved. Museums often have collection of skeletons of animals too. 1.4.4 Zoological parks. These are places where wild animals are kept in protected environment under human care, and which enables us to learn about their food habits and behavior. All animals in a zoo are provided as far as possible the conditions similar to their natural habitat. Children love visiting this park, commonly known as zoo. Picture showing animals, different zoological parks of India: rhinoceros, swan, elephant, tiger. Key is another taxonomic aid used in the identification of plants and animals based on similarity and dissimilarity. The key are based on the contrasting characters, generally in pair called couplet. It represents choice made between the two opposite options. This represents the acceptance of one and only one and rejection of other. Each statement is called a lead. Separate taxonomic keys are required for taxonomic categories such as family, genus, species for identification purpose. Keys are generally analytical in nature. Flora, manuals, monograph, and catalogs are some other means of recording descriptions. They also help in correct identification. Flora contains the actual account of habitat and distribution of plant of a given area. These provide the index of the plant species found in a particular area. Manuals are useful in providing information for identification of names of species found in an area. Monograph contain information of any one taxon. Summary: the living world is rich in variety. Millions of plants and animals have been identified and described, but a large number of still remain unknown. The very range of organisms in terms of size, color, habitat, physiological and morphological features make us seek the defining characteristics of living organisms. In order to facilitate the study and kinds of diversity in organisms, biologists have evolved certain rules and principles due to identification, nomenclature, and classification of organisms. The branch of knowledge dealing with these aspects is referred to as taxonomy. The taxonomic studies of various species of plants and animals are useful in agriculture, forestry, industry, and in general for knowing our bioresources and their diversity. The basics of taxonomic, like identification, naming, and classification of organisms, are universally evolved under international code based on resemblance and distinct differences. Each organism is identified and assigned a correct scientific biological name comprising two words as per binomial system nomenclature. An organism represents or occupies a place or position in system of classification. There are many categories. Ranks are generally referred to as taxonomic category or taxa. All the categories constitute a taxonomic hierarchy. Taxonomists have evolved a variety of taxonomic aids to facilitate identification, naming, and classification of organisms. These studies are carried out from actual specimens which are collected from the field and preserved as reference in the form of herbaria, museum, and in botanical garden and zoological parks. It requires special techniques for collection and preservation of specimens in herbaria and museum. Live specimens, on the other hand, of plants and animals are found in botanical garden or in zoological park. The taxonomists also prepare the disseminate information through manuals and monographs for further taxonomic studies. Taxonomic key are tools that help in identification based on characters. सो भाई फर्स्ट चैप्टर अपना कंप्लीट हो गया. ये तो टैक्सोनॉमिक उतना आने वाला नहीं है, इन सबको छोड़ सकते हो, हर्बेरिया वगैरह. टैक्सोनॉमिक एड्स ये मोस्ट इम्पॉर्टेंट मैं लिख कर दे रहा हूँ. 2025 में अगर इस चार्ट से क्वेश्चन ही आया ना भाई, नहीं आया क्वेश्चन तो क्या कुछ नहीं, लेना आएगा, पढ़ लो. ये कोहली खेल रहा है, प्ले क्रिकेट ऑन फ्लैट ग्राउंड, ट्रिक से याद कर लो. ओके. सो फाइनली एक चैप्टर कंप्लीट हुआ. अब टोटल बचे हैं अपने इस यूनिट में एक बचा है बायोलॉजिकल क्लासिफिकेशन, प्लांट किंगडम, एनिमल किंगडम. तो इन बालजन 1983 में दिया और इंटरनेशनल प्राइज ऑफ बायोलॉजी 1994, क्राफोर्ड प्राइज 1999 और नेक्स्ट में 100 साल जिया बंदा, देख रहा हो. लिविंग वर्ड से रिलेटेड कुछ यहाँ पढ़ा गया. लिविंग कौन है उसका कुछ कैरेक्टर पढ़ा गया. इधर रिप्रोडक्शन कैरेक्टरिस्टिक है या नहीं है, क्योंकि मान लो जैसे म्यूल वगैरह हैं या वर्कर भी नॉन रिप्रोडक्टिव होते हैं, इनफर्टाइल इन लिविंग है. मेटाबॉलिज्म हाँ है, ये कैरेक्टर होगा. डिफाइनिंग फीचर है. कॉन्शियसनेस ये डिफाइनिंग प्रॉपर्टी है. सेल्फ कॉन्शियसनेस ये सिर्फ ह्यूमंस में होता है. ओके ब्रो, ओके. डाइवर्सिटी 1.7 to 1.8 मिलियन होता है. इतना हो गया नॉमेनक्लेचर आइडेंटिफिकेशन. ICV कोड करता है प्लांट वगैरह के लिए, ICZ एनिमल्स के लिए हो गया. जेनरिक स्पेसिफिक बाइनोमियल नोमेनक्लेचर कैरोलस लीनियस दिया हो गया. मैंजिफेरा इंडिका, इसके आगे उसका नाम लिख देते हैं किसका, जो साइंस जो साइंटिस्ट उसको डिस्कवर किया उसके बाद लिखा जा सकता है. यहाँ पे लैटिन होता है स्पेसिफिक इफिटेड. चलो भाई ये कंप्लीट हुआ ना, बहुत हुआ. अब आगे का यहाँ पे कुछ इम्पोर्टेंट जीनस जैसे पैंथेरा, लायन, लियोपार्ड, टाइगर, तीनों इसी में आ रहा. और पोटैटो के लिए सोलेनम में तुम्हारा ब्रिंजल भी आ रहा, पोटैटो भी आ रहा. जीनस है. फैमिली देखो, फैमिली का ये वेजिटेटिव और रिप्रोडक्टिव फीचर के बेस पे फैमिली को बांटा गया. सोलेनम, पेटूनिया, डटूरा. फैमिली किस में आता है, सोलनेसी में. सोलनेसी फैमिली में ये सब जीनस आते हैं, सोलेनम, पेटूनिया, डटूरा. और फेलिडाई और कैनिडाई फैमिली फेलिडाई और कैनिडाई में इनको रखा गया. इसमें कैट आता है और इसमें डॉग आएगा. और ये सब ऑर्डर कार्निवोरा में आ जाएंगे, फेलिडाई और कैनिडाई. और कन्वोलेसी, सोलनेसी जो फैमिली है, इसको ऑर्डर पॉलीमोनियल्स में रखा गया. अब देखो मैमेलिया along with carnivora include animals like tiger, cat, class Mammalia, other order also. फाइलम में देखो हॉलो डॉर्सल हारो न्यूरल किस में पाया जा रहा है, कॉर्डाटा में होता है. ठीक है. किंगडम के बारे में इसमें दिया है कुछ, ये दोबारा देख लेना. चलो भाई दूसरा चैप्टर स्टार्ट करते हैं, लेट नहीं करेंगे ज़्यादा. चैप्टर 2 बायोलॉजिकल क्लासिफिकेशन. रेडी हो भाई, नॉन स्टॉप। तो इसमें अपन क्या पढ़ेंगे, फर्स्ट किंगडम मोनेरा, उसके बाद क्या करेंगे किंगडम प्रोटिस्टा, उसके बाद किंगडम फंजाई, उसके बाद किंगडम प्लांटी, उसके बाद किंगडम एनिमिलिया, वायरस वायराइड एंड लाइकेन। तो इसका कुछ ओवरव्यू कर लेते हैं। सिंस dawn of civilization there has been many attempts to classify living organism. It was distinct not using criteria that were scientific but born out need to be organism of our own use for shelter, food, shelter and clothing. Aristotle was earliest to attempt more scientific basis for classification. He used simple morphological character to classify plant into herbs, shrubs, and shrubs, herbs. He also divided animals into two groups: those which had red blood cell and those that did not. In lineage time, a two kingdom system classification with plantae and animalia. Animalia kingdom was developed that include all plants and animals respectively. This system did not distinguish between eukaryotic and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi. Classification of organism into plant and animalia was easily done and was easy to understand, but a large number of organisms that did not fall into either category since two kingdom classification used for a long time was found inadequate. Beside gross morphological, a need was also felt of including all characteristics like cell structure, nature of cell, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system from the living organism have hence undergone several changes over the time. Though plant and animals kingdom have been a constant under all different systems, the understanding of what group and organism be included under this kingdom have been changing. The number and nature of other kingdom also been understood differently by different scientists over the time. Table 2.1 Characteristic of 5 kingdom. Characters: cell type Monera prokaryotic, Protista eukaryotic, Fungi eukaryotic, Plantae eukaryotic, Animalia eukaryotic. तो देखो इसको ऐसे देख लेते हैं। मोनेरा में सेल टाइप प्रोकैरियोटिक, सेल वॉल क्या होता है नॉन सेलुलोजिक, पॉलीसेकेराइड एंड प्लस अमीनो एसिड, न्यूक्लियर मेम्ब्रेन एब्सेंट होता है, बॉडी ऑर्गेनाइजेशन सेलुलर होता है, मोड ऑफ न्यूट्रिशन ऑटोट्रॉपिक, कीमोसिंथेटिक एंड फोटोसिंथेटिक एंड हेट्रोट्रॉफिक, सैप्रोफाइटिक एंड पैरासाइटिक। प्रोटिस्टा में यूकैरियोट, सेल वॉल प्रेजेंट इन सम, न्यूक्लियर मेम्ब्रेन प्रेजेंट, बॉडी ऑर्गेनाइजेशन सेलुलर, ऑटोट्रॉपिक, फोटोसिंथेटिक एंड हेट्रोट्रॉफिक, मोड ऑफ न्यूट्रिशन। फंजाई में सेल वॉल यूकैरियोट, सेल टाइप यूकैरियोट, सेल वॉल प्रेजेंट विथ काइटिन, न्यूक्लियर मेम्ब्रेन प्रेजेंट, मल्टीसेलुलर एंड लूज टिश्यू, सेल ऑर्गेनाइजेशन, मोड ऑफ न्यूट्रिशन हेट्रोट्रॉफिक, सैप्रोफाइटिक एंड पैरासाइटिक। प्लांटी सेल टाइप यूकैरियोट, सेल वॉल present made of cellulose, nuclear membrane present, body organization tissue or organ, mode of nutrition autotrophic, photosynthetic. Animalia cell type eukaryote, cell wall absent, nuclear membrane present, body organization tissue organ or organ system, mode of nutrition heterotrophic, holozoite and or saprophytic. So let's start करो भाई, खेले यहाँ से। R.H. Whitaker 1969 proposed a 5 kingdom classification The kingdom defined by him was named Monera protista fungi plantae and animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of living five kingdoms. The three-domain system has also been proposed that divide the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain, and thereby a six-kingdom classification. You will learn about the system in detail at higher classes. Let us look at this five kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, fern, gymnosperm, and angiosperm, and the plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together group which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other group which were eukaryote. It also grouped together unicellular organisms and multicellular organisms in one example. For example, chlamydomonas and spirocheta were placed together under algae. The classification did not differentiate between heterotrophic group like fungi and autotrophic group like green plant. Though they also shows a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had cellulosic cell wall. When such characteristics were considered, the fungi were placed in separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas colladella, earlier placed in algae within plants and both having cell wall, with Paramecium and amoeba, which were earlier placed in animal kingdom which lacked cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will not take place in future too, depending on improvement in our understanding of characteristic and evolutionary relationships. Over time, an attempt has been made to evolve classification system which reflect not only the morphological, physiological, and reproductive similarities, but it also phylogenetic, that is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista, fungi of Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plants and animal kingdom respectively, will deal separately in chapters 3 and 4. 2.1 Monera. Bacteria are sole members of kingdom Monera. They are most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow, and deep ocean where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shapes: the spherical coccus, pleural cocci, and the rod-shaped bacillus, pleural bacilli, the common-shaped vibrio, pleural vibrio, and spirillum, pleural spirilla. Bacteria of different shapes: cocci, bacilli, spirilla, vibrio. देखो स्पाइरला में फ्लैजिलम है और बैसिली में स्पोर है. क्वेश्चन आ जाएगा स्पोर्स प्रेजेंट इन बैसिलिस एंड फ्लैजिला प्रेजेंट इन स्पाइरला. Though the bacterial structure is very simple, they are very complex in behavior. तो बैक्टीरियल स्ट्रक्चर क्या है, सिंपल है, बट बिहेवियर क्या है, कॉम्प्लेक्स है. Compared to many other organisms, bacteria as a group shows the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophic, that is, they depend on other organisms or dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, halophils, hot springs, thermohydrophils, and marshy areas. Methanogens. Archaebacteria differ from other bacteria in having different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals, such as cow, buffaloes, and they are responsible for production of methane biogas from dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by presence of a rigid cell wall. If motile of flagellum, the cyanobacteria also differ from blue-green algae and have a chlorophyll A similar to green plants and are photosynthetic autotroph. The cyanobacteria are unicellular, colonial, filamentous, freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They are often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst. That is nostoc and anavena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume, root, etc. Some are pathogen causing damage to human beings, crop, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker, as well as known disease caused by different bacteria. Bacteria are produced mainly by fission. Sometimes under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primary type of DNA transfer from one bacterium to other. The mycoplasma are the organisms that completely lack a cell wall. They are the smallest living organism known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Or dividing bacterium: DNA, cell membrane, cell wall. Filamentous blue-green algae nostoc, heterocyst, mucilaginous sheath, and this is heterocyst. 2.2 Kingdom Protista. A single-celled eukaryote are placed in protista, but the boundary of this kingdom are not well defined. What may be the photosynthetic protista to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenites, slime molds, and protozoans under protista. Members of protista are primarily aquatic. This kingdom form a link with other dealing with plants, animals, and fungi. Being eukaryotic, the protestant cell body contain of well-defined nucleus and other membrane-bound organelles. Some have a flagella or cilia. Protista reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. The group Group includes diatoms and golden algae desmites. They are found in freshwater as well as in marine environment. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatom, the cell wall form two thin overlapping cells which fit together in a soap box. The walls are embedded with silica and thus the walls are indestructible. The diatom have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years referred as diatomaceous earth. Being gritty, this soil used in polishing, filtration of oil, syrups. Diatoms are chief producer of ocean. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on their pigments present in their cell. The cell wall has steep cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in furrow between two wall plates. Very often, red dinoflagellates, example, goniologs, undergo such rapid multiplication that they make the sea appear red. Red tide toxins released by such large number may even kill other marine animals such as fishes. Here is dinoflagellate euglenoids. Majority of them are freshwater, found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle, which make their body flexible. They have two flagella, a short and long one. Though they are photosynthetic in presence of sunlight, when deprived sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoids are identical to those present in higher plants. Example, euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and limbs, engulfing organic material under suitable conditions. They form an aggregation called plasmodium, which may grow and spread over several feet. During the unfavorable conditions, the plasmodium differentiate and form floating bodies bearing spores at their tip. The spore passes through walls. They are extremely resistant to survive for many years, even under adverse conditions. The spores are dispersed by air current. Here is slime mold. Here is euglenoids. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predator or parasite. They are believed to be primitive relative of animals. There are four major groups of protozoans. First, amoeboid protozoans. These organisms live in fresh water, seawater, and moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica cell on their surface. Some of these, such as ant amoeba, are parasite. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause diseases such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms. Live because of presence of thousands of cilia. They have a cavity gullet that opens to outside of the cell surface. The coordinate movement of rows of cilia cause the water laden with food to slide into gullet. Example, paramecium. Sporozoans. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease with staggering effect on human population. 2.3 Kingdom Fungi. Fungi constitute a unique kingdom of heterotrophic organisms. They show great diversity in morphology and habitat. You must have seen fungi on moist bread and rotten fruit. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaf are due to parasitic fungus. Some unicellular fungi that is yeast are used to make bread and beer. Other fungi cause disease in plants and animals. Wheat rust causes Puccinia is an important example. Some are the sources of antibiotics, that is penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in refrigerator? Yes, it prevents food from going bad due to bacterial and fungal infection. With the exception of yeast, which are unicellular fungi, are filamentous. Their bodies consist of a long thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous through tube find with multinucleated cytoplasm. These are called xenocytic hyphae. Others have separate cross wall in their hyphae. The cell wall of fungi are composed of chitin polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence called saprophytes. Those that depend on living plants and animals are parasites. They can also live as symbionts in association with algae, as lichen with the roots of higher plant mycorrhiza. Reproduction of fungi can take place by vegetative means fragmentation, fission, and budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and sexual reproduction is by oospores or ascospores and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile and non-motile gametes called plasmogamy. Second, fusion of two nuclei called chirogamy. And third, meiosis of zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately results in diploid cell 2n. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei each per cell occur. Such condition is called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cell become diploid. The fungi form floating body in which reduction division occur, leading to formation of haploid spores. The morphology of the mycelium mode of spore formation and floating body form basis of division into various kingdom. The morphology of mycelium mode of spore formation, floating body form the basis of kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes found in aquatic habitat and decaying woods in moist and damp places or as obligate parasite on plants. The mycelium is aseptate and xenocytic. Asexual reproduction takes place by zoospore motile or by aplanospore non-motile. These spores are endogenously produced in a sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogametes, or dissimilar in isogametes or oogametes. Some common examples are mucor rhizopus, the bread mold mentioned earlier, albugo-parasitic fungi, and mustard. Here is mucor aspergillus, and this is agaricus. Ascomycetes 2.3.2 Commonly known as sac fungi, the ascomycetes are mostly multicellular, that is penicillium, or rarely unicellular, that is yeast Saccharomyces. They are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is a branch and septate. The asexual spore motile conidia produce exogenously on special mycelium called conidiophore. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are reproduced endogenously in a sac like asci, singular ascus. These asci are arranged in different type of fruiting bodies called ascocarps. For some examples are Aspergillus, Claviceps, Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and considered delicacies. 2.3.3 Basidiomycetes. Commonly known from Basidiomycetes are mushroom bracket fungi and puffballs. They grow in soil or on logs and tree stumps, and in living plant bodies are parasites, that is rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent. Plasmogamy is brought about by fusion of two vegetative or somatic cells of different strands or genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, mushroom, Ustalago is not, Puccinia, rust fungus. 2.3.4 Deuteromycetes. Commonly known as imperfect fungi because only the asexual or vegetative phase of these bacteria are known. When the sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative state have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkage were stabilized, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect sexual stage of members of Deuteromycetes were discovered, they often moved to Ascomycetes or Basidiomycetes. The Deuteromycetes reproduce only by sexual spores known as conidia. Only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposer on litter and help in mineral recycling. Some examples are Alternaria, Colentotrichum, Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plant or parasite. Bloodwort Venus flytrap, for example, an insectivorous plant, and Cuscuta is parasitic. The plant cell have an eukaryotic structure with prominent chloroplast, and cell were mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plant include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Life cycle of plant has distinct phases: the diploid, saprophytic, and haploid gametophytic, that alternate with each other. The length of haploid and diploid phase and whether these phases free living or dependent on others vary among different groups in plants. This phenomenon is called alternation of generation. You will study further in detail in class chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell like cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have definite shape and size. higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development, silent feature of various phyla described in chapter 4. 2.6 Virus, viroid, prions, and lichens. In 5 kingdom classification, we take there is no mention of lichen, and some acellular organisms like virus, viroids, prions. These are briefly introduced here. All of us who have suffered ill effect on common cold flu know what effect of virus and can have on us, even if we do not associate it with our condition. Viruses did not find a place of classification since they are not considered truly living. If we understand the living as those organisms that have cell structure, the viruses are non-cellular organisms that are characterized by having inert crystalline structure outside the living cell. Here is TMB, bacteriophage, capsid, RNA, capsids, bacteriophage, tail fiber, sheath, collar, head. Once they infect the cell, take over machinery of host cell to replicate themselves, killing the host. Would you call virus cell living or non-living? The name of virus, that means venomous or poisonous fluid, was given by Dmitry Ivanovsky in 1892 to recognize certain microbes as casual organism of mosaic disease of tobacco. These were found to be smaller than bacteria because they pass through the bacteria-proof filters. M.W. Vizernik, 1898, demonstrated that the extract of infected plants of tobacco could cause infection in healthy plant and called fluid Candidium ovum fluidium, infectious living fluid. W. Stanley, 1935, showed a virus that could crystalline crystal consist of largely of protein. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to protein, viruses also contain genetic material that could be RNA or DNA. No virus contain both RNA or DNA. A virus is nucleoprotein and genetic material is infectious. In general, viruses that infect plants are single-stranded RNA. Viruses that infect animals have single or double-stranded RNA or double-stranded DNA. Bacterial virus, bacteriophage, infect bacteria are usually double-stranded viruses. The protein coat called capsid, made of a small subunit called capsomeres, protect the nucleic acid. These capsomeres are arranged in helically polyhedral geometrical form. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by agent consisting of abnormally folded proteins. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSC), commonly called mad cow disease in cattle, and analogous to variant CR-Jacob disease (CJD) in humans. Lichen is a symbiotic association, mutually useful association between algae and fungi. The algae component is called phycoband and fungal component called mycoband, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb nutrients and water for partner. So a close association of these are like in nature would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plant and animal are first proposed by Aristotle on the basis of their morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed the elaborates five kingdom classification: Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of five kingdom classification were cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. In the five kingdom classification, the bacteria included kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extent to metabolic diversity. Bacteria may be autotrophic, heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophyte, dinoflagellate, euglenoids, slime mold, protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom Fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are four classes under this kingdom. The plant include all eukaryotic chlorophyll-containing organisms. Algae, bryophyte, teratophyte, gymnosperm, and angiosperm are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and saprophytic generation, which the heterotrophic eukaryote multicellular organisms lacking cell wall are included in animal in kingdom Animalia. The mode of nutrition of organism is holozoic. They reproduce mostly by sexual mode. Some asexual acellular organisms like virus and viroids are as well as lichens are not included in फाइव किंगडम सिस्टम क्लासिफिकेशन. तो भाई अपना कंप्लीट हुआ चैप्टर सेकेंड. बहुत टाइम लगा रहा हूँ यार, बहुत स्लो मैं पढ़ रहा हूँ. ऐसे में तो लग रहा है कि दिन के 12 बजने वाले हैं कंप्लीट करते करते. थोड़ा फास्ट करना पड़ेगा. तो चैप्टर सेकेंड कंप्लीट हुआ. बायोलॉजिकल क्लासिफिकेशन. इसमें कैरेक्टरिस्टिक ऑफ फाइव किंगडम क्लासिफिकेशन पढ़े. उधर किंगडम मोनेरा में कुछ पढ़े. इसमें आर्के बैक्टीरिया, यू बैक्टीरिया पढ़े. उसके बाद प्रोटिस्टा पढ़े. उसके बाद इस प्रोटिस्टा में क्राइसोफाइट्स, डाइनोफ्लैजेलेट्स, यूग्लिनोइड्स पढ़े. स्लाइमोड्स भी था. प्रोटोजोअन्स भी था. प्रोटोजोअन्स में आता है एमेबॉइड था, फ्लैजेलेटेड प्रोटोजोअन्स था, सिलेटेड प्रोटोजोअन्स था, स्पोरोजोअन्स था. उसका फंजाई की है. फंजाई में अपन एक ये प्लाज्मोगेमी, कैरियोगेमी, उसका मियोसिस ऑफ जाइगोट, और फाइकोमाइसिटीज करे, एस्कोमाइसिटीज करे, बेसिडियोमाइसिटीज करे, ड्यूट्रोमाइसिटीज करे. उसका प्लांटिकोरम कुछ यहाँ पे दिया. ब्लैडर वॉट, विनस फ्लाइट ऑफ इंसेक्टिवोरस है. कस्क्यूटा क्या है, पैरासाइटिक. किंगडम एनिमिलिया करा. उसके बाद वायरसेस, वायरोइड, एम्ब्रियॉन्स एंड लाइकेंस के बारे में पढ़ाई हुआ. ठीक. वायरोइड्स क्या होते हैं, वायरस क्या होते हैं, लाइकेंस. यहाँ पे ये याद कर लेने वाली बात है. डिमेट्रियोनस की 1892 में मोज़ेक टीएमबी को डिस्कवर किया. उसके बाद बैक्टीरिया प्रूफ, एमडब्ल्यू विजिटिंग कंटेजियम फ्लूइडियम, डब्ल्यू में स्ट्राइंग गया क्रिस्टल फॉर्म में. उसका वायरोइड क्या है, आरएनए लो मॉलिक्यूलर वेट का. ठीक. प्रियॉन्स क्या है, प्रोटीन है. प्रोटीन का बना हुआ इन्फेक्शियस पार्टिकल्स. लाइकेन के बारे में पढ़े. तो लो भाई अब चैप्टर 3 प्लांट किंगडम. इसमें पहले करेंगे एल्गी, ब्रायोफाइट, टेरिडोफाइट, जिम्नोस्पर्म, एंजियोस्पर्म. प्लांट लाइफ साइकिल. अल्टरनेशन ऑफ जनरेशन नहीं करना है क्योंकि ये हट गया. तो इतना करेंगे तो 2 मिनट का भाई मैं रेस्ट चाहता हूँ. 2 मिनट 41 हो रहा है. 43 पे स्टार्ट करेंगे ये करना. 2 मिनट का रेस्ट करने दो भाई. सो लेट्स गो फुल एनर्जी के साथ भाई लोग. भाई जिंदा है अभी. पूरा पढ़ना है भाई. रुकना नहीं है, थकना नहीं है, हारना नहीं है, हार मानना नहीं है. दिखा देना है बायोलॉजी को, बायोलॉजी की औकात क्या है. ठीक है भाई. दिखा देना है बायोलॉजी को, बायोलॉजी की औकात क्या है, और अपने अंदर कितनी एनर्जी है, कितनी ऊर्जा है, उसको दिखाना है यहाँ पे. तुम्हारा भाई तुम्हारे साथ है भाई. लग जाओ, आ जाओ. करते हैं अब प्लांट किंगडम स्टार्ट मेरे भाई लोग. तो भाई रेडी हो भाई. करते हैं स्टार्ट. कैसे? चलो मेरे भाई लोग स्टार्ट. आ जाओ, करते हैं प्लांट किंगडम. In various chapter we looked broad classification of living organism under the system proposed by Whitaker in 1969, wherein he suggested 5 kingdom classification which Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and the member of Monera and Protista have cell wall have now been excluded from Plantae through earlier classification based them in same kingdom. So the cyanobacteria that also referred as blue-green algae are not algae anymore. In this chapter we'll describe algae, bryophyte, pteridophyte, gymnosperm and angiosperm under Plantae. Let us also look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habits, color, number and shapes of leaves, etc. They were based mainly on vegetative characters or on the endoecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system classification are equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As again, this natural classification system developed which were based on natural affinities among the organism and consider not only external feature but also internal feature like ultrastructure, anatomy, embryology and phytochemistry. Such a classification of flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxon have a common ancestor. We now use information from many other sources to help resolve difficulty in classification. These become more important when there is no supporting fossil evidences. Numeric taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Some are codes are assigned to all characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on the cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituent of plant. Resolve confusion are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thyroid autotrophic and largely aquatic, both freshwater and marine organisms. They occur in a variety of both other habitats, moist stone, soil, and wood. Some of them also occur in association with fungi, lichens, and animals on sloth bear. The form and size of algae is highly variable, ranging from colonial form like wallbox and filamentous form like Eleutheryx spirogyra. A few of marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being Jew spores. They are flagellated, motile, and germination gives rise to a new plant. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eleutheryx, and non-flagellated and non-motile but similar in size as in Spirogyra. So reproduction is called isogynous. Fusion of two gametes is similar in size and as in species of Eudorina is termed anisogamy. Fusion between one large and one motile static female gamete and smaller motile male gamete is termed oogamous. Example in wallbox and fucus. Algae, green algae, wallbox and Eleutheryx. Here is wallbox and here is Eleutheryx. Brown algae, laminaria, fucus, and dictyota. Laminaria, fucus, dictyota. Frond, stiff, hold fast. Ear bladder, frond, midrib, hold fast. Frond, stiff. Here is dictyota, funaria, laminaria. Laminaria, fucus, and dictyota. Red algae, porphyra, polysphonia. And polysphonia. Algae are useful in many variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are the paramount importance in primary producer of energy-rich compound which form the basis of food cycle of all aquatic animals. Many species of porphyra, laminaria, and sargassum are among 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water-holding substance, algin from brown algae and charagen from red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice cream and jellies. Chlorella, a unicellular algae rich in protein, are used in food supplement even in by space travelers. The algae are divided into three main classes: chlorophyces, pheophyce, and rhodophyce. 3.1.1 Chlorophyce. The member of chlorophyce are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in different chloroplasts. The chloroplast may be discoid plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of members have one or more storage body called pyrenoids located in their chloroplast. Pyrenoids contain protein beside starch. Some algae may store food in the form of oil droplet. Green algae usually have rigid cell wall made of inner layer cellulose and outer layer pectose. outer layer pectose, inner layer cellulose and outer layer pectose. Vegetative reproduction usually take place by fragmentation or by formation of different type of spores. Asexual reproduction is by flagellata spores produced by producing zoosporangia. The sexual reproduction shows considerable variation in type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Chyra. 3.1.2 Phaeophyce. The member of Phaeophyce brown algae are found primarily in the marine habitat. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched form are represented by scales, which may reach up to the height of 100 meters. They possess chlorophyll A, C carotenoids, and xanthophylls. They vary in color from olive green to various shades brown depending upon the amount of xanthophylls pigments fucoxanthin present in them. Food stores as complex carbohydrate which may be in the form of laminarin or mannitol. The vegetative cell have a cellulose wall usually covered on outside by gelatinous coating of algin. The protoplast contain, in addition to protoplast, are centrally located vacuole in the nucleus. The plant body is usually attached to substratum by a holdfast and has a stalk, the stipe, and the leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most of brown algae is by flagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. The sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form are ectocarpus, dictyota, laminarin, sargassum, and fucus. Rhodophyce. The member of Rhodophyce are commonly called red algae because the predominance of red pigment are phycoerythrin in their body. Majority of red algae are marine with their greater concentration found in the warmer area. They occur in both well-lighted region close to the surface of water and also in great depths of ocean where relatively little light penetrates. Red thalli of most of red algae are multicellular. Some of them have complex body organization. The body is told Floridian starch, which is very similar to amylopectin and glycogen in structure. Red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile. Table 3.1 Division of algae and their main characteristics. Classification. Common name, major pigment, food stores, cell wall, flagellate number, and position of insertion, habitat. Chlorophyce, green algae, chlorophyll A and B, starch present, cellulose 2 to 8, equal apical. Freshwater, brackish water, salt water. Phaeophyce, brown algae, chlorophyll A, C, B, fucoxanthin, mannitol, and laminarin, cellulose and algin, 2 unequal lateral flagella. Habitat fresh water, rare, brackish water, salt and water. Rhodophyce, red algae, chlorophyll A and D, phycoerythrin, Floridian starch, cellulose, pectin, and polysulfate ester. Cell wall made up of this absent flagella absent in Rhodophyce. Present in fresh water, some brackish water, salt water. Okay. Sexual reproduction is oogamous accompanied by complex post-fertilization development. Common example: Polysphonia, Porphyra, Gracilaria, and Gelidium. 3.2 Bryophyte. Bryophytes include the various mosses and liverwort that are found commonly in growing in moist, shaded areas in hills. Example of bryophyte: liverwort, mosses. In liver, Mertensia, female thallus and male thallus. Here is Mertensia, female thallus. and male thallus. Female thallus का arconiophore, gamma cup, and here is rhizoid in male markensia, and thetiophore, gamma cup, and here is rhizoid. Funaria, gametophytes and sporophyte. Here is funereal gametophyte and sporophyte. This is capsule, seta leaves, manaxia, and this is rhizoid. Is sphagnum gametophytes. This is gametophytic from plant body, and this is what? This is arcogonial branch, and this is antheridial branch. So 2025 में भाई लीक कर दे रहा हूँ, ये क्वेश्चन आएगा. एंथ्रीडियल ब्रांच एक डायग्राम देगा और क्वेश्चन भी देगा भाई. मैं लीक कर दे रहा हूँ यहाँ पे. सब्सक्राइब कर लेना तुम लोग जितने भी लोग, अगर आ गया तो 4 मार्क्स. ये डायग्राम आएगा और एक क्वेश्चन पूछेगा नीट 2025 में. Bryophytes are called amphibian of plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostrate on erect and attached to substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structure. The main plant's body or bryophytes is haploid. It produces gametes, hence is called gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated enterozoids. The female sex organ is known as archegonium. Is flask-shaped and produces a single egg. Antherozoids are released into water where they come in contact with archegonium. An antherozoid fuse with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not living but attached to the photosynthetic gametophyte and derive nourishment from it. Some cell of the gamete sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species sphagnum of moss provide peat that have long been used as fuel and packing of material for transshipment in living material because of their capacity to hold water. Mosses along with lichen are the first organism to colonize rock, hence are great ecological importance. They decompose rocks, making the substrate suitable for growth of higher plant. Since mosses form dense mats on soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are derived into liverworts mosses. 3.2.1 liverworts. The liverworts grow usually in moist, sandy habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of liverwort are thalloid and markensia. The thallus is dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in 2 rows of the stem-like structure. Asexual reproduction in liverwort takes place by fragmentation of thalli or by the formation of a specialized structure called gemma. Singular gemma. This is gemmae. This is gemma. भाई इसको gemma पढ़ेंगे gemma. Gemmae are green multicellular asexual pods which developed in small receptacles called gemma cup located on thalli. Gemmae become detached from parent body, germinate to form new individual. During sexual reproduction, the male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into food set and capsule. After the meiosis, spores are produced within the capsule. These spores germinate to form free living gametophyte. 3.2.2 Mosses. The predominant stage of life cycle of mosses in the gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is leafy stage, which develops from secondary protonema and lateral bud. They consist of a upright cylinder axis bearing spirally arranged leaves. They are attached to soil through multicellular branch rhizoids. This stage bears sex organs. Vegetative reproduction in mosses is by fragmentation and budding in their secondary protonema. In sexual reproduction, the sex organs Enteridia and Archegonia are produced at the apex of leafy shoots. After fertilization, the zygote develops into sporophyte consisting of a fruit, seta, and capsule. The sporophyte in mosses are more elaborate than that of liverwort. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses funaria, polytrichum, sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetail and fawns. Pteridophytes are used in medicinal purposes and soil wonders. They are also frequently grown as ornamentals. Evolutionarily, they are involved in first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The pteridophytes are found in cool, damp and sandy places, though some may flourish well in sandy soil conditions. You may recall that bryophytes, the dominant phase in the leaf-like life cycle, is the gametophytic plant body. However, in pteridophyte, the main plant body is sporophyte, which is differentiated into root, stem, and leaves. These organ possess well-differentiated vascular tissue. The leaf in pteridophyte are small microphyll as in Selaginella or large in microphyll as in fern. The sporophyte bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones, Selaginella or equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular pteridophytes. Selaginella, equisetum, fern, salvia. This is salvinia, fern, Selaginella, equisetum. So leaves, roots, stem, this is Selaginella, and this is equisetum, rhizome, branch, internal node, strobilus, and this is Salvinia. Freely, mostly photosynthetic thyroid gametophyte called prothallus. These gametophytes require cool, damp, sandy places to grow. Because of this specific requirement and their need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophytes bear male and female sex organs called Antheridia and Archegonia, respectively. What is required for transfer of enterozoite? The male gamete to release from Antheridia to mouth of the Archegonium. Fusion of male gamete with egg present in the Archegonium result in the formation of zygote. Zygote thereafter produced in multicellular, well-organized, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophyte, only spores are of similar kind. Such plants are called homospores. General like Selaginella and Salvinia, which produce two kinds of spore, macro large and micro small spores, are known as heterospores. The megaspore or microspore germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of zygote into young embryo take place within female gametophytes. This event is precursor to the seed habit, considered the important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Coelom, Lycopsida, Selaginella and Lycopodium, Synapsida, Equisetum, Pteropsida, Triopteris, Teres and Adiantum. 3.4 Gymnosperm. The gymnosperm sperm seed are plant in which the ovule are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seed that develop post fertilization are not covered, that is, are naked. Gymnosperm included in medium-sized tree and tall trees and shrubs. One gymnosperm, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproot. Roots in some genera have fungal association in the mycorrhiza finus, while in some other cycas, small specialized roots called coralloid root are associated with endotoxin cyanobacteria. The stems are unbranched cycas and branching finus and cedrus. The leaves may be simple or compound in cycles. The pinnate leaf persists for a few years. The leaves in gymnosperm are well adapted to withstand extreme temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help in reduce water loss. Gymnosperm are heterosporous. They produce haploid microspore and megaspores. The two kinds of spores are produced within sporangia are borne on sporophytes, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophyll and microsporangia are called microsporangiate or male strobili. The microspore develop into male gametophyte and generation, which is highly reduced and is confined to only limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called microsporangiate or female strobili. The male and female cones or strobili may be born on the same tree pinus. However, in cycas, male and cone females are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and composite structure is called an ovule. The ovule is born on megasporophytes, which may be clustered to form female cones. The megaspore mother cell divided meiotically to form 4 megaspore. The one megaspore enclosure within the megasporangium develop into multicellular female gametophyte that bear 2 or more archegonia or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm the male and female gametophyte do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with opening of the ovule borne on megasporophyte. The pollen tube carrying the male gametes grow towards archegonia in the ovules and discharge their content near the mouth of the archegonia. Following fertilization, zygote develop into embryo and ovules into seed. These seeds are not covered. Gymnosperm, cycas, pinus, ginkgo. Here is cycas and here is pinus, branched stem, and here is ginkgo, dwarf shoot. This is dwarf shoot, and this is long shoot, and this is seed. So angiosperm 3.5. Unlike gymnosperm, where ovules are naked, angiosperm or flowering plant, the pollen grain and ovules are developed in specialized structure called flowers. In angiosperm, the seeds are enclosed in a fruit. The angiosperm are exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest olivia to tall tree eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, the dicotyledons and monocotyledons. The dicotyledons are characterized by the seed having two cotyledons, reticulate venessa in leaves, and tetramerous, pentamerous flower, that is having 4 or 5 members of each floral whorls. The monocotyledons, on the other hand, are characterized by a single cotyledon, seeds, parallel venation in leaves, and trimerous flower having 3 members of each floral whorls. The male sex organs in flower are each stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, the pollen mother cell divide by meiosis to produce microspores, which mature into pollen grain. The female sex organ in the flower is a pistil. Pistil consists of swollen ovary at its base, a long slender style of style and stigma. Inside the ovary, ovule present. Generally, each ovary has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3-celled egg apparatus, 1 egg cell, 2 synergid, 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce diploid secondary nucleus. Pollen grains, after dispersal from anther, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains are made to form stigma, and the resulting pollen tube grows through the tissue of stigma and style and reach the ovule. The pollen tube enters the embryo sac where 2 male gametes are discharged. One of the 2 gametes fuse with egg cell syngamy to form zygote. The other gamete fuse with the diploid secondary nucleus to produce triploid primary endosperm nucleus (PEN). Because of the occurrence of 2 fusion, that is syngamy and triple fusion, this event termed as double fertilization, an event unique to angiosperm. The zygote develop into an embryo with one or 2 cotyledons, and PEN develop into endosperm, which provide nourishment to the developing embryo. The synergid and antipodal degenerate after fertilization. Occurring during this event, the ovules develop into seeds and ovaries develop into fruit. The life cycle of angiosperm. So, if you are 3.6, here is life cycle of angiosperm. Here is sporophyte, stigma, style, ovary, megasporangium, anther, filament, microsporangium. Microsporangium develop microspore. This microspore develop pollen grain. Microspore, gametophyte in generation, sporophyte to in generation. So here is female gametophyte, and here is female gamete, and here is male gamete. Male gametophyte, and here is gamete. After fusion, in this form zygote, and zygote develop into embryo. This latter we studied in class 12th, sexual reproduction in flowering plant in detail. So this is deleted from latest NCERT syllabus, so we are not going to cover this, but we are going to read summary. Plant kingdom including algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple cellulite, autotrophic, and largely aquatic organism depending on the type of pigment possessed and the type of stored food. Algae are classified into 3 classes, namely chlorophyceae, phaeophyceae, and rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexual formation, different type of spores sexually by formation of gametes, which may show isogamy, anisogamy, and oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is different and more differentiated than that of algae. It is thallus-like, prostate, and erect, attached to substratum by rhizoids. They possess root-like, leaf-like, and stem-like structure. The bryophytes are divided into liverwort and mosses. The plant body of bryophyte, liverwort, are thalloid and dorsiventral, where mosses are upright. This is very important. In liverwort, thalloid and dorsiventral. In mosses upright. Cylinder axis bearing spirally arranged leaves. The main plant body is bryophyte, is gamete producing, and is called a gametophyte. It bears the male sex organ called antheridia and female sex organ called archegonia. The male and female gamete fuse to form zygote, which produce a multicellular body called sporophyte. It produces a haploid spore. The spores germinate to form gametophytes. The pteridophyte, the main plant body is sporophyte, which differentiate into true root, stem, leaves. These organs possess well differentiated vascular tissue. The sporophyte wear sporangia, which produces spores. The spores germinate to form gametophyte, which require cool damp places to grow. The gametophyte wear male and female sex organ called antheridia and archegonia, respectively. What is required for transfer of male and female archegonia, where zygote formed after fertilization? The zygote produces a sporophyte. The gymnos are plant which ovule are not included by any ovary wall. After fertilization, seed remain exposed, and therefore the plant body can next seed plant. The gymnosperm produces microspore and megaspore, which are produced in microsporangia and megasporangia born on sporophylls. Sporophytes, microsporophytes, and megasporophytes are arranged spirally on axes to form male and female cones, respectively. The pollen grain germinate and pollen tube release male gamete into ovule, where it fuses with egg cell in archegonia. Following fertilization, the zygote develops into embryo and ovule into seed. In angiosperm, the male sex organ stamen and female sex organ pistil are born on flowers in a flower. Each stamen consists of filament and an anther. An anther produces a pollen grain male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is a female gametophyte or embryo sac which contains egg cell. The pollen tube enters into embryo sac, where two female gametes are discharged. One male gamete fuses with the egg cell, syngamy, and other fuses with diploid secondary nuclear triple fusion. This phenomenon of two fusion is called double fertilization and is unique to angiosperm. The angiosperms are divided into two classes, dicotyledon and monocotyledon. So, during the life cycle of sexual reproducing plant, there is alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant group well differentiated and may do different pattern life form haplontic, diploidic and intermediate. So, plant kingdom अपना reading complete हुआ. अरे bro, what this? Oh sorry भाई. क्या क्या हो रहा है भाई, क्या क्या हो रहा है. सो अपना ये कंप्लीट हुआ भाई. ये तो करना नहीं था. अपना प्लांट किंगडम एनजीओस्पर्म अपने करा और जिम्नोस्पर्म करा, ट्रेडोफाइट करा, मोसेस करा, लिवरवॉर्ट, ब्रायोफाइट, रोडोफाइसी, फियोफाइसी, क्लोरोफाइसी और एल्गी. प्लांट किंगडम ये सारे पार्ट कंप्लीट हुए. अब अपन करने जा रहे हैं एनिमल किंगडम. चैप्टर 4 करेंगे अपन अब एनिमल किंगडम भाई. So let's start bro. चैप्टर 4 एनिमल किंगडम। तो इसमें पहले बेसिक ऑफ क्लासिफिकेशन करेंगे, उसका क्लासिफिकेशन ऑफ एनिमल करेंगे। सो लेट्स स्टार्ट। Basic of classification. In spite of difference in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, pattern of digestive, circulatory, and reproductive system. These features are usually on the basis of animal classification, and some of them are discussed here. 4.1.1 Level of organization. Though all members of animal are multicellular, all of them do not exhibit the same pattern of organization of cell. For example, in sponges, the cells are arranged in loose cell aggregate that they exhibit cellular level of organization. Some division of labor activities occur among the cell cylindrids. The arrangement of cells is more complex. Here, the cell performing the same function are arranged into tissue. Hence, is called tissue level of organization. A still higher level of organization, that is organ level of, is exhibited by members of platyhelminthes and other higher phyla, where tissue are grouped together to form organs. Each is specialized for particular function. In animals like annelids, arthropods, and mollusks, echinoderms and chordates, organ have associated to form functional system. Each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ system is in different group of animal exhibit various patterns of complexity. For example, the digestive system in platyhelminthes has a single opening to the outside of the body that serves as opening mouth and anus, and is hence called incomplete. A complete digestive system has two opening mouth and anus. Similarly, the circulatory system may be two types. First, open type, in which blood is pumped out of the heart and the cell and the tissue directly brought into it. Second, closed type, in which the body is circulated throughout serous vessels varying diameters, artery veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical. Any plane passing through the center does not divide animals into equal halves. When any plane passing through the center of axis of animal divides the organism into two identical halves, it is called radial symmetry. Ctenophore and echinoderms have this kind of plane. Animals like annelid, arthropod, etc., where the body can be divided into two identical left and right halves in one plane, exhibit bilateral symmetry. Here is bilateral symmetry and here is radial symmetry. 4.1 Diploblastic and triploblastic organization. Animals in which the cells are arranged into two embryonic layers, an external ectoderm and internal endoderm, are called diploblastic animals. Cylindrids. An undifferentiated layer, mesoglea, is present between the ectoderm and endoderm. Those animals which developing broad germinal layer, mesoderm, in between ectoderm and endoderm, is called triploblastic animal. There are plenty elements to call that. Showing germinal layer, diploblastic and triploblastic. Here mesoderm, ectoderm, endoderm, and here mesoglea present in diploblastic animals. Triploblastic are platyhelminthes to chordate. 4.1.1 Coelom. Diagrammatic sectional view of coelomate, pseudocoelomate and acoelomate. Coelom present in coelomate, pseudocoelomate, and here is acoelomate. Coelom. Presence or absence of cavity between the body wall and gut wall is very important in classification. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates, that is annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. In some animals, the body cavity is not lined by mesoderm. Instead of mesoderm, it is present as a scattered pouch between ectoderm and endoderm. Such body cavity is called pseudocoelom, and animal possessing them is called pseudocoelomates, that is aschelminthes. The animals in which body cavity is absent is called acoelomate, platyhelminthes. 4.1.5 Segmentation. In some animals, the body is externally and internally divided into segments with serial repetition at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and phenomena is known as metamerism. 4.1.6 Notochord. Notochord is mesodermally derived rod-like structure formed on the dorsal side during embryonic development. In some animals with notochord is called chordate, and those animals which do not form this structure is called non-chordate. Example: porifera and echinoderms. 4.2 Classification of animals. The broad classification of animalia based on common fundamental features mentioned in preceding section given in figure 4.4. Kingdom. Animalia multicellular. Level of organization: cellular symmetry mostly asymmetrical. Body cavity: coelom, acoelomate. Example: porifera. And multicellular: tissue level, organ level, or organ system level. Here is radial and bilateral. Radial में acoelomate, coelenterata, cnidaria, and ctenophora. Tissue में bilateral में without body cavity acoelomate, platyhelminthes. With false coelom, pseudocoelom, aschelminthes. With true coelom, coelomates. Annelida, arthropoda, mollusca, echinodermata, hemichorda, chordata. Echinodermata exhibit radial bilateral symmetry depending on its figure. Broad classification of animal kingdom: Animalia based on common fundamental features. The important characteristic features of these different phyla are described. 4.2.1 Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport canal system. Water enters through minute pores, ostia, in the body wall into central cavity, spongocoel, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Choanocytes or collar cell line spongocoel and the canal. Digestion is intracellular. The body is supported by a skeleton made of spicules or spongin fiber. Sexes are not separate hermaphrodite, that is, egg and sperm are produced by same individual. Sponges are reproduced asexually by fragmentation and sexually by formation of gametes. Fertilization is internal, development is indirect, having larval stage which morphological distinct from adult. Example of porifera: Sycon, Spongilla. Example: Sycon, also known as Scypha. Spongilla, common in freshwater sponge. Spongia, common in bath sponge. 4.2.2 Phylum Coelenterata or Cnidaria. They are aquatic, mostly marine, sessile, and free-living, radially symmetrical animals. The name cnidarians are derived from cnidoblasts and cnidocytes, which contain stinging capsule or nematocysts present on the tentacles of the body. Cnidoblasts are used for increased defense and for capture of prey. Cnidarians exhibit tissue level of organization and are diploblastic. They have central gastrovascular cavity with single opening mouth called a hypostome. Digestion is extracellular and intracellular. Some of cnidarians, that is corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms, polyps and medusa. The former is sessile and cylindrical and form like hydra, adamsia, etc., where the latter umbrella shaped with swimming like Aurelia and jellyfish. Those cnidarians which exist both form of exhibit alternation of generation metagenesis, that is polyp produce medusa asexually and medusa form polyp sexually, that is present in ovalia. Here is outline diagram for Aurelia, medusa, and polyp adamsia. So example of Example: Physalia podca is metaphor, Adamsia sea anemone, Peninsula sea fawn, Gorgonia sea fan, Amandra brown claw. Here shape of Nidolast. So Adamsia, Hydra and Asia are the silent cynical form of Hydra. To 4.2.3 phylum Dinofauna. Dinofauna commonly known as sea walnut or comb jelly are exclusively marine, radially symmetrical and diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated complets which help in locomotion. Digestion is both extracellular and intracellular bioluminescence deeper body of living organism to emit light well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Example: Pleurovachia and Ctenoplana. Here is a diagram of Ctenophora Pleurovachia. 4.2.4 Phylum Platyhelminthes. They have dorsoventral flattened body, hence are called flatworms. These are mostly endoparasites found in animals, including human beings. Flatworms are bilateral, symmetrical, triploblastic and acellular animals with organ level of organization. Hooks and suckers are present in the parasitic form. Some of them absorb nutrients from the host directly through their body surface. Specialized cell called flame cell help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and developed into through many larval stages. Some members like Planaria possess high regeneration capacity. Examples are Taenia, tapeworm, fasciola, liverflow. Here is Platyhelminthes, tapeworm and liverflow. 4.2.5 Phylum Escalmenthes. The body of Escalmenthes is circular in cross section, hence the name roundworm. They may be free living, aquatic and terrestrial, parasitic in plants and animals. Roundworms have organ system level of body organization, and they are bilateral, symmetrical, triploblastic and pseudosolvent animals. Alimentary canal is complete with well-developed musculopharynx and excretory tube. Remove body waste from body cavity through the excretory pores. Sexes are separate, dioecious, that is, male and female sex are distinct. Other females are long than males. Often females are long than men. Fertilization is internal development, may be direct or indirect. Example: Ascaris roundworm, Augeria, filarial worm, and Cyclostoma hookworm. 4.2.6 Phylum Annelida. They may be aquatic, marine or freshwater or terrestrial, free living and sometimes parasitic. They exhibit organ system level of organization of body organization and bilateral symmetry. They are triploblastic, metamerically segmented and coelomate animals. Their body surface is distinctly marked out into segment or metamers and is hence the phylum name Annelida, Latin endless little ring. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereis possess lateral appendages, parapodia, which help in swimming. A closed circulatory system is present. Nephridia, singular nephridium, help in osmoregulation and excretion. Neural system consists of paired ganglion connected by lateral nerve to double ventral nerve cord. Nereis and aquatic form is dioecious, but earthworm and leeches are monoecious. The reproduction is sexual. Example: Nereis ferretum, earthworm, and Hirudinea blood-seeking leech. Here is Annelida, Nereis, and Hirudinea. 4.2.7 Phylum Arthropoda. This is the largest phylum of Animalia, which includes insects. Over 2/3 of all animal species on Earth are arthropods. They have organ system level organization. They are bilateral, symmetrical, triploblastic, segmented, and coelomate animals. The body of arthropod is covered by chitinous exoskeleton. The body consists of head, thorax, and abdomen. They are jointed appendages, organ or through joint podal appendages. Respiratory organ are gills, book lungs, or tracheal system. Circulatory system consists of open type sensory organs like antenna, eye, compound eye, and simple. Statocyst or balancing organ are present. Excretion take place through Malpighian tubules. They are mostly dioecious. Fertilization is usually internal. They are mostly oviparous. Development may be direct or indirect. Example: economically important insect: Apis, honeybee, bombyx, silkworm, lattice, lacifer, lag insect. Vectors: Anopheles, culex, and Aedes mosquitoes. Gregarious fish: locust, living fossil, limbless king crab. Here example of Latinoid arthropods: locust, butterfly, scorpion, prawn. 4.2.8 Phylum Mollusca. This second largest animal phylum. Mollusca are terrestrial, aquatic, marine, freshwater, having organ system level of body organization. They are bilateral, symmetrical, and triploblastic and coelomate animals. Body is covered by a calcareous cell and is unsegmented with distinct head, muscular foot, and vascular visceral hump. A soft and spongy layer of skin form a mantle over the visceral hump. The space between the hump and the mantle is called mantle cavity, in which feather-like gills are present. They have respiratory and excretory function. The anterior head region has sensory tentacles. The mouth contains file-like grasping organs for feeding called radula. Here is example of mollusca: Pila and octopus. They are usually dioecious and oviparous with indirect development. Example: Pila, Appanation, Pinctada, Paloestra, Sepia, cuttlefish, Loligo, squid, octopus, devilfish, Aplysia, sea hare, Dentalium, tusked, and Cutopelura chiton. 4.2.9 Echinodermata. These animals have endoscleral calcareous ossicles and hence the name Echinodermata. Spiny body figure. All the marine animals organ system level of body organization. The adult Echinodermata radial symmetrical but larva are lateral symmetrical. They are triploblastic and coelomate animals. Digestive system is complete with mouth on lower ventral side and anus on upper side dorsal side. The most distinctive feature of echinoderms is the presence of water vascular system, which help in the locomotion, capture, and transport of food and respiration. An excretory system is absent. Sexes are separated. Reproduction is sexual. Fertilization is usually external. Development is indirect with free living larvae. Example: Asteris, starfish, kind sea urchin, Antenna, sea lily, and Cucumeria, sea cucumber, Ophira, Vita star. 4.2.10 Phylum Hemichordata. Here is example: Asteris and Ophira. Phylum Hemichordata. Hemichordata is earlier considered a subphylum under phylum Chordata, but now it is considered a separate phylum under non-Chordata. Hemichordata have a rudimentary structure in collar region called stromochord. The structure similar to notochord. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic, and coelomate animals. The body is cylindrical and composed of an anterior proboscis, collar, and trunk. Circulatory system is open type. Respiration takes through gills. Excretory organ is proboscis gland. Sexes are separate. Fertilization is external. Development indirect. Example: Blanoglosson and Sacoglosson. 4.2.11 Chordata. Animal belonging to phylum Chordata is fundamentally characterized by presence of notochord or dorsal, hollow nerve cord, and paired pharyngeal gill slit. These are bilaterally symmetrical, triploblastic, coelomate with organ system level of body organization. They possess a postnasal tail and closed circulatory system. Here nerve cord and here notochord. Postnasal tail, gill slit. Chordata characteristics. Comparison of chordata and notochord. Chordates and non-chordates. Notochord present. Central nervous system is dorsal and hollow single. Pharynx portion is gill slit. Heart is ventral. Postnasal part of tail is present. Notochord is absent. Central nervous system ventral solid double. Gill slit absent. Heart is dorsal if present. Post-anal tail is absent. Phylum Chordata is divided into two subphylum: Urochordata and Tunicata. Cephalochordata and Tuberata is subphylum. Urochordata and Cephalochordata are often referred to as protochordates, and the exclusive marine and Urochordata notochord is present in between the larval tail, while in Cephalochordata it extends from head to tail region and is persistent throughout their life. Example: Urochordata, Acidia alpha, Dolium, Cephalochordata, Branchiostoma, and Amphioxus or lancelet. The members of subphylum Vertebrata possess notochord during the embryonic period The notochord replaced by a cartilaginous bony vertebral column in the adult. Thus, all vertebrates are chordate, but all chordates are not vertebrate. Besides the basic chordate character, the vertebrates have a ventral muscular heart with 2 or 3 or 4 chambers, kidney for excretion and osmoregulation, and paired appendage which may be fins and limbs. Subphylum Vertebrata further divided in following parts. Vertebrata division: Agnatha, leg jaw, and Gnathostomata, bear jaw. Agnatha contains cyclostomata, and Gnathostomata contains two superclasses: feces, bear fins, and tetrapoda, bear limbs. So feces contain class Chondrosis and Osteichthyes, and tetrapoda consist amphibia, reptilia, and avian mammals. 4.2.11.1 Class Cyclostomata. All living members of Cyclostomata are ectoparasite and some species. They have an elongated body bearing 6 to 15 pair of gill slits for respiration. Cyclostomes have a sucking and circular mouth without jaw. Their body is divided of scale and paired fins. Cranium and vertebral column are cartilaginous. Circulation is a closed type. Cyclostomes are marine but migrate for spawning to fresh water. After spawning within a few days, they died. Their larvae, after metamorphosis, return to the ocean. Example: Petromyzon lamprey and magazine hagfish. 4.2.11.2 Class Chondrichthyes. Here Jales word will Petromyzon. Chondrichthyes, they are marine animals with streamlined body and have cartilaginous endoskeleton. Figure 4.19. Here is cartilaginous fish, Scoliodon, Prestige, Sawfish. Mouth elongated ventrally, notochord is persistent throughout their life. Gill slits are separated without operculum or gill cover. The skin is tough and containing minor placoid scale. Teeth are modified placoid scale which are backwardly directed, and their jaws are very powerful. These animals are predaceous. Due to absence of air bladder, they are constantly swimming in water to avoid sinking. Heart is 2 chambered and 1 auricle and 1 ventricle. Some of them are electric organ in torpedo, and some possess poison sting trigon. They are cold-blooded poikilothermous animals, that is, they lack the capacity to regulate their body temperature. Their sexes are separate, and males and pelvic fins bear claspers. In males, pelvic fins bear claspers. They have internal fertilization, and many of them are viviparous. Example: Oscilion, many of them are viviparous. Here very important. Scolion, dogfish, Prestige, Sawfish, Carcharodon, great white shark, trigon, stingray. 2.1.11.3 Class Osteichthyes. It includes both marine and freshwater fish with bony endoskeleton. Their body is streamlined, mouth is mostly terminal, and they have 4 pair of gill slits which are covered by operculum on each side of his skin and covered with cycloactinoid scale. Air bladder is present which regulates the buoyancy. Heart is 2 chambered, 1 auricle and 1 ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Example of bony fish is Hippocampus catla. Example: marine, Exocetus flying fish, Hippocampus seahorse. Freshwater, Labiorohu, Catla, Calarius maur, aquarium, beta or fighting fish, Pterophyllum, angelfish. 4.2.11.4 Class Amphibia. The name indicated amphi, dual, bios, life. Amphibia can live in aquatic as well as terrestrial habitat. Most of them have 2 pair of limbs. Body divisible into head and trunk. Tail may be present in some. The amphibian skin is moist without scale. The eyes have eyelids. A tympanum represents the ear. Alimentary canal, urinary and reproductive tract are open into common chamber called cloaca, which open to exterior. Respiration is by gills, lungs, and through skins. The heart is three chambered, two auricle and one ventricle. These are cold-blooded animals. Sexes are separate. Fertilization is external. They are oviparous and development is indirect. Examples: bufo, toad, rana, frog, hyla, tree frog, salamander, ichthyophos, limbless amphibia. Here is example of amphibia, salamander and rana. Here is example of reptiles. 4.2.11.5 Reptilia. The class name refers to their creeping and crawling mode of locomotion. Latin reaper or reptum, to creep and crawl. They are mostly terrestrial animals and their body is covered by dry and conified skin, epidermal scales or scutes. They do not have external ear opening. Tympana represent ear. Limbs when present are two pairs. Heart is usually three chambered but four chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales and skin cards. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Example: chelone, turtle, testudo, tortoise, chameleon, tree lizard, calotus, garden lizard, crocodilus, crocodile, alligator, hemidactylus, wall lizard, poisonous snake, naja, cobra, bankerus, carrot, vipera, viper. Here is chameleon, crocodile, chelone and naja. 4.2.11.6 Class Aves. The characteristic feature of Aves birds are presence of feather. Most of them are fly except flightless bird ostrich. They possess beak. The following modified into wing. The hind wings generally have scales are modified for walking, swimming and clasping. The tree's branches is skin dry without glands except the oil and the bases of tail. Endoskeleton is fully ossified, bony and long. Bones are hollow and with air cavity pneumatic. The digestive tract of birds has additional chamber. The crop and gizzard. The heart is completely four chambered. They are warm-blooded homoiothermous animals. That is, they are able to maintain constant body temperature. Respiration is by lungs. Their air sac connected to the lungs supplement respiration. Ear sac connected to lungs supplement respiration. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Example: corvus crow, columba pigeon, Cetacula parrots, Stuthio, ostrich, peacock, Apernodactylus penguin, neophron vulture. Here is some birds: neophron, Struthio, Cetacula, pavo. 4.2.11.7 Class Mammalia. They are found in a variety of habitats: polar ice cap, deserts, mountains, forests, grasslands and dark caves. Some of them have adapted to fly or live in water. The most unique mammalian characteristic is the presence of milk producing gland, mammary gland, by which the young one nourishes. They have two pairs of limbs adapted to walking, running and climbing, burrowing, swimming and flying. The skin of mammals are unique, possessing hair. External ears are pinna present. Different type of teeth present in the jaw. Heart is four chambered. They are homoiothermous. Respiration is by lungs. Sexes are separate and fertilization is internal. They are oviparous with few exceptions and development is direct. Example: oviparous Ornithorhynchus, platypus; viviparous Macropus, kangaroo; pteropus, flying fox; camelus, camel; macaca, monkey; ratus, rat; canis, dog; felis, cat; elephas, elephant; equus, horse; delphus, common dolphin; Blenoptera, blue whale; Panthera tigris, tiger; Panthera leo, lion. The silent distinguishing feature of all phyla under animal kingdom comprehensively given in table 4.2. Silent feature of different phylum of animalian kingdom. Here are phylum: level of organization, symmetry, coelom, segmentation, digestive, circulatory, respiratory, distinctive feature. तो इसको वही जो पढ़े हैं उसकी का रिवीजन है. ठीक, तो एक कॉमन कैरेक्टर देख लेते हैं, डिस्टिंक्टिव फीचर. तो पोरीफेरा का क्या होता है? बॉडी विथ पोर कार्ड कैनाल सिस्टम, सिलेंड्रेटम एंड निडोब्लास्ट, टिनोफोरम कॉम्प्लेक्स, फ्लैटियल में फ्लैट बॉडी एंड सकर्स, Scale means worm like shape, elongated, annelida body ring like structure, merism शो कर रहा है. आर्थ्रोपोडा जो स्केलेटन ज्वाइंट अपेंडेज. मोलस्का एक्सटर्नल स्केलेटन सेल यूजुअली प्रेजेंट. एकाइनोडर्म वाटर वास्कुलर सिस्टम रेडियल सिमेट्री. हेमिकोडाटा वर्म लाइक विथ प्रोबोजिक कोलेंट्रम. कोडाटा नोटोकॉर्ड डोर्सल हॉलो एंड नर्व कॉर्ड गिल स्लिट विथ लिम्स. समरी इसका कर लेते हैं. Basic fundamental feature of such organization, symmetry, cell organization, phylum segmentation, notochord, etc. have enabled us broadly classify animal kingdom. Beside the fundamental feature of these, many other distinctive characteristics which specific for each phyle or class. Porifera include multicellular animals which exhibit cellular level of organization and have characteristic flagellators, conocytes. These cylindrids have tentacles and bear nidoblast. They are mostly aquatic, sessile, and free-floating. The ctenophores are marine animals with complex ability elements. They have a flat body, exhibit bilateral symmetry. The parasitic form so distinct with sucker and hooks. Skin length is pseudocellular, include parasitic as well as non-parasitic roundworm. Annelids are metamerically segmented animals with true pseudome. The arthropoda are mostly abundant group of animals characterized by the presence of joint appendages. The mollusks are soft bodies surrounded by external calcareous cell. The body is covered by external skeleton made of chitin. The echinoderm possesses spiny skin. Their most distinct feature is presence of vascular system. The hemichordates are a small group of worm-like animals. They are cylindrical body, proboscis, and coelentrum. Phylum Chordata include animals which possess a notochord either throughout or during early embryonic life. Other common feature observed in the chordates are dorsal hollow and nerve cord, paired pharyngeal gill slits. Some of vertebrates do not possess jaw agnatha, whereas most of them possess jaw gnathoistomata. Gnatha are represented by the class Cycloistomata. They are most primitive chordates and are ectoparasites on fishes. Gnathoistomata have two superclasses, the fishes and tetrapoda. Class Condylithis and Osteothis bear fins for locomotion and are grouped in the fishes. The Condylithis are fishes with cartilaginous endoskeleton and are marine. Classes Amphibia, Reptilia, Aves, and Mammalia have two pair of limbs and thus grouped under tetrapoda. The amphibia have adapted to live under both land and water. Reptiles are characterized by the presence of dry and conified limbs. Limbs are absent in snakes, fishes, amphibia, reptiles, poikilotherms, cold-blooded apes, and warm-blooded animals with feathers, and their bodies are forelimbs modified into wings for flying. Hind limbs are adapted to walking, swimming, perching, clasping. The unique feature of mammalians are presence of mammary gland and hair in skin. They commonly exhibit viviparity. So एनिमल किंगडम अपना खत्म हुआ भाई. ठीक, ये कुछ एग्जांपल है इसका. वो भी पैरा और निथोराइंगस आएगा. टेरोपस फ्लाइंग फॉक्स भी इसका एग्जांपल है. ये है टेरोपस. विविपैरस होता है क्या है, बैट. ब्लाइनोपेटेरा भी इसी का एग्जांपल मैमल्स का. ठीक, ये कुछ याद कर लिए. ठीक, ये कुछ देख लो, सब रट्टा मार जाओ इसको. और क्या ही है, ये रहा अपना एनिमल किंगडम में ख़त्म हुआ. तो भाई हम लोग 2 बजे से मतलब कि 5 मिनट का थोड़ा स्टार्ट करेंगे यूनिट 2. ठीक, फ्रॉम देखो 5 मिनट ब्रेक. इसके बाद 2 AM से स्टार्ट करा जाएगा क्या, स्ट्रक्चरल ऑर्गेनाइजेशन इन प्लांट्स एंड एनिमल. इसमें आएगा चैप्टर 6 मॉर्फोलॉजी इन फ्लावरिंग प्लांट, चैप्टर 6 एनाटॉमी इन फ्लावरिंग प्लांट, चैप्टर 7 स्ट्रक्चरल ऑर्गेनाइजेशन इन एनिमल. इसका सेकेंडरी ग्रोथ नहीं करेंगे इसको, क्योंकि वो डिलीट कर देगा. तो भाई 2 बजे से स्टार्ट करते हैं इसको, और कोशिश करेंगे 2 बजे से लेके इन तीनों चैप्टर को निपटा दें. 2-3 मुश्किल तो 5 बजे के अंदर इन सबको निपटा देना. ठीक, ख़त्म कर देना भाई. लग रहा है आज 12 बजने वाला है यार सुबह का. लेट्स सी क्या होता है, करते हैं, करेंगे. 2 बजे तक आते हैं भाई, थोड़ा रेस्ट कर लो. सो आई एम बैक गाइस। तो भाई 2 बज गया, चलो स्टार्ट करते हैं अपना स्ट्रक्चरल ऑर्गेनाइजेशन इन प्लांट्स एंड एनिमल। तो कैसे हो भाई लोग, रेडी? गाड़ी की सीट बेल्ट बांध लीजिए, क्योंकि हम लोग स्पीड को अपना थोड़ा इंक्रीज करेंगे, नहीं तो इस जन्म में तो नहीं होने वाले हैं भाई। लेट्स गो ब्रो, लेट्स सी व्हाट हैपेंस। कुछ करके ही जाएंगे भाई, मरने से पहले। जन्म लिए हैं। So guys, let's start structural organization in animals. So let's the description of diverse form of life on Earth was earlier made only by observation through naked eyes or later through magnifying lenses, the microscope. The discussion is mainly gross structural features, both external and internal. In addition, observable and perceivable living phenomena were also recorded as part of this description. Before experimental biology, or more specifically physiology, was established as a part of biology, naturalists described only biology. Hence, the biology remained as natural history for a long time. The description by itself was amazing in terms of details. While the initial reactions of a student could be boredom, one should keep in mind that the detailed descriptions were utilized in the later day reductionist biology, where living processes drew more attention from the scientists than the description of life forms and their structure. Hence, this description become meaningful and helpful in framing research questions in physiology and evolutionary biology. In the following chapter in this unit, the structure organization of plants and animals, including the structure basic of physiology or behavioral phenomena, is described. For convenience, this description or morphologically and anatomically feature presented separately for plants and animals. Catherine Izzo was born in Ukraine in 1898. She studied agriculture in Russia and Germany, received her doctorate in 1931 in United States. She reported in her early publications that the curly top virus is spread through a plant by the way the food conducting or phloem tissue. Dr. Izzo Plant Anatomy, published in 1954, took a dynamic developmental approach designed to enhance one's understanding of plant structure and an enormous impact worldwide, literally bringing about a revival of discipline. The Anatomy of Seed Plant by Catherine Izzo was published in 1960. It was referred to as Webster of Plant Biology. It is encyclopedic. In 1957, she was elected to the National Academy of Science, becoming the 6th woman to receive that honor. In addition to this prestigious award, she received the National Medal Science from President George Bush in 1989. When Catherine Izzo died in the year 1997, Peter Raven, director of anatomy and morphology, Missouri Botanical Garden, remembered that she absolutely dominated the field of plant biology even at the age of 99. Chapter 5, Morphology of flowering plant. इसमें पढ़ेंगे रूट, स्टेम, लीफ, इन्फ्लोरेसेंस, द फ्लावर, फ्रूट, सीड, सेमी technical description of flowering plant, description of some important families. The wide ranges of structure of higher plants will never fail us to fascinate us, even though the enjoys from such large diversity. External structure morphology, they are characterized by presence of root, stem, leaves, and flowers. Chapter 2 and 3 we talked about classification of plants based on morphology and other characteristics. For any successful attempt at classification and at understanding any higher plant, or for that matter any living organism, we need to know that standard technical terms and standard definition. We also need to know about the possible variation and different part found as adaptation of plant to their environment, that is adaptation to various habitat for production, climbing, and storage. If you pull out any weed, you will see that all of them have roots, stem, and leaves. They may be bearing flowers and fruits. The underground part of flowering plants is the root system, while the portion above the ground form our shoot system. Figure 5.1, the root. In majority of dicotyledonous plants, the direct elongation of radicle led to the formation of primary root, which grow inside the soil. It bears lateral roots, several orders that are referred to as secondary, tertiary, etc. roots. The primary roots is branch constitute the taproot system as seen in mustard plant. The monocotyledonous plant primary root is short-lived and replaced by large number of roots. These roots originate from the base of the stem, constitute the fibrous root system as seen in the wheat plant. In some plants like grass, monstera, and banyan tree, root arises from parts other than the radicle and called adventitious root. The main function of root system is absorption of water and minerals from the soil, providing proper anchorage of plant part, storing reserve food material, and synthesis of plant growth regulators. Here is flower, fruit, stem, leaf, shoot system, and here is root system, primary root, and here is secondary root, bud, node, internode. Type of roots: taproot, fibrous, and adventitious root, fibrous root, and here is taproot. Lateral, main root. Region of roots: region of maturation, root hair, region of elongations, and here is root cap, region of meristematic activity, the region of root cap. The root is covered at apex by a thimble-like structure called root cap. By a root cap, it protects the tender apex of root as it makes its way through the soil. A few millimeters from the root cap is a region of meristematic activity. The cell of this region is very small, thin-walled, and with dense protoplasm. They divided rapidly. The cell proximal to this region undergo rapid elongation and enlargement and is responsible for growth of root in length. This region is called the region of elongation. This cell of elongation zone gradually differentiate and mature. Hence, this zone proximal to the region of elongation is called region of maturation. From this region, some of epidermal cells form very fine and delicate thread-like structure called root hair. These root hairs absorb water and minerals from the soil. 5.1.2 Modification of root. Modification of root to support banyan tree. Root of some plants change in their shape and structure, become modified for function other than absorption, conduction, and water mineral. They are modified to support storage of food, respiration. The taproot of carrot and adventitious root of sweet potato get swollen and store food. You can give some more such examples. Have you ever wondered what those hanging structures that support a banyan tree? These are called prop roots, similar to the stem mash and sugar can have supportive root coming out other lower nodes and stem. These are called stilt root. In some plants such as rhizophora growing swampy area, many roots come out of the ground and grow vertically upwards. Such roots are called pneumatophora. Hence, help to get oxygen for respiration. Modification of root: storage, asparagus, turnip, carrot, sweet potato, and respiration pneumatophora in rhizophora. 5.2 stem. What are the features that distinguish the stem from the root? The stem is the ascending part of axis bearing branches, leaf, flower, and fruit. It develops from the plumule of embryo of germinating seed. The stem bears nodes and internode. The region of stem where the leaves are born called nodes, while internode are portion between the two nodes. The stem bears buds, which may be terminal. Axillary stem are generally green when young, later often become woody and dark brown. The main function of stem is spreading out the branches, bigger leaves, flowers are fruit. It conduct water, mineral, and photosynthesis. Some stem perform a function of storage of food, support and production, and vegetative propagation. 5.2.1 Modification of stem. The stem may not always be typically like what they are expected to be. They are modified to perform different functions. Example, figure 5.6. Underground stem of potato, turmeric, jamaican, calacasia are modified to store food in them. They also act organs perennial to tide over conditions unfavorable for growth. Stem tendrils which develop from axillary but slender spiral required help in plants to climb, such as in gourds, cucumber, pumpkin, watermelon, and grapevines. Axillary buds of stem may also be get modified into woody, straight, and pointed thorns. Thorns are found in many plants, such as bougainvillea. They protect plants from browsing animals. Some plants are arid region modify their stem into flattened opuntia or freshly cylindrical euphorbia structure. They contain chlorophyll and carry out photosynthesis. Underground stem of such grass, strawberry, etc. New when mother died, new plants form. In plants like mint and jasmine, slender lateral branch arise base man growing aerially for some time arc downward to touch the ground. A lateral branch with short internode. Each node bears rosette of flower and top root is found in aquatic plant fish, Tianicornia, in banana, pineapple, and chrysanthemum. The lateral branches originate from the basal underground portion of stem, grow horizontally beneath the soil, and come out obliquely upward. गिव राइट तो ये इसको मैं ही पढ़वाई क्योंकि हट गया ना. जितने भी मॉडिफिकेशन एनसीआर से हट गए उसको हटा ही दो. लीफ द लीफ लैटरल फ्लैट और स्ट्रक्चर बोन ऑन स्टेम इन डेवलप टू एट द नोड एंड बियर अ बर्ड इन इट्स एक्सियल. The axillary bud later develop into branches. Leaves originate from shoot apical meristem and are arranged in acropetal order. They are most important vegetative organ for photosynthesis. A typical leaf consists of three main parts: leaf base, petiole, and lamina. The leaves are attached to stem by leaf base and may bear two lateral small leaf-like structure called stipule. The monocotyledon, the leaf base expand into a sheath covering the stem partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called pulvinous. The petiole helps hold the blade to light. Long, thin, flexible petiole along leaf blade are floated in wind, thereby cooling the leaf and bringing fresh air to leaf. The lamina or the leaf blade in the green expanded part of leaf with veins and veinlet. There is usually middle prominent vein which known as midrib. Veins provide rigidity to leaf blade and acts as a channel of transport of water, minerals, and food material. The shape, margin, apex, surface, and extend to incision lamina vary in different leaves. Venation. Here is axillary bud, petiole, leaf base, stipule, and here is lamina. Venation. Arrangement of leaf, arrangement of veins and veinlet in lamina of leaf terminus venation. When the veinlet form a network of venation is termed as reticulate. When the veins run parallel to each other within the lamina, the venation is called parallel. Leaves of dicotyledon plants generally possess reticulate venation, while parallel venation is the characteristic of most monocotyledons. Structure of leaf: part of leaf B reticulate venation, and here is part of leaf. Here is reticulate venation, and here is parallel venation. Type of leaves: a leaf set with simple when its lamina is entire or when incised. The incision do not touch the midrib. When the incision of lamina reach up the midrib, breaking into the number of leaflets, the leaf is called compound. A bud is present in the axilla petiole in both simple and compound leaves, but not in the axilla leaflet of the compound leaf. The compound leaf may be of two types. First, infinitely compound leaf. A number of leaflets are present on the common axis. The branches, which represent the midrib of the leaf, as neem. Compound leaf, neem. Infinitely compound and palmately compound, silk cotton. Palmately compound leaf. The leaflet are attached to common point at petiole in silkworm. 5.3.3 Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem or branch. This is usually three types: alternate, opposite, and hold. In alternate type of phyllotaxy, a single leaf arises at each node alternate manner, as in China rose, mustard, and sunflower plants. In opposite type, a pair of leaves arise at each node and lie opposite to each other, as in calotropis and guava plants. If more than two leaves arise at a node and form a whole, it is called hold, as in Alstonia. Here, different type of phyllotaxy: alternate in China rose, opposite in guava, and hold in Alstonia. So here is modification of leaves. 5.3.4. Leaves are often modified to perform function other than for photosynthesis. They are converted into tendrils for climbing, as in pea, or into spines for defense, as in cacti. The fleshy leaves of onion and garlic store food. In some plants, such as Australian acacia, the leaves are small and short-lived. The petiole in these plants expand, become green, and synthesize food. Leaves of certain insectivorous plants, such as pitcher plant, Venus flytrap, are also modified leaves. Modification of leaves: here is to support. This is tendril. And protection: for protection, leaves modified into spines, and for storage, fleshy leaves. That is, this is present in onions. 5.4. Inflorescence. A flower is modified shoot wherein the shoot apical meristem change into floral meristem. Internode do not elongate and axis get condensed. The apex produce different kinds of floral appendage laterally at the successive node instead of leaves. Where a shoot tip transforms into flower, it always solitary. The arrangement of flower on the floral axis is known as inflorescence. Depending on whether the apex gets developed into flower or continues to grow, two major types of inflorescence are defined: racemose and cymose. In racemose type of inflorescence, the main axis continues to grow. The flowers borne laterally in acropetal succession. In cymose type of inflorescence, the main axis terminates to flower, hence limited in growth. The flowers are borne on basipetal order. So, in cymose type of inflorescence, main axis terminate and limited its growth. The flower is borne in known as basipetal order. Why cymose का क्या basipetal order में हो रहा है, याद करने की जरूरत है. देखो, रेसमोज एक्रोपिटल, साइमोज बेसिपिटल. CBRA रेसमोज inflorescent, and here is cymose inflorescence. 5. The flower, most important. The flower, the reproductive unit of an angiosperm, is meant for sexual reproduction. Of typical flower has 4 different kind of holes arranged successively on the swollen end of the stalk or pedicel called thalamus or receptacle. These are the calyx, corolla, androecium, and gynoecium. Calyx and corolla are accessory organ, while androecium and gynoecium are reproductive organ. In some flower like lily, the calyx and corolla are not distinct and are termed as perianth. When a flower has both androecium and gynoecium, it is bisexual. A flower having either only stamens or only carpels is unisexual. In symmetry, the flowers may be actinomorphic, or also known as radial, or zygomorphic, also known as bilateral symmetry. When flower can be divided into 2 equal radial halves in any radial plane through the center, it is said to be actinomorphic. For example, it is present in mustard, datura, chili. When it can be divided into 2 similar halves only in one particular plane, known as zygomorphic. Example, pea, gulmohar, bean, cassia. A flower asymmetrical irregular cannot be divided into any 2 similar type of halves into a vertical plane passing through the center present in canal. A flower may be trimerous, tetramerous, or pentamerous when floral appendages are 3, 4, and 5. Multiple of 3, 4, and 5 respectively. Flower with bracts reduced leaf found in the base at the base of petal are called bracteate, and those without bracts are called ebracteate. Here is position of floral parts on thalamus. A. Hypogynous, and B and C are perigynous, and D are epigynous. Epigynous, perigynous, and hypogynous. So, based on the position of calyx and corolla androecium respect to ovary of thalamus, the flower is described hypogynous, perigynous, and epigynous. The hypogynous flower, the gynoecium, occupy the highest position, while the other parts situated below it. The ovary in such flower is said to be superior, that is mustard, china rose, and brinjal. If gynoecium is situated in the center and other part of flower are located in the rim of thalamus almost at the same level, it is called perigynous. The ovary here is said to be half inferior, that is plum, peach, rose. In epigynous flower, the margin of thalamus grow upward and enclosing the ovary completely and getting fused with it. The other part of flower arises above the ovary. Hence, the flower is said to be inferior, as in flower of guava and cucumber, and the ray florets of sunflower. 5.5.1 parts of flower. Each flower normally has 4 floral holes, with calyx, corolla, androecium, and gynoecium. Figure 5.14. 5.5.1 Calyx. The calyx is the outermost hole of flower, and the member is called sepals. Generally, sepals are green, leaf-like, and protect the flower in the bud stage. The calyx may be gamosepalous, sepal united, or polysepalous, sepals free. 5.1.2 Corolla. Corolla is composed of petals. Petals are usually brightly colored to attract insects for pollination. Like calyx, corolla may also be gamopetalous, petal united, or polypetalous, petals free. The shape and color of corolla vary greatly in plants. Corolla may be tubular, bell-shaped, funnel-shaped, or wheel-shaped. Here a part of flower: androecium, gynoecium, corolla, calyx, pedicel. Calyx, corolla, androecium, gynoecium. Estivation: the mode of arrangement of sepals or petal in floral bud with respect to the other member of same whole is known as estivation. The main type of estivation are valvate, twist, imbricate, and auxiliary. When the sepals or petal or whole just touch one another at the margin without overlapping, as in calotropis, it is said valvate. If one margin of the appearance overlap to that of the next, so on, as in china rose, ladyfinger, and cotton, it is called twisted estivation. If the margin of sepal and petal overlap each other, one another, but not in any particular direction, as in cassia and gulmohar, the activation is said to be imbricate. In P and B flower, there are 5 petals. The largest, known as standard, overlap the 2 lateral petal wings, which is turn overlap the 2 smallest anterior petal keel. This type of activation known as axillary or papillonaceae. Here is valvate activation, twisted, imbricate and axillary. So valvate, twisted, imbricate and axillary. Axillary examples of axillary: P and imbricate, cassia, gulmohar and valvate. Examples of valvate are calotropis, and example of twist are china rose, ladyfinger and cotton. Cotton, china rose and ladyfinger, twisted, valvate, calotropis, imbricate, cassia and gulmohar. P axillary. Androsium 5.5.1.3. Androsium is composed of stamen. Each stamen, which represents the male reproductive organ, consists of stalk or a filament and an anther. Each anther is usually bilobed and each lobe has 2 chambers. The pollen sac. The pollen grain are produced in pollen sacs. A sterile stamen is called staminode. Stamens of flower may be united with other members such as petals or among themselves. When stamens are attached to the petals, they are epipetalous, as in brinjal, or epiphyllous when attached to the perianth, as in the flower of lily. So, epiphyllous in lily and epipetalous in brinjal. The stamen in flower may either remain free or may be united varying degree. The stamen may be united into one bunch or one bundle monodelphous, as in china rose, 2 bundle diadelphous, as in P, and more than 1 bundle polydelphous, as in citrus. There may be variation in length of filament within flower, as in salvinia and mustard. 5.1.4 Gynosium. Gynosium is the female reproductive part of flower and is made up of 1 or more carpels. A carpel consists of 3 parts, namely stigma, style and ovary. Ovary is the enlarged basal part on which lies the elongated tube, the style. The style connects the ovary to the stigma. The stigma is usually at the tip of style and is the receptive surface of pollen grain. Each ovary bears 1 or more ovule attached to the flattened cushion-like placenta. When more than 1 carpel is present, they may be free, as in lotus and rose, and are called apocarpus. They are termed as syncarpus when carpels are fused, as in mustard and tomato. After fertilization, the ovule develop into seeds and the ovary mature into fruits. Placentation. The arrangement of ovules within the ovary is known as placentation. The placentation are different types, namely marginal, axial, parietal, basal, free central and free central. In marginal placentation, the placenta form a ridge along the ventral strata of ovary and ovule are born on this ridge, and this ridge forming 2 rows, as in P. When the placenta in axial and the ovule are attached to it in a multilocular ovary, the placentation is said to be axial, as in china rose, tomato and lemon. In parietal placentation, ovule developed on the inner wall of ovary or on the peripheral part. Ovary is 1 chambered, but it becomes 2 chambered due to the formation of false septum, that is mustard and ergemum. When the ovules are born on central axis and septa are absent, as in dianthus and primrose, the placentation is called free central. In basal placentation, the placenta develops at the base of ovary and a single ovule is attached to it, as in sunflower and marigold. Here are examples: marginal and axile, parietal, central एंड बेसल. तो इसका एग्जांपल बहुत इंपॉर्टेंट कर लेना भैया. 5.6 द फ्रूट एक कैरेक्टराइज फीचर ऑफ फ्लावरिंग प्लांट. It is mature or ripened ovary developed after fertilization. If a fruit is formed by fertilization of ovary, it is called parthenocarpic fruit. Generally, the fruit consists of whole pericarp and seeds. The pericarp may be dry, fleshy. When pericarp is thick, fleshy, it is differentiated into outer epicarp and the middle mesocarp and the inner endocarp. Here is epicarp, mesocarp, seed, and endocarp. Part of fruit. Here is mango and here is coconut. Mango and coconut, the fruit is also known as drupe fruit. They develop from the monocarpellary superior ovary. And are end one-seeded. In mango, the pericarp is well differentiated into an outer thin epicarp, a middle fleshy edible mesocarp, and inner stony hard endocarp. In coconut, which is also known as drupe, the mesocarp is fibrous. 5.7 the seed. The ovule after fertilization develops into seed. A seed is made up of a seed coat and an embryo. The embryo is made up of radicle, an embryonal axis, and one as in wheat match, and one in wheat and match, or two cotyledon in gram and P. 5.7.1 structure of dicotyledonous seed. The outermost covering of seed coat. The seed coat has two layers, the outer testa and the inner tegmen. The hilum is a scar on the seed coat through which the developing seed were attached to the fruit. Above the hilum is small pore called micropyle. Within the seed coat is an embryo, consists of an embryonal axis and two cotyledon. The cotyledon often fleshy and full, rich of food material. At the two ends of the embryonal axis are present the radicle and the plumule. In some seeds, such as castor endosperm, are formed as a result of double fertilization. Is a food storing tissue and called endospermic seed. In plants such as bean, gram, and pea, the endosperm is not present in mature seeds, and such seeds are called non-endosperm. Structure of dicotyledonous seed. Here is micropyle, hilum, seed coat, cotyledon, plumule, radicle. 5.7.2 Structure of monocotyledonous seed. Generally, monocotyledonous seed are endospermic, but some, as in orchids, and non-endospermic. In the seeds of cereals such as maize, the seed coat is membranous and generally fused with fruit wall. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by proteinous layer called alluring layer. The embryo is small and situated in the groove at one end of endosperm. It consists of one large shield-shaped cotyledon known as scutellum and a short axis known as plumule and radicle. The plumule and radicle are enclosed with a sheath which is called coleoptile and coleorhiza. Here is the structure of monocotyledonous seed. Here is embryo and endosperm, coleorhiza cover radicle, and coleoptile cover plumule, and scutellum and endosperm. This represent what? Cotyledon. Yes. Seed coat of fruit wall, alluring layer made of protein, store protein, and endosperm. 5.8 Semi-technical description of a typical flowering plant. Here is a floral diagram with floral formula. Brassicaceae. Brassicaceae means mustard. So, zygomorphic male and female both present. Calyx 2+2. Corolla 4. Endosperm 2+4. Gynosium hypogynous 2 united. Various morphological features are used in describe and flowering plant. This description has been brief and simple. and scientific language represent a proper sequence. The plant is described beginning with the habit, vegetative character, root, stem, and leaves, and then floral characters, inflorescence, and flower parts. After describing various parts of plant, a floral diagram and a floral formula are presented. The floral formula is represented by some symbols. In the floral formula, BR stands for bracteate, K for calyx, C for corolla, P for perianth, A for androecium, G for gynoecium, G to the base, a small line for superior ovary, and G under a small line is for inferior ovary. This represents male, this represents female, and this represents bisexual plant. This represents actinomorphic and this represents zygomorphic nature of flower. Fusion is indicated by enclosing the figure within bracket and adhesion by a line draw above the symbol of the floral parts. A floral diagram provides information about the number of parts of flower, their arrangement, and the relation they have with one another. The position of the mother axis with respect to flower is represented by a dot on top of the floral diagram. Calyx, corolla, androecium, and gynoecium are drawn in successive holes, calyx being the outermost and gynoecium being in the center. Floral formula also shows cohesion and adhesion within the parts of holes and between holes. The floral diagram and floral formula in 5.20 represent the mustard brassicaceae. Here is the floral formula and diagram of brassicaceae. 5.9 description of some important family. 5.9.1 Fabaceae. The family was earlier called Papilionoideae of subfamily of family Leguminaceae. It is distributed all over the world. In figure 5.1 to 5.21 vegetative characters: trees, herbs, roots with root nodules, stem erect and or climber, leaves alternate pinnately compound or simple, leaf base pulvenated, stipulate, venation reticulate. 5.1 Pygium sativum, pea plant. A flowering twig, B flower, C petals, and D this represent reproductive part, and LS of carpel. And here floral diagram of Pygium sativum. This example of Fabaceae. So floral character: inflorescence racemose, flower bisexual, zygomorphic, calyx, sepal 5, gamosepalous, valvate, imbricate, aestivation, corolla, petal 5, polypetal. Papilionaceae consist of posterior standard 2 lateral wings and 2 anterior ones forming keel enclosing stamens, auxiliary aestivation, androecium 10 didelphus, and dioecious. Gynoecium ovary superior monocarpal unilocular with many ovules, style single. Fruit, legumes, seeds, one many non-endospermic. Floral formula zygomorphic. This represents zygomorphism. Yes. No. This is zygomorphic, bisexual, calyx 5 united, corolla 1 plus 2 plus 2 united, androecium 9 plus 1, gynoecium 1. Economic importance: many of the plants belonging to the family source pulses, gram, arar, same, mung, soybean, edible oils, and edible oils obtained from soybean and groundnut, dyes, indigofera, fiber, sun hemp, fodder, sesame, trifolium, ornamental, lupin, sweet pea, medicine, mulathy. 5.9.2 Solanaceae. It is large family commonly called the potato family. It is wider distribution in tropics, subtropics, even temperate zone. Vegetative character: plant mostly herbs and rarely small tree. Stems herbaceous, rarely woody, aerial, erect, cylindrical, branched, and solid. Solanum nigerum, Makoi plant. Flowering twig, flower, LS of flower, stamens, carpels. Floral diagram. or leaves hairy globulous underground stem. Potato solanum tuberosum. Leaves alternate, simple, rarely pinnately compound, stipulation, reticulate. Floral character, inflorescence, solitary, axillary, cymoid, as in solanum. Flower bisexual, actinomorphic, calyx, sepal, 5 united, persistent, valvate, aestivation. Corolla petal, 5 united, valvate, aestivation. Androecium, stamens, 5 epipetal. Gynosium bicarpally obligately placed, syncarpus, ovary superior, bilocular placenta, swollen with many ovule, exile, fruit, berry or capsule, seeds, many endosperms, floral, zygomorphic, both male and female present, calyx, 5 united, corolla 5 united, and androecium and corolla both united, gynosium. Economic importance. Many plants belonging to this family are source of some food: tomato, brinjal, potato, spices, chili, medicine, belladonna, ashwagandha, fumigatory, tobacco, ornamental, petunia, and liliaceae is data from this syllabus. So summary: flowering plant exhibit enormous variation in shape, size, structure, and mode of nutrition, lifespan, habitat, and they have well-developed root and shoot system. Root system is either taproot or fibrous root. Generally, dicotyledonous plants have taproot, while monocotyledonous plants have fibrous root. The roots in some plants get modified for storage of food, mechanical support, and respiration. The root system differentiated into leaf, stemless, flower, and fruit. The morphological feature of stems like presence of nodes and internodes, internal multicellular hair, and positively phototropic nature help to differentiate the stem from roots. Stem also get modified to perform diverse functions such as storage of food, vegetative propagation of and protection under different conditions. Leaf is lateral outgrowth of stem developed exogenously at the node. These are green in color, perform the function of photosynthesis. Leaves exhibit marked variation in their shape, size, margin, apex, and extend the incision of leaf blade lamina. Like other parts of plant, leaf also get modified into other structures such as truncated spine, climbing, and production respectively. Flowering. The flower modified shoot mean sexual reproduction. The flower are arranged different type of inflorescence. They exhibit enormous variation, structure, symmetry, position, and ovary relation. Other part, arrangement of petal, sepal, ovule. After fertilization, the ovary is modified into fruit and ovules into seeds. Seeds either may be monocotyledonous or dicotyledonous. They vary in shape, size, period, viability. The floral characteristics form the basis of classification and identification of flowering plant. This can be illustrated through semi-technical description of families. Hence, the flowering plant described in different sequence by using scientific terms. The floral features are represented in the summarized form as flower diagram and floral formula. So bro, भाई साहब अपना ये भी चैप्टर खत्म हुआ. Morphology in flowering plant. So morphology in flowering plants भी complete हुआ भाई लोग. तुम्हें मुझको बता दे चाहूँ मैं इतना बता दूँ तुझको. भाई अभी क्या है ना, इतना अभी कितना 4 ही चैप्टर हुआ ना अपना. इतना में ये देखो, ये याद कर लो, ये बढ़िया. वाल्वेट में क्या आएगा तुम्हारा, कैलोट्रॉपिस. ट्विस्टेड में क्या आएगा, ट्विस्टेड में देखो ओवरलैपिंग किस तरीके से. ये रहा ट्विस्टेड, इसका ओवरलैपिंग देखो, ये इसके ऊपर है और इसका ओवर इसके ऊपर और ये इसके ऊपर। ट्विस्टेड में फॉर्मल में तो इसमें आता है चाइना रोज और लेडी फिंगर और कॉटन। तो ट्विस्टेड में क्या गया, चाइना रोज, लेडी फिंगर, कॉटन। और इम्ब्रिकेट में क्या है, इम्ब्रिकेट में कैसिया और गुलमोहर। वेक्सिलरी में क्या आएगा, वेक्सिलरी में अपना पी आएगा। ठीक, तो भाई लोग ये अपना हुआ कंप्लीट। तो दो इतना अभी कितना ही चैप्टर हुआ भाई, अभी 4 चैप्टर अभी कंप्लीट हुआ यार। चलो नेक्स्ट। सॉरी ब्रो, व्हाट द हेक इज दिस। So let's start bro. एनाटॉमी ऑफ फ्लोरिंग प्लांट्स चैप्टर 6। भाई साहब हिम्मत नहीं कर रहा यार, लेकिन रुकेगा नहीं साला, झुकेगा नहीं साला। लेट्स स्टार्ट चैप्टर 6। एनाटॉमी ऑफ फ्लोरिंग प्लांट। 6.1 the tissue, 6.2 the tissue system, 6.3 anatomy of dicotyledons and monocotyledons plants, and 6.4 secondary growth। ये नहीं पढ़ेंगे बिकॉज नीट के सिलेबस में नहीं है, अपन वही पढ़ेगा जो नीट के सिलेबस में है। बाकी अपना काम बनता, भाड़ में जाए जनता। सो लेट्स स्टार्ट ब्रो। You can very easily see the structure similarity and variation in the external morphology of large living organisms, both plants and animals. Similarly, if we were to study the internal structure, we also find several similarities as well as differences. This chapter introduces the internal structure and functional organization of higher plants. Study of internal structure of plants is called anatomy. Plants have cells as the basic unit. Cells are organized into tissue and intern of tissue are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperm, the monocotyledons and dicotyledons are seen anatomically different. Internal structure are also shown adaptation to diverse environments. 6.1 the tissue. A tissue is a group of cells having a common origin and usually perform a common function. A plant is made up of different kinds of tissue. The tissue are classified into two main categories, namely meristematic and permanent tissue, based on whether the cells being formed are capable to dividing or not. 6.1.1 meristematic tissue. Growth in plant is largely restricted to a specialized region of active cell division called meristems. Greek word meristems, divide. Plants have different kind of meristems. The meristems which occur at the tip of roots and shoots are produced primarily tissue called apical meristem. Here is apical meristem, root and shoot. Central cylinder cortex, protoderm, initials of root cap, root apical meristem. Here is shoot, leaf primordium, shoot apical meristematic zone, axillary bud differentiating vascular tissue. Shoot apical meristem occupy the different root, while the shoot apical meristem occupy the distant-most region of stem axis. During the formation of leaf, the elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in axil of leaves and are capable of forming branch or flower. The meristem which occur between mature tissue is known as intercalary meristem. They occur in grass and regenerate parts removed by grazing herbivores. Both apical meristem and intercalary meristem are primary meristem because they appear early in life of plants or contribute to the formation of primary plant body. The meristem that occur in mature region of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, are called secondary or lateral meristem. They are cylindrical meristem, fascicular, vascular cambium, interfascicular cambium, and cork cambium are example lateral meristem. These are responsible for producing the secondary tissue. Following division of cell, both primary as well as secondary meristem, the newly formed cell become structurally and functionally specialized and lose ability to divide. Such cells are termed as permanent or mature cell and constitute the permanent tissue. During the formation of primary plant body sex, a specific region apical meristem produce dermal tissue, ground tissue, and vascular tissue. 6.1.2 Permanent tissue. The cell of permanent tissue do not generally divide further. Permanent tissue having cells similar to structure and function are called simple tissue. Permanent tissue having many different types of cells called complex tissue. 6.1.2.1 is simple tissue. A simple tissue is made of only one type of cell. The various simple tissue in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma form the major component within the organ. These cells of parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either closely packed or have a small intercellular space. The parenchyma perform various functions like photosynthesis, storage, and secretion. The collenchyma occur in layers below the epidermis in some of the dicotyledonous plants. It is found either as homogeneous layer or in patches. It consists of cells which are much thickened at the corner due to deposition of cellulose, hemicellulose, and pectins. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intracellular spaces are absent. They provide mechanical support to growing part of plants such as young stem and petiole of leaf. Sclerenchyma consists of a long narrow cell with thick lignified cell wall having a few or numerous pits. They are usually dead with and without protoplast. On the basis of variation in the form, structure, origin, and development, sclerenchyma may be either fibers or scleroid. The fibers are thick-walled, elongated, and pointed cells generally occurring in group in various parts of the plants. The scleroids are spherical, oval, or cylindrical, highly thickened cell with very narrow cavities in lumen. These are commonly found in fruits, walnuts, pulp fruits like guava, pear, sapota, seed coats of legumes, and leaves of tea. Sclerenchyma provide mechanical support to organ. Parenchyma, collenchyma, thickened corner protoplast, vacuole, cell wall, and sclerenchyma of fiber or scleroid. Scleroid, here is pits, here is lumen, and here is thick cell. Lumen, thick cell wall. 6.1.2.2 Complex tissue. The complex tissue are made up of more than one type of cell, and these work together as unit. Complex xylem and phloem constitute the complex tissue in the plants. Xylem function as conducting tissue for water and minerals, for roots, for stem and leaves. It also provides mechanical strength to plant parts. It is composed of 4 different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Gymnosperm lack the vessel in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layer of cell wall have thickening which vary in form. In flowering plant, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube-like structure made up of many cells called vessels member, each with lignified wall and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforation in their common walls. The xylem of vessel is a characteristic feature of angiosperms. Xylem fiber have highly thickened wall and obliterate central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin walled, and their cells are made up of cellulose. They store food material in the form of starch, fat, and other substances like tannins. Radial conduction of water takes place by the parenchymatous cell. Primary xylem is of two types, protoxylem and metaxylem. The first form, primary xylem element are called protoxylem. A later form, primary xylem, is called metaxylem. In stem, the protoxylem lies toward the centropiece and the metaxylem lies toward the periphery. In stems, protoxylem lies towards center, okay, and the metaxylem lies toward periphery of organs. The latter primary xylem is called endarch. The roots, protoxylem lies toward the periphery and metaxylem lies toward center. Such arrangement is called exarch. Phloem to transport food material usually from leaf to other part of plant. Phloem in angiosperm is composed of septive element, companion cell, phloem parenchyma, and phloem fiber. Gymnosperm have albuminous cell and sieve tube. They lack sieve tubes and companion cell. Sieve tube elements are also long tube-like structure arranged longitudinally and are associated with companion cell. Their end walls are perforated in a sieve-like manner to form of sieve tubes. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks the nucleus. The function of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with the sieve tube elements. The sieve tube element or companion cell are connected by pit field present within their common longitudinal wall. The companion cell help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated tapering in cylindrical cell which have dense cytoplasm and nucleus. The cell wall is composed of cellulose, has pit through which plasmodesmal metal connection exists between the cell. The phloem parenchyma store food material and other substances like resin, latex, and mucilage. The phloem parenchyma is absent in most of the monocotyledons. Phloem fibers, bast fibers, are made up of sclerenchymatous cell. These are generally absent in primary phloem, but are found in secondary phloem. These are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers are of joint flax and hemp are used to commercially The first formed primary phloem consists of narrow sheath tube and the referred as protophlloem, and later formed phloem is bigger tube and referred as metaphloem. Here is xylem tracheaidal vessel, and here is phloem. Sheath pore, sheath tube, filament, phloem parenchyma companion cell. The tissue system. We were discussing type of tissue system based on the type of cell present on. Now consider how tissue vary depending upon their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. These tissue system: epidermal tissue system, ground fundamental tissue system, and the vascular conducting tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms outermost covering of the whole plant body and comprises epidermal cell, stomata, and epidermal appendages. The trichomes and hair. The epidermis is the outermost layer of primary cell body. It's made of elongated, compactly arranged cells which form continuous layer. Epidermis is usually layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall or large vacuole. The outside of the epidermis is often covered with waxy thick layer called cuticle, which prevents the loss of water. Cuticle is present in roots, stomata, and structure are present in epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells called guard cell, which includes a stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of guard cell away from the stomatal pore thin, and inner wall toward the stomatal pore are highly thickened. The guard cell possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells are vicinity of guard cell become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture guard cells are surrounded by subsidiary cells are called stomatal apparatus. The cell of epidermis bear a number of hairs. The root hairs are unicellular elongation of epidermal cell, help in absorb water, minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in shoot system are usually multicellular. They may be branched and soft and stiff. They may even be secretory. The trichomes help in preventing water loss from transpiration. Here a diagrammatic representation. A stomata is bean-shaped guard cell. B stomata is dumbbell-shaped guard cell. Here is epidermal cell, and here is subsidiary cell, and here is chloroplast. Here is guard cell. And here is stomatal aperture. 6.2.2 The guard tissue system. All tissue except epidermis and vascular bundle constitute ground tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary range. In the primary stem and the roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cell called mesophyll. The vascular tissue system. The vascular system consists of complex tissue, the phloem and xylem. The xylem and phloem together constitute vascular bundle. In dicotyledonous system, the cambium is present between phloem and xylem. Such vascular bundle, because of presence of cambium, possess the ability to form secondary xylem and phloem tissue, and hence called open vascular bundle. In the monocotyledonous, the vascular bundle have no cambium present in them. Hence, since they do not form secondary tissue, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial. Such as in roots. In conjoint type of vascular bundle, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stem and leaves. The conjoint vascular bundle usually have phloem located only on outer side. Various type of vascular bundle: radial, conjoint open. Radial, conjoint open. 6.3 Anatomy of dicotyledonous and monocotyledonous plant. For better understanding of tissue organization of roots, stem and leaves, it is convenient to study transverse section of the mature zones of these organs. 6.3.1 Dicotyledonous root. Look at the figure 6.6. It shows the transverse section of sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin walled parenchyma cell with intercellular space. The innermost layer of cortex is called endodermis. It comprises of single layer of barrel-shaped cell without any intercellular space. The tangential as well as radial wall of the endodermal cells have deposition of water impermeable waxy material suberin in the form of Casparian strip. Next to the endodermis lies a few layers thick walled parenchymatous cell referred to as pericycle. Initiation of lateral root and vascular cambium during secondary growth take place in this cell. The pith is small and inconspicuous. The parenchymatous cell which lies between the xylem and phloem are characterized by conjunctival tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissue on the inner side of endodermis such as pericycle, vascular bundle, and pith constitute stele. 3.2 Monocotyledonous root. The anatomically of monocot root is similar to dicot root in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. As compared to dicot, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in monocot root. Pith is large and well developed. Monocotyledon root do not undergo any secondary growth. Transverse section of dicot root, primary and monocot root. Here in dicot root hair, epidermis, cortex, epidermis, pericycle, protoxylem, metaxylem, pith, and phloem. Root hair, epidermis, cortex, endodermis, pericycle, and here phloem, xylem, protoxylem, pith, metaxylem. Dicotyledonous 6.3.3 Dicotyledonous stem. The transaction of typical young dicotyledonous stem shows the epidermis, the outermost protective layer of the stem. Figure 6.7 covered with a thin layer of cuticle. It may be bare trichomes and fewer stomata. The cell arranged in multiple layers between epidermis and pericycle constitute a cortex. It consists of 3 subzones. The outer pericycle the outer hypodermis consists of a fewer layer of parenchymatous cell just below epidermis, which provide mechanical strength of young cells. Cortical layer below the hypodermis consists of rounded, thin-walled parenchymatous cell which conspicuous to intercellular space. The innermost layer of cortex is called endodermis. This cell of endodermis rich in starch layer, also referred to as starch sheath. Pericycle is present in endodermis above the phloem, which forms semilunar patches, sclerenchyma. In between the vascular bundle, there are few radially placed parenchymatous cells which constitute the medulla. A large number of vascular bundles are arranged in a ring arrangement. Vascular bundle is a characteristic of dicot. Each vascular bundle is conjoined, open, and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular space which occupy the central position stem constitute pith. 6.3.4 monocotyledonous stem. The cotyledonous stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath, and a large conspicuous parenchymatous growth tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the central located one. The phloem is present and water-containing cavity are present within the vascular bundle. 6.3.5 dorsiventral leaf. The dorsiventral leaf lamina shows three main parts, namely epidermis, mesophil, and vascular system. The epidermis, which covers both upper surface adaxial and epidermis, lower surface abaxial epidermis of leaf, has a conspicuous cuticle. Abaxial epidermis generally bear more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and lower epidermis is called mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cell: the palisade parenchyma and spongy parenchyma. The adaxial placed palisade parenchyma is made up of elongated cells which are arranged vertically in parallel to each other. The oval and round, loosely arranged spongy parenchyma situated below the palisade cell extend to the lower epidermis. There are large number of air cavities between these cells. Vascular system include vascular bundle, which can be seen in the veins of midriff. Size of vascular bundle dependent on size of veins. The veins vary in thickness in reticulate veins or dicot leaves. The vascular bundle are surrounded by a layer of thick walled bundle sheath. The long fine position of xylem bundles. Isobilateral 6.3 isobilateral monocotyledonous leaf. In anatomy of isobilateral leaf is similar to that of dorsiventral leaf in many ways. Its following characteristic differences: in an isobilateral leaf, stomata present on both the surface of epidermis and also mesophyll is not differentiated in palisade and spongy parenchyma. In grasses, the certain adaxial epidermal cell along with vein modified themselves into an empty colorless cell known as a bulliform cell. When the bulliform cell in the leaf have absorbed water and are turgid, the leaf surface is exposed. When they are palisade due to water stress, they make the leaves curl inward and minimize water loss. The parallel venation of monocot leaves reflect the near similar of vascular bundle, except main veins, as seen in vertical section of leaf. Tiers of leaf, dicot leaf and monocot leaf. Here dicot leaf और ये है monocot leaf. So monocot leaf में abaxial epidermis, stomata, substomatal cavity, xylem, mesophyll, adaxial epidermis, stoma, abaxial epidermis, substomatal cavity, spongy is mesophyll, air cavity, palisade, mesophyll, adaxial epidermis, phloem, xylem, bundle sheath. Summary: Anatomically, a plant is made up of different kind of tissue. The plant tissue are broadly classified into meristematic, apical, lateral, intercalary, and permanent, simple and complex. Assimilation of food and its storage, transportation of water, mineral and photosynthesis, and mechanical support are main function of tissue. There are three types of tissue system: epidermal, ground, and vascular. The epidermal tissue system is made up of epidermal cell, stomatal, and epidermal appendages. The ground tissue system is form of main bulk of the plant. It is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is made up of many xylem and phloem. On the basis of presence of cambium location, xylem, phloem, and vascular bundle are different type. Vascular bundles form conducting tissue and translocate water, mineral, and food material. Monocotyledon and dicotyledon plant marked by variation in their internal structure. They differ in type, number, location, and vascular bundle. The type and growth of most of dicotyledon's roots and stem increase with girth, diameter, organs, activity, vascular core, cambium with actually secondary ग्रोथ पढ़ना नहीं था, हटाओ इसको. सो ये हम लोग का रीडिंग कंप्लीट हुआ. ये रहा अपना मोनोकॉट लीफ का स्ट्रक्चर. ये रहा डाइकॉट लीफ का स्ट्रक्चर और डोर्सिवेंट्रल लीफ. ये रहा ट्रांसवर्स सेक्शन ऑफ स्टेम. तो ये पहला मोनोकॉट है, उसका डाइकॉट. ये ऊपर डाइकॉट का, ये डाइकॉट का ये अपना ट्रांसवर्स सेक्शन है लीफ का, स्टेम का. और ये अपना किसका है, मोनोकॉट का. इसके बाद अपन ये पढ़े डाइकॉट का, वो पढ़े मॉर्फोलॉजी ऑफ डाइकॉट, एनाटॉमी डाइकॉट का किए यहाँ पे रूट का. उसके बाद मोनोकॉट का किए. उसके बाद ये देखें रेडियल वास्कुलर बंडल, कंजवाइंट क्लोज, कंजवाइंट ओपन पड़े. उसके बाद ये स्टोमेटा का डायग्राम डाइकॉट का और मोनोकॉट का करें. उसके बाद ये जाइलम एंड फ्लोएम का स्ट्रक्चर देखें. परमानेंट टिश्यू ये कंप्लीट हुआ. अब अपन भाई करेंगे क्या, अब करेंगे नेक्स्ट चैप्टर स्ट्रक्चरल ऑर्गेनाइजेशन इन एनिमल्स. सो रेडी गाइस, चैप्टर्स चैप्टर 7 स्ट्रक्चरल ऑर्गेनाइजेशन इन एनिमल्स. तो यार रेडी स्टार्ट करते हैं इसको भी. 7.1 तो इसमें कौन सा टॉपिक्स पढ़ना अपने को, एनिमल टिश्यू. 7.2 ऑर्गन्स एंड ऑर्गन सिस्टम. 7.3 अर्थवॉर्म नहीं पढ़ेंगे इसमें. 7.4 कॉकरोच को पढ़ेंगे, हाँ फ्रॉग भी पढ़ेंगे. तो इसमें देखो पहले एनिमल टिश्यू पढ़ेंगे, मतलब रीड करेंगे. उसके बाद ऑर्गन एंड ऑर्गन सिस्टम करेंगे, जो छोटा टॉपिक है इसमें. फ्रॉग को नहीं करेंगे, कॉकरोच और फ्रॉग दोनों इसमें रीड करेंगे. तो रेडी हो जाओ, रेडी करते हैं, स्टार्ट, लेट्स गो. सो इन द प्रीसीडिंग चैप्टर यू केम अक्रॉस द लार्ज वैरायटी ऑफ ऑर्गेनिजम्स बोथ यूनिसेलुलर एंड मल्टीसेलुलर of the animal kingdom. In unicellular organism, all functions like digestion, respiration, and reproduction are performed by a single cell. In the complex body of multicellular animals, the same basic functions are carried out by different group of cells in a well-organized manner. The body of a simple organism like Hydra is made up of different type of cells, and the number of cells in each type can be thousands. The human body is composed of billions of cells to perform various functions. How do these cells in the body works together? In multicellular animals, a body group similar cell along with intracellular substances performed a specific function. Such an organization is called tissue. You may be surprised to know that all complex animals consist of only 4 basic types of tissue. These tissues are organized into a specific proportion and pattern to form an organ like stomach, lungs, heart, and kidney. When two or more organs perform a common function by their physical and chemical interaction, they together form organ system. That is digestive system, respiratory system, etc. Cell, tissue, organs, and organ system is split up to work in a way exhibit division of labor and contribute to survival of body as whole. 7.1 Animal tissue. The structure of the cell vary according to their functions. Therefore, the tissues are different and broadly classified into 4 types. First, epithelial. Second, connective. Third, muscular. And fourth is neural. 7.1.1 Epithelium tissue. We commonly refer to an epithelium tissue is epithelium. Epithelial tissue is epithelium. Plural, epithelia. This tissue has a free surface which faces either a body fluid or outside environment and thus provide a covering or lining of some part of the body. These cells are compactly packed with a little intercellular matrix. There are 2 types of epithelial tissue, namely simple epithelium and compound epithelium. Simple epithelium is composed of single layer of cell function as lining of body cavity, ducts, and tubes. The compound epithelium consists of 2 or more cell layers and has protective function as it does in our skin. On the basis of structure modification of cells, simple epithelium is further divided into 3 types. First is squamous, second is cuboidal, third is columnar. Figure 7.1 Simple epithelium. This is squamous, this is cuboidal, and here is columnar. And here is columnar cell bearing cilia. The squamous epithelium is made of a single thin layer of flattened cell with irregular boundaries. They are found in the wall of blood vessels, air sacs of lungs, and involved in function like forming diffusion boundary. The cuboidal epithelium is composed of a single layer of cube-like cell. This is commonly found in the duct of glands and tubular parts of nephron in kidney, and its main function are secretion and absorption. The epithelium of proximal convoluted tubule PCT of nephron in the kidney has microvilli. The columnar epithelium is composed of a single layer of tall and slender cells. Their nuclei are located at the base. Free surface may have microvilli. They are found in lining of stomach and intestine and help in secretion and absorption. If the columnar and cuboidal cell bear cilia on their free surface, they are called ciliated epithelium. Their function is to move particles or mucus in specific direction over epithelium. They are mainly present in the inner surface of hollow organs like bronchioles and fallopian tubules. Some of the columnar or cuboidal cells get specialized for secretion and are called glandular epithelium. They are mainly of 2 types: unicellular, consisting of isolated glandular cell, goblet cell of alimentary canal, and multicellular, consisting of cluster of cells, salivary glands. On the basis of mode of pouring their secretion, glands are divided into 2 categories, namely exocrine and endocrine glands. Exocrine glands secrete mucus, saliva, air wax, oil, milk, digestive enzymes, and other cell products. These products are released through ducts or tubes. In contrast, endocrine glands do not have ducts. Their products called hormone are secreted directly into the fluid bathing the gland. Compound epithelium. So here is a diagram of glandular epithelium, unicellular and multicellular. This is multicellular gland. and this is unicellular. So compound epithelium is made of more than one layer, multi-layered of cell, and thus has limited role in secretion and absorption. Their main function is to provide protection against chemical and mechanical stresses. They cover the dry surface of skin, the moist surface of buccal cavity, pharynx, inner lining of duct and salivary gland, end of pancreatic duct. All cells in epithelium are held together with little intercellular material. In nearly all animals' tissue, specialized junctions provide both structural and functional links between individual cells. Three types of cells junction are found in epithelium and other tissue. These are called as tight adhering or gap junction. Tight junction are held to stop substance from leaking across a tissue. Adhering junction perform cementing to keep neighboring cells together. Gap junction facilitate the cell to communicate with each other by connecting the cytoplasm of adjoining cell for rapid transfer of ions, small molecules and sometimes big molecules. 7.1.2 Connective tissue. Connective tissue are most abundant and widely distributed in the body of complex animals. They are named connective tissue because of their special function linking up separately other tissue organs of the body. They range from soft connective tissue to specialized tissue, which include cartilage, bone, adipose, and blood. In all connective tissue except blood, the cell secrete fiber of structural protein called collagen or elastin. The fibers provide strength, elasticity, and flexibility to the tissue. These cells also secrete modified polysaccharide which accumulate between cells and fibers and act as matrix. Ground substance connective tissue are classified into three categories: loose connective tissue, second dense connective tissue, and third is specialized connective tissue. 7.4 Loose connective tissue. First is areolar and second is adipose. In adipose tissue, here is fat storage area, here is nucleus, and here plasma membrane. And areolar tissue, this is macrophage, this is fibroblast, this is collagen fiber. Loose connective tissue. Loose connective tissue has a cell and fibers loosely arranged in semi-fluid ground substance. For example, areolar tissue present beneath the skin. Often, it serves as a support framework for epithelium. It contains fibroblast cells that produce and secrete fibers, macrophages, and mast cells. Adipose tissue is another type of loose connective tissue that located mainly beneath the skin. The cells of this tissue are specialized to store fats, the excess of nutrients which are not used immediately, and convert fat and store in this tissue. Fibers and fibroblasts are compactly packed in the dense connective tissue. Orientation of fibers shows a regular and or irregular pattern and are called dense regular and dense irregular tissue. In the dense regular connective tissue, the collagen fibers are represented in a row between many parallel bundles of fibers. Tendons which attach to skeletal muscle of bones, and ligament which attach to the one bone to another are example of this tissue. Dense irregular connective tissue has fibroblasts and many fibers, mostly collagen, that are oriented differently. This tissue is present in skin. Cartilage bones and blood are various type of specialized connective tissue. Here is dense connective tissue, dense regular, dense irregular. Intercellular material of cartilage is a solid, pliable, and resist compression. Cells of this tissue, chondrocytes, are enclosed in a small cavities within a matrix secreted by them. Most of cartilage in vertebrate embryo are replaced by bones in adults. Cartilage is present in tip of nose, outer ear joints, between adjacent bone of vertebral column, limbs, hands, and adults. Specialized tissue here is cartilage, collagen fiber, cartilage of cell or chondrocytes. Bones have a hard and non-pliable ground substance rich in calcium, salt, collagen fiber, which give bone its strength. It is main tissue that provide structure frame to the body. Bones support to protect softer tissue of organs. The bones of osteocytes are present in the space called lacunae. Limbs bones such as the long bones of legs serve weight-bearing functions. They also interact with the skeletal muscle attached to them to bring about movement. The bone marrow in some bones is the site production of blood cells. Blood is a fluid connective tissue containing plasma, red blood cells, RBC, white blood cells, WBC, and platelets. And it is a main circulating fluid that help in the transport of various substances. You will learn in chapter 17 and 18. 7.1.3 muscle tissue. Each muscle is made up of many long cylinder fibers arranged in parallel arrays. These fibers are composed of numerous fine fibrils called myofibrils. Muscle fibers contract to shorten in response to stimulus, then relax, lengthen, and strengthen their uncontracted state in coordinated fashion. Their action moved to the body to ascend to the changing environment and to maintain the position of various parts of the body. In general, muscle play an active role in all the movement of the body. Muscle are three types: skeletal and smooth and cardiac. Skeletal muscle tissue is closely attached to skeletal bone. In typical muscles such as biceps, stripped skeletal muscle fibers are bundled together in a parallel fashion. A sheath of tough connective tissue enclosing the several bundles muscle fiber. Learn from chapter 20. Muscle tissue: skeletal muscle, striated muscle, smooth muscle, and cardiac muscle. Here is skeletal muscle, smooth muscle, and cardiac muscles. Striation, nucleus, junction between adjacent cells. Smooth muscle fiber taper at both ends, fusiform, and do not show striation. Cell junction hold them together and they are bundled together in connective tissue sheath. The wall of internal organs such as blood vessel, stomach, intestine contain this type of muscle tissue. Smooth muscle are involuntary as their functioning cannot indirectly control. We usually are not able to make it contract merely by thinking about it as we can do with the skeletal muscle. Cardiac muscle system is a contractile tissue present only in the heart. Cell junction fusion with the plasma membrane of cardiac muscle cell are make them stick together. Communication junction intercalated disc at the some fusion point allow them to contract at unit. When cell receive signal contract there, its neighbor are also stimulated contract. Neural tissue 7.1.4 Neural tissue exerts greatest control over the body responsiveness in the change condition. Neurons in the unit of neural system and excitable cell. The neuroglial cell, which constitute the rest of neural system, protect and support neurons. Neuroglial make up some more than one half of the volume of neural tissue in our body. When neurons suitably stimulate, an electrical disturbance generated which swiftly travels along plasma membranes arrives of disturbance neurons ending or output zone trigger event that may cause stimulation or inhibition of adjacent neurons and other cell organs and organ system. The basic tissue of mentioned above organism to form organs which in turn associated with the form organ system in multicellular organism. Such an organization is essential for more efficient and better coordinated activities of millions of cells constituting an organism. Each organism in our body is made up of one or more type of tissue. For example, our body consists of all four type of tissue: epithelial tissue, connective tissue, muscular tissue, and neural. We also notice after some careful study that the complexity of our organ system displays certain discernible trend. This discernible trend is called evolutionary trend. You will study in detail in class 12. You are being introduced morphological and anatomic three organisms at different evolutionary levels to show their organization and functioning. Morphologically refers to study form and externally visible feature. In the case of plants, the microbes in terms of morphology precisely means only this. In case of animals, this refers to external appearance of organs or part of the body. The word anatomy conventionally is used for the study of morphology and internal organ in animals. You will learn the morphology and anatomy of earthworm, cockroach and frog representing invertebrates and vertebrates. अर्थफॉर्म तो भाई सिलेबस में नहीं है तो नहीं पढ़ूंगा मैं, हटाओ. इंसेक्ट और फ्रॉग दिया है, ठीक. तो इंसेक्ट में अपना कॉकरोच आएगा और फ्रॉग आएगा. चलो 7.4 कॉकरोच. Cockroach are brown and black bodied animals that are included in class Insecta of phylum Arthropoda. Bright yellow and green colored cockroach have also been reported in tropical region. Their size ranges from 1 by 4 inches to 3 inches, 0.6 to 7.6 centimeter. And they have long antenna legs and flat extension of upper body walls and conceals head. They are nocturnal omnivores that live in damp places throughout the world. They have become residents of human home and thus serious pest and vector in several diseases. 7.4.1 Morphology. The adult of common species of cockroach Periplaneta americana are about 34 to 54 millimeter long with wings that extend beyond the tip of the abdomen in males. The body of cockroach is segmented and divisible into three distinct regions: head, thorax, and abdomen. The entire body is covered by a hard chitinous exoskeleton, brown in color. In each segment, exoskeleton has hardened plates called sclerite, tergite dorsally, and sternite ventrally that are joined to each other by a thin flexible articular membrane, anthroidal membrane. External feature of cockroach: anus cerci, abdomen, hindwing, tegmina, head, filiform antenna, compound eye, pronotum, mesothorax, prothorax, metathorax, mesothoracic leg, metathoracic leg. Head is a triangular in shape and lies entirely at right angle to the longitudinal body axis. It is formed by fusion of 6 segments and show great mobility in all directions due to flexible neck. The head capsule bears a pair of compound eye. A pair of thread-like antennae arises from membranous socket lying in the front of eye. Antennae have sensory receptors that help in monitoring the environment. Anterior end of head bears appendages forming biting and chewing type of mouth parts. The mouth part consisting of labrum, upper lip, and a pair of mandible, a pair of maxillae, and a labium. Lower lip. A median flexible lobe acting as a tongue. Hypopharynx lies within the cavity enclosed by your mouth parts. The thorax consists of three parts: prothorax, mesothorax, and metathorax. The head is connected with thorax by a short extension of prothorax known as neck. Each of thoracic segment bears a pair of walking legs. This first pair of wings arises from mesothorax and the second pair from metathorax. Forewings mesothoracic called tegmina are opaque, dark and leathery and cover the hind wings when at rest. The hind wings are transparent, membranous and are used in flight. The abdomen in both males and female consists of 10 segments. In females, the seventh segment is boat-shaped and together with 8 segments and 9 segments sternum form a broad or genital pouch, whose anterior part contain a female gonopore, spermathecal pores and collateral glands. In males, genital pouch or chamber lie at the hind end of abdomen, bounded dorsally by 9th and 10th terga and ventrally by 9th sternum. It contains dorsal anus, ventral male genital pores, and gonapophysis. Males bear a pair of short thread-like anus styles which are absent in females. In both sexes, the 10th segment bears a pair of jointed filament structure called anal cerci. Okay. Figure 7.15, head region of cockroach. A, part of head region. B, mouth part. So it is labium, maxilla, compound eye, ocelus, mandible and labrum. And mouth parts, it is maxilla, labium, maxilla, mandible, labrum, hypopharynx, and in mandibulum this is grinding region and this is incising region. 7.4.2 Anatomy. Alimentary canal present in the body cavity is divided into 3 regions: foregut, midgut and hindgut. The mouth open into short tubular pharynx leading to the narrow tubular passage called esophagus. This in turn open into sac-like structure called crop, used for storing food. The crop is followed by gizzard or proventriculus. It has an outer layer of thick circular muscle and thick inner cuticle forming 6 highly chitinous plates called teeth. Gizzard help in grinding the food particle. The entire foregut is lined by cuticle. A ring of 6 to 8 blind tubules called hepatic or gastric ceca is present at the junction of foregut and midgut, which secrete digestive juice. At the junction of midgut and hindgut is present another ring, 100 to 150 yellow colored thin filamental malphizian tubules. They are help in removal of excretory product from the hemolymph. The hindgut is broader than midgut and is differentiated into ileum, colon and rectum. The rectum opens out through anus. Here is alimentary canal of cockroach: pharynx, salivary gland, salivary reservoir, esophagus, crop, gizzard, hepatic ceca, mesenteron or midgut, malphizian tubules, ileum, it is ileum, colon and it is rectum. Blood vascular system of cockroach is an open type. Blood vessels are poorly developed and open into space hemocoel. Visceral organs located in the hemolymph is bathed in blood hemolymph. The hemolymph is composed of colorless plasma and hemocytes. Heart of cockroach consists of elongated muscular tube lying along the mid-dorsal line of thorax and abdomen. It is differentiated into funnel-shaped chamber with ostia on either side. Blood from sinuses enter into heart through ostia and pumped internally to sinuses. The respiratory system consists of network of trachea that open through 10 pair of small holes called spiracles present on lateral side of body. Thin branching tubes, tracheal tubes subdivided into tracheoles, carry oxygen from air to all parts. The opening of spiracles regulated by sphincters. Exchange of gases take place by tracheoles by diffusion. Excretion is so it is open circulatory system in cockroaches. Anterior aorta, alari muscles, chambers of hearts. So there are 10 pair of small holes, spiracles. And here we go, excretion performed by Malpighian tubules. Each tubule is lined by glandular or ciliated cell. They absorb nitrogenous waste products and convert them into uric acid, which is excreted out through the hindgut. Therefore, this insect is called ureotelic. In addition, the fat body nephrocytes and urocose gland also help in excretion. The nervous system of cockroach consists of a series of fused, segmentally arranged ganglia joined by a paired longitudinal connectives on ventral side. 3 ganglia lie in the thorax and 6 in the abdomen. The nervous system of cockroach is spread throughout the body. The head holds the beat nervous system while the rest are situated along the ventral belly side part of its body. So now you understand what if the head cockroach cut off. It will still live for as long as 1 week. In the head region, the brain represented by supraosophageal ganglion, which supplies nerve to antenna and compound eyes. In cockroach, the sense organ are antenna, eyes, maxillary papilla, labial palps, anal cerci, etc. The compound eye are situated at the dorsal surface of the head. Each eye consists of about 2000 hexagonal ommatidia, singular nomen ommatidium. With the help of several ommatidia, a cockroach can receive several images of objects. This kind of vision is known as mosaic vision, with more sensitivity but less resolution, being common during night, hence called nocturnal vision. Cockroaches are dioecious and both sexes have well developed reproductive organs. Male's reproductive system consists of a pair of testes lying at each other lateral side as 4 to 6 abdominal segments. From each testis arises a thin wire difference which open into ejaculatory duct through a seminal vesicle. The ejaculatory duct open into a male gonophore situated ventral to the anus. A characteristic mushroom-shaped gland present at 6th to 7th abdominal segment, which function as accessory reproductive gland. The external genitalia are represented by male gonophysis or phallomere, chitinous asymmetrical structure surrounding the male gonopore. The sperm are stored in the seminal vesicle and are glued together in the form of bundle called spermatophores, which are discharged during the copulation. The female reproductive system consists of 2 large ovaries lying laterally in the 2 to 6 abdominal segment. Each ovary is formed of a group of 8 ovarian tubules or ovules containing a chain of developing ova. Oviduct of each ovary unite into a single median oviduct, also called vagina, which open into the genital chamber. A pair of spermatheca is present at 6th segment, which open into the genital chamber. Sperms are transferred through spermatophore. Their fertilized eggs are encased in a capsule called ootheca. Ootheca is a dark reddish to blackish brown capsule about 3 to 8 mm long. They are dropped or They are domed or glued to suitable surface, usually in crack or crevices, highly relative humidity near a food source. On an average, female produce 9 to 10 ootheca, each containing 14 to 16 eggs. The development of Periplaneta americana is porometabolous, meaning there are development through nymphal stage. The nymph to look very much like adults. The nymph grows by molting about 13 times to reach the adult form. The next to last nymphal stage has wing pad, but only adult cockroach have wings. Many species of cockroaches are wild and are no known economic importance yet. A few species thrive and contain around human habitats. They are pests because they spoil food and contaminate it with their smelly excreta. They can transmit a variety of bacterial diseases and containing food material. So here is reproductive system of cockroach, and it is male organ and it is female reproductive organ. So this is testes and this is phallic gland, small tubules, long tubules, seminal vesicle, vas deferens, ejaculatory duct. This is right phallomere. This is ventral phallomere. This is anus cersus, and this is caudal style, pseudopenis, titillator, left phallomere. And in female, ovary, oviduct, common oviduct or vagina, collateral gland, genital chamber, vestibulum, and gonopophysis, spermatotheca, and the genital pouch consists of genital chamber and vestibulum. And let's go 7.5 frogs. Frogs can live both on land and fresh water and belongs to class Amphibia or phylum Chordata. The most common species of frog found in India is Rana tigrina. They do not have constant body temperature, that is, their body temperature varies with temperature of the environment. Such animals are called cold-blooded or poikilotherms. You might have also noticed change in the color of the frogs while they are on grasses and dry land. They have ability to change their color, hide them from their enemies, camouflage. This protective coloration is called mimicry. You may also know that frogs are not seen during peak summer and winter. During this period, they take shelter in deep burrows to protect them from extreme heat and cold. This is known as summer sleep and estivation, and winter sleep known as hibernation, respectively. 7.5.1 Morphology. Have you ever touched the skin of frog? The skin is smooth and slippery due to the presence of mucus. The skin always contains the moist condition. The color of dorsal side of frog generally olive green and darker irrelevant spot. On the ventral side of the skin is uniformly pale yellow, and the frog never drink water but absorb it through skin. Body of frog is divisible into head and trunk, a neck and tail absent. Above the mouth, a pair of nostrils present. Eyes are bulged out, covered a nictitating membrane that protects them. While in water, on the either side of eye membranous tympanum, ear receive sound signals. The forelimbs and hind limbs help in swimming, walking, leaping, burrowing. The hind limbs and in 5 digits they are larger and muscular than forelimbs and that end in 4 digits. Feet have webbed digits that help in swimming. Frogs exhibit sexual dimorphism, Male frogs can be distinguished by the presence of sound-producing vocal sacs and also copulatory pad uh, on the first digit of forelimbs, which absent in female frog. So here is the external feature of frog: hind limb, forelimb, eye, head, and trunk. 7.5.2 Anatomy. The body cavity of frogs accommodated different organ systems such as digestive, circulatory, respiratory, nervous, excretory, and reproductive system with a well-developed structure and function. The digestive system consists of alimentary canal and digestive gland. The alimentary canal is short because frogs are carnivores and hence the length of intestine is reduced. The mouth is open into buccal cavity that leads to the esophagus through pharynx. Esophagus is a short tube that opens into stomach, which in turn continues as intestine, rectum, and finally opens outside the cloaca. Liver secretes bile that is stored in the gallbladder. Pancreas, a digestive gland, produces pancreatic juice containing digestive enzyme. Food is captured by the bilobe tongue. By bilobe tongue, digestion of food takes place by action of HCl and gastric juice secreted from walls of stomach. Partially digested food called chyme is passed from stomach to the first part of small intestine. The duodenum receives bile from gallbladder and pancreatic juice from the pancreas through common bile duct. Bile emulsifies the fat. Pancreatic juice digests the carbohydrate and protein. Final digestion takes place in the intestine. Digested food is absorbed by numerous finger-like folds in wall of intestine called villi or microvilli. The undigested solid waste moves into rectum and passes out through cloaca. Frogs respire on land and in water by two different methods. In water, skin acts as aquatic respiratory organ. Cutaneous respiration. Dissolved oxygen in water is exchanged through skin by diffusion. So here is a diagrammatic representation of internal organ of frogs showing complete digestive system. Heart, esophagus, liver, stomach, intestine, it is rectum, it is cloacal aperture, and this is cloaca, and this is urinary bladder, ureter, kidney, fat bodies, very important. This is fat bodies, lungs, gallbladder. On the land, buccal cavity, skin and lungs act as a respiratory organ. The respiration of lung is called pulmonary respiration. The lungs are pair elongated pink color sac-like structure present in upper part of trunk region, thorax. Air enters through the nostril into the buccal cavity and then the lungs. During the aestivation and hibernation, gaseous exchange take place through skin. The vascular system of frog is well developed closed type. Frog have lymphatic system also. The body vascular system involves heart, blood vessels, and blood. The lymphatic system consists of the lymphatic system. 1 minute. What happens? The lymphatic system consists of lymph channels, lymph nodes. Heart is a muscular structure situated in the upper part of body cavity. It has chambers, 2 artery and 1 ventricle, and the covered with membrane called pericardium. A triangular structure called sinus venosa join the right atrium and it receives blood through the major veins called vena cava. The ventricle opens into sac-like conus arteriosus on the ventral side of heart's side. The blood from the heart is carried out all part of the body by arterial system. The veins collected blood from different part of the body is called is from heart and from heart the venous system. Special venous connection between the liver and intestine as well as kidney and lower parts of the body are present in the frog. The former is called hepatic portal system and the latter is called renal portal system. The blood is composed of plasma and plasma cells and the blood cell are अरे भाई चार्ज हो जाए यार the blood cell are RBC, erythrocyte WBC, white blood cell leukocyte, and the platelets. RBC are nucleated and contain red colored pigment, namely the hemoglobin. The lymph is different from the blood. It lacks few proteins and RBC. The blood carries nutrient, gases, and water to the respective site during the circulation. The circulation of blood is achieved by pumping action and muscular heart. The elimination of nitrogenous waste is carried out by a well-developed excretory system. The excretory system consists of a pair of kidney, ureter and cloaca and urinary bladder. These are compact, dark and red bean-like structures situated a little posterior in body cavity. On the other side, vertebral column. Each kidney is composed of several structural and functional units called uriniferous tubules and nephrons. Two ureters emerge from kidney in the male frogs. The ureters act as urogenital duct which open into the cloaca. The female in the female the ureter and oviduct open separately in cloaca. The thin-walled urinary bladder is present ventral to the rectum which also open into cloaca. The frog excretes urea and thus is a ureotelic animal. Excretory waste is carried by blood into kidney where it separates and excretes. The system for control and coordination is highly evolved in the frog. It includes both neural system and endocrine glands. The chemical coordination of various organs and blood is achieved by hormones which are secreted by endocrine glands. The prominent endocrine glands found in frog are pituitary, thyroid, parathyroid, thymus, and pineal glands, pancreatic islets, adrenals, gonads. The nervous system is organized into central nervous system, brain, and spinal cord, of peripheral nervous system, cranial, spinal nerves, and autonomic nervous system, sympathetic and parasympathetic. There are 10 pair of cranial nerves arising from the brain. Brain is enclosed in a bony structure called brain box or cranium. The brain is divided into forebrain, midbrain, and hindbrain. Forebrain include olfactory lobe, paired cerebral hemisphere, and unpaired diencephalon. The midbrain is characterized by a pair of optic lobes. The hindbrain is consists of cerebellum and medulla oblongata. The medulla oblongata pass out through the foramen magnum and contribute into the spinal cord, which is enclosed in the central vertebral column. Frog has different type of sense organ, namely organs of touch, sensory papillae, taste buds, smell, nasal epithelium, vision, eye, and hearing tympanum with internal ears. Out of these, eye and internal ears are well organized structure and the rest are cellular or aggregation around nerve ending. Eyes in the frog are a pair of spherical structures situated in the orbit of a skull. There are simple eyes possessing only one unit. External ear is absent in frog. Only tympanum can be seen externally. The ear is an organ of hearing as well as balancing equilibrium. Frogs have a well organized male and female reproductive system. Male reproductive organ consists of a pair of yellowish oval testes which are found in adult upper part of kidney by the double fold peritoneum called mesoneum. Vesi franci are 10 to 12 in number that arises from testes. They enter the kidney on their side and open into bladder canal. Finally, it communicates with the urinogenital duct that come out of the kidneys and open into cloaca. The cloaca is a small medium chamber that is used to pass fecal matter, urine, and sperm to the exterior. The female reproductive organs include pair of ovaries. The ovaries are situated near the kidney and there is no functional connection with kidney. A pair of oviduct arising from the ovary open into the cloaca separately. A matured female can lay 200, 500, 3000 ova at a time. Fertilization is external and takes place in water. Development involves a larval stage called tadpole. Tadpole undergo metamorphosis to form the adult. Frogs are beneficial for mankind because they eat insects and protect the crop. Frogs maintain ecological balance and because of this serve as an important link food chain and food web in the ecosystem. In some countries, the muscular legs of frog are used to food by man. Summary: since tissue, organs, and organ system is split up, the work in a way that ensures the survival of body as a whole. The and exhibit division of labor. A tissue is defined as a group of cells along with intercellular substance performing one or more function in the body. Epithelia is a sheet-like tissue lining the body surface and its cavity. Ducts and tubes. Epithelia have one or free surface facing the body fluid or the outside environment. Their cells are structurally and functionally connected at junction. Diverse type of connective tissue bind together, support strength and protect fiber insulated other tissue in the body. Soft connective tissue consists of protein, fibers as well as variety of cells arranged in around ground substance. Cartilages, bone, blood, and adipose tissue are separated connective tissue. Cartilage and bone are both structural material. Blood is fluid tissue with transport function. Adipose tissue is a reservoir of stored energy. Muscle tissue which contract shorten in response to stimulation. Help in movement of body specific body part. Skeletal muscle is a muscle tissue attached to the bone. Smooth muscle is a component of internal organ. Cardiac muscle makes up the contractile walls of the heart. Connective tissue cover all three types of tissue. Nervous tissue exerts greatest control over the response of the body. Neurons are the basic unit of nervous tissue. अर्थवर्म नहीं करना है. Cockroach and frogs show characteristic feature of the body. Threat में हटाओ. Body of cockroach Periplanta americana is covered by chitinous exoskeleton. It is divided into head, thorax, and abdomen. Segment bear jointed appendages. There are 3 segments of thorax, each bearing a pair of walking legs. 2 pairs of wings are present. 1 pair each is second and third segment. There are 10 segments in abdomen. Alimentary canal is well developed with a mouth surrounded by mouth parts. A pharynx, esophagus, grope, gizzard, midgut, hindgut, anus, hepatic ceca are present at the junction of foregut and midgut. Malphizian tubules are present at the junction of midgut and hindgut. Helps in excretion. A pair of salivary glands are present near the crop. The blood vascular system is open type. Respiration takes place by network of trachea. Trachea opens outside with spiracles. Nervous system is represented by a segmentally arranged ganglia, ventral nerve cord. A pair of testes is present in 4 to 6 segments of an ovary in a 2 to 6 segment. Fertilization is internal. Female produce 9 to 10 ootheca bearing developing embryos. After rupturing the single ootheca, 16 young ones called nymphs come out. The Indian bullfrog Rana tigrina is the common frog found in India. Body is covered by a skin. Mucus gland is present in the skin, which is highly vascularized and helps in respiration in water and lands. Body is divisible into hair, trunk. A muscular tongue is present, which is bilobed at the tip and is used in capturing the prey. The alimentary canal consists of esophagus, stomach, intestine, and rectum, which open into cloaca. The main digestive system of liver and pancreas. It can respire water through skin through lungs on land. Circulatory system is closed with single circulation. RBC are nucleated. Nervous system is organized into central, peripheral, and autonomic. The organs of urinogenital system of kidney are urinogenital ducts, which open into cloaca. The male reproductive organ is a pair of testes. The female reproductive organ is a pair of ovaries. A female layer 2,500 to 3,000 ova at a time. The fertilization and development is external. The egg hatch into tadpoles, which metamorphose into frogs. So अभी 4 बज रहे हैं और हम लोग का कितना चैप्टर हुआ? 2 यूनिट, मतलब कि 2 यूनिट कंप्लीट हुए टोटल. अभी टाइम नहीं देखना चाहिए हमको. कौन कौन से चैप्टर कवर हो गया? तो चैप्टर अपना कवर हो गया. फर्स्ट लिविंग वर्ड प्लस प्लांट किंगडम प्लस एनिमल किंगडम. लिविंग वर्ड के बाद फर्स्ट चैप्टर, सेकेंड चैप्टर भी था, थर्ड, फोर्थ. इसके बाद अपन कर रहे हैं क्या? मॉर्फोलॉजी इन प्लांट प्लस एनाटॉमी इन प्लांट्स. उसके बाद मॉर्फोलॉजी इन एनिमल. कंप्लीट हो गया अपना. अब अपन करेंगे क्या? सेल यूनिट एंड स्ट्रक्चर ऑफ लाइफ. रेस्ट. 7 minutes. चार 12 4 20. हजारों में बहन. भाई खतरनाक लड़का है भाई. कौन है? खुद को देख वे, है चांद में है दाग फिर न तुझ में एक भी, तुझ पे हक मेरा तेरे हवाले कर दिया। जरा तुम्हें मेरी नजर से खुद को देखेंगे, है चांद में है दाग फिर सो गाइस हो गया रेस्ट, बहुत रेस्ट टाइम हो गया अपने कर्तव्य पाथ पे चलने के लिए। सो गाइस लेट्स स्टार्ट नेक्स्ट जर्नी ऑफ दिस चैप्टर। सो जरा कभी मेरी नजर, तो भाई ये स्टार्ट करने से पहले कुछ शायरी हो जाए। ठोकरे खा के भी ना संभले तो मुसाफिर का नसीब, वरना पत्थरों ते तो अपना फर्ज निभा ही दिया। तो ये कुछ शायरी ऐसे हैं, ठोकरें खाकर भी अगर नहीं संभल रहा ना, तो ये नसीब ही है भाई। नहीं तो पत्थरों ने तो अपना वो क्या फर्ज निभा ही दिया है ठोकर देकर। अब नहीं संभल पाए तो गलती तुम्हारा है भाई। तो ठोकर लगे तो अच्छी बात है, उसको देख के बुरा नहीं मानो, उससे सीखो कुछ। ठीक है मेरा भाई, ठोकर लगती हैं कुछ सिखाने के लिए, उसको पॉजिटिव वे में लो और आगे बढ़ो। तो चलो भाई हम लोग अपना सेल बायोलॉजी स्टार्ट करते हैं। तो चैप्टर 8, सेल द यूनिट ऑफ लाइफ। इस यूनिट में हम लोग पढ़ेंगे सेल यूनिट ऑफ लाइफ, बायोमोलिक्यूल्स, और चैप्टर 10 सेल साइकिल एंड सेल डिवीजन। तो अपन क्या करेंगे, पहले सेल यूनिट ऑफ लाइफ पढ़ेंगे, बायोमोलिक्यूल्स, उसका सेल साइकिल एंड सेल डिवीजन, पूरा सीक्वेंस से पढ़ेंगे, कुछ भी इधर उधर नहीं करेंगे अपन। यूनिट 3 सेल स्ट्रक्चर एंड फंक्शन। सो biology is the study of living organisms. The detailed descriptions of their forms and appearance only brought out their diversity. It is the cell theory that emphasizes the unity of underlying this diversity and of forms. That is the cellular organization of all life forms. A description of cell structure and cell growth of by division is given by in chapter comprising this unit. Cell theory also created a sense of mystery around the living phenomena, that is, physiological and functional behavioral processes. This mystery was requirement of integrity of cellular organization of living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral process, one can take physical and chemical approach and use cell-free system to investigate. This approach enables us to describe the various processes in molecular terms. The approach is stabilized by analysis of living tissue for elements and compounds. It will tell us what type of organic compound are present in living organism. In the next stage, one can ask the question, what are these compounds doing inside the cell, and in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain abnormal processes that occur during any disease condition. This physicochemical approach to study and understand a living organism is called reductionist biology. The concept and technique of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules are provided. So let's start. तो जी.एन. रामचंद्रन के बारे में कुछ देखें. G.N. Ramachandran, an outstanding figure in the field of protein structure, was founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of triple helical structure of collagen published in Nature in 1954, and his analysis of allowed conformation of protein through the use of Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922, in a small town not far from Cochin, on the southwest coast of India. His father was professor of mathematics at a local college and thus had a considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as the top-ranking student in BSc Honours Physics courses of the University of Madras. He received PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publication on models of alpha helix and beta sheet structure, directed his attention to solving structure of collagen. He passed away at the age of 78 on 7 April 2001. Chapter 8, Cell, the Unit of Life. तो इसमें अपन पढ़ेंगे व्हाट इज सेल, सेल थ्योरी, एन ओवरव्यू ऑफ सेल, प्रोकैरियोटिक सेल, यूकैरियोटिक सेल. So let's start. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it an inanimate thing does not have which a living thing has? The answer to this presence of basic unit of life, the cell in all living organism. All organisms are composed of a cell. Some are composed of single cell and are called unicellular organisms, while others, like us, composed of many cells called multicellular organisms. 8.1. What is cell? Unicellular organisms are capable of first, independent existence, and second, performing essential function of life. Anything less than a complete structure of cell does not ensure independent life. Hence, cell is a fundamental structure and functional unit of living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to electron microscope revealed all the structural detail of the cell. 8.2 Cell theory. In 1838, Matthias Leiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form tissue of plants. At about same time, Theodore Swan, 1839, a British zoologist, studied different type of animal cell and reported that cell had a thin outer layer which today known as plasma membrane. He also concluded based on his study on plant tissue that the presence of cell was unique characters of plant cell. On the basis of this, Swan proposed the hypothesis of that bodies of animals and plants composed of cell and product of cells. Slide and Swan together formulated the cell theory. This theory, however, did not explain how new cells were formed. Rudolf Virchow, 1855, first explained that cell divided and new cells are formed from pre-existing cell, omnicellular acellular. He modified the hypothesis of Slider and Swan to give the cell theory in final shape. Cell theory, as you understand today, is first, all living organisms are composed of cells and product of cells. Second, all cells arise from pre-existing cell. 8.3 An overview of a cell. You have earlier observed cell in an onion peel and on or human cheek, cell under microscope. Let us recollect their structure. The onion cell is a typical plant cell, has distinct cell wall as its outer boundary, and just within it is cell membrane. The cell of human cheek have an outer membrane as a delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contains the genetic material, DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas that lack membrane bound nucleus are called prokaryotic. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both plant and animal cells. Various chemical activities occur in it, keep the cell living state. Beside the nucleus, the eukaryotic cell have membrane-bound distinct structure called organelles like endoplasmic reticulum, Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cell lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in cytoplasm but also within two organelles, chloroplast and mitochondria of endoplasmic reticulum. Animal cell contain another non-membrane bound organelle called centrosomes, which help in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasma, the smallest size is only 0.2 micrometer in length, while the bacteria could be 3 to 5 micrometer. The largest isolated single cell of egg of ostrich among multicellular organisms. Human red blood cells are about 7.0 micrometer diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-shaped, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of cell may be vary with the function they perform. Diagram showing different shapes. RBC, round and concave, white blood cells, amoeboid, columnar epithelium, long and narrow, nerve cell branch and long, and tracheoid, elongated, mesophyll, round and oval. So 3.4 prokaryotic cell. The prokaryotic cell is represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organism. They are generally smaller and multiply more rapidly than eukaryotic cell. They may vary greatly in shape and size. The four basic shape of bacteria are bacillus rod-like, coccus spherical, vibrio comma-shaped, and spirillum spiral. The organization of prokaryotic cell is fundamentally similar, even though prokaryotic exhibits a wide variety of shapes and functions. All prokaryotics have cell wall surrounding the cell membrane except mycoplasma. The fluid matrix filling the cell in the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to genomic DNA, the single chromosome circular DNA. Many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmids DNA conforms certain unique phenotypic characters such bacteria. One such character is resistant to antibiotic. In higher classes, you will learn that this plasmid DNA used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryote. No organelles like the ones in eukaryote are found in prokaryotic cell except for ribosomes. Prokaryotes have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryote. They are essentially enfolding of cell membrane. 8.2 diagram showing comparison of eukaryotic cell with others. Typical eukaryote 10 to 20 micrometer. Typical bacteria 1 to 2 micrometer. PPLO 0.1 micrometer. Viruses 0.01 to 0.2 micrometer. 0.4.1 cell envelope and its modification. Most prokaryotic cells, particularly the bacteria cell, have the chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure, that is the outermost glycocalyx followed by a cell wall and then plasma membrane. Although each layer of envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of their difference in cell envelopes and manner of their response to staining procedure developed by Gram, which namely those that take up Gram strain, Gram positive, and other that do not take Gram negative bacteria. Glycocalyx differ in composition of thickness among different bacteria. It could be loose sheath called slime layer in some, while in others it may be thick or tough. Bacteria, the capsule. The cell wall determines the shape of cell and provide strong structural support to prevent the bacterium from bursting and collapsing. The plasma membrane is selectively permeable and nature are interacts with outside world. This membrane is similar structure to that of eukaryotes. A special membranous structure is the mesosome, which is formed by extension of plasma membrane into cell. These extensions are in the form of vesicle, tubules, and lamella. They help in cell wall formation, DNA replication, and distribution of daughter cell. They also help in respiration, segregation process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryote like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophore which contain pigments. Bacterial cell may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number of arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, basal body. The body is the longest portion and extends from the cell surface to the outside. Beside flagella, pili fimbriae are also surface structure of the bacteria but do not play a role in motility. Pili is a elongated tubular-like structure made up of special protein. The fimbriae is small bristle-like fiber sprouting out of the cell. In some bacteria, they are known to help attach the bacterium rock and stream and also to the host tissue. 8.4.2 Ribosomes and inclusion body. In prokaryotes, ribosomes are associated with plasma membrane of the tissue. They are about 15 nanometer to 20 nanometer in size and are made up of two subunits, 15S and 30S. Units which, when presented together, form 70S prokaryotic ribosomes. Ribosomes are site of protein synthesis. Several ribosomes may be attached to single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryote cell are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in cytoplasm. That is phosphate granules, synophysian granules, glycogen granules, gas vacuoles, and are found in blue, green algae, purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes include all the protists, plants and animals and fungi. In eukaryotic cells, there is extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear membrane envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as farmer possess cell wall, plastid and large central vacuoles which are absent in animal cell. On the other hand, animal cells have centrioles which are absent in almost all plant cell. Here is plant cell and animal cell. In plant cell, this is smooth endoplasmic reticulum. This is lysosome. This is rough endoplasmic reticulum. This is nucleus. Whole is nucleus, and this is nucleolus, and this is Golgi apparatus, nuclear envelope, plasma membrane, vacuole. This is vacuole, middle lamella, the outer cell wall, mitochondria, ribosomes, chloroplast, cytoplasm, and peroxisome, microtubule, plasmodesmata, okay, and animal cell, microvilli, plasma membrane, centriole, peroxisome, lysosome. This is ribosomes, mitochondria, rough endoplasmic reticulum, cytoplasm. These are all cytoplasm, and Golgi apparatus, smooth endoplasmic reticulum, nuclear envelope, nucleolus. This is nucleolus, and this whole is nucleus. Oh, what's happened, bro? Sorry for this disruption, bro. Let us now look at individual cell organelles and understand their structural functions. 8.5.1 Cell membrane. The detailed structure of membrane was studied only after the advent of electron microscope in 1950. Meanwhile, chemical studies on the cell membrane, especially in human red blood cell (RBC), enabled the scientists to deduce the possible structure of plasma membrane. These studies shows that the cell membrane is mainly composed of lipids and proteins. The major lipids and phospholipids that arrange in a bilayer are also the lipids are arranged within the membrane with the polar head toward the outer side and hydrophobic tail toward the inner side. This ensures that the non-polar tail of saturated hydrocarbon is protected from aqueous environment. In addition to phospholipid, membrane also contain cholesterol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different types of cells. In human beings, the membrane of the erythrocytes has approximately 52% of protein and 40% lipid and 52% protein. Depending on ease of extraction, membrane protein can be classified as integral or peripheral. Peripheral protein lies on the surface of membrane, while integral proteins are partially or totally buried in the membrane. Fluid mosaic model of plasma membrane: cholesterol, sugar, peripheral protein, integral protein, and phospholipid bilayer. Important: cholesterol, sugar, and peripheral protein. An improved model of structure of cell membrane was proposed by Singer and Nicholson in 1972. Widely accepted fluid mosaic model. According to this, the quasi-fluid nature of lipid enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as fluidity. Okay, the fluidity nature of membrane is also important for the point of view function like cell growth, formation of intercellular junction, secretion and endocytosis, cell division, etc. One of the most important functions of plasma membrane is the transport of molecule across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient from the higher concentration to the lower. Water may also move across the membrane from higher to lower concentration. Movement of water diffusion is called osmosis. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar bilayer, they require a carrier protein for the membrane to facilitate their transport across the membrane. A few ions or molecules that are transported across the membrane against their concentration gradient, that is from the lower to higher concentration. Such transport is an energy dependent process in which ATP utilized, and this is called active transport, that is sodium potassium pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called cell wall form an outer covering for plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from the mechanical damage and infection, it also helps to cell-to-cell interaction and provide barrier to understable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of young plants cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on inner toward membrane side of the cell. The middle layer is a The middle lamella is a layer mainly of calcium peptide which hold or glue different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm or neighboring cell. 8.5.3 Endomembrane system. While each of the membrane organelles are distinct in terms of its structure and function, many of these are considered together as endomembrane system because their function and are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the function of mitochondria, chloroplast, and peroxisomes are not coordinated with the above component, these are not considered as a part of endomembrane system. Endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuole. 8.5.3.1 Endoplasmic reticulum. Electron microscope study the eukaryotic cell reveal the presence of network or reticulum or obtaining tubular structure is scattered in cytoplasm. That is called endoplasmic reticulum here. Hence, endoplasmic reticulum includes intracellular space into two distinct compartments, that is luminar inside endoplasmic reticulum and extraluminar cytoplasm compartment. The endoplasmic often shows ribosome attached to their outer surface. Their endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear small and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum frequently observe the cell actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In the animal cell, lipid like steroidal hormones are synthesized in smooth endoplasmic reticulum. Endoplasmic reticulum. This is smooth endoplasmic reticulum. If ribosome present, then rough endoplasmic reticulum. This is nucleus and this is nuclear pore. 8.5.3.2 Golgi apparatus. Camille Golgi in 1898 first observed densely distant reticular structure near the nucleus. These were later named Golgi body after him. They consist many flat discs shaped sac or cisternae of 0.5 micrometer to 1 micrometer diameter. These are stayed parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentric, arranged near the nucleus with distinct convex sheaths or forming phase and concave or maturing phase. These sheath phase transphase organelles are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging material to be delivered either in the intracellular target or secreted outside the cell wall. Material to be packaged or form a cycle from endoplasmic reticulum fused with the sheath phase of Golgi apparatus and move toward the maturing phase. This explains why Golgi apparatus remain in the close association with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in cisternae of Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Eight 8.5.3.4 vacuoles. The vacuole is a membrane-bound space found in cytoplasm. It contains water, sap, excretive product, and other materials not useful for cell. The vacuole is bound by single membrane called tonoplast. In plant cell, The vacuole can occupy it up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of number of ions and other material against concentration gradient into vacuole. Hence, their concentration is significantly higher in vacuole than in cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion in many cells, as its protist food vacuole are found by engulfing food particles. 8.5.4 Mitochondria. Mitochondria are, unless specifically stated, are not easily visible under microscope. The number of mitochondria per cell is variable depending on physiological activity of the cell. In terms of shape, size also considered degree of variability is observed. Typically, the saucer shape cylindrical having diameter 0.1 to 1.0 micrometer, average 0.5 micrometer. The length 1 to 4.1 micrometer. Each mitochondria is double membrane bound structure with outer membrane and inner membrane, dividing its element distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of organelles. The inner membrane forms the number of infolding called the cristae or crista toward the matrix. The crista increase the surface area. The two membranes have their own specific enzyme associated with mitochondrial function. Mitochondria are the site aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule of few RNA molecules, the ribosomes and the components required for the synthesis of protein. The mitochondria divide by fission. Here is the structure of mitochondria: longitudinal section, outer membrane, inner membrane. It is matrix. It is this intermembrane space, and this is cristae. 8.5.5 Plastid are found in all plant cells and euglenoids. These are easily observed under the microscope as they are large and they bear some specific pigment, thus imparting specific color to the plant based on the type of pigment. Plastid can be classified in chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat soluble carotenoid pigment like carotene, anthophylls, and other pigments. This gives part yellow, orange, and red color. The leucoplast are colorless plastid, varied shapes, size, and store nutrients. Amyloplast store carbohydrate, starch, that is potato. Elaioplast store oils and fat, whereas aleuroplast store proteins. Majority of the chloroplasts of green plants are found in mesophyll cells of the leaves. These are lens-shaped, oval, spherical, squared, and even ribbon-like organelles having variable length 5 to 10 micrometer, width 2 to 4 micrometer. Their number varies from 1 per cell to 20 to 14 per cell in mesophyll, 1 in chlamydomonas. Like mitochondria, chloroplasts are also double membrane bound to the inner chloroplast. The membrane is relatively less permeable. The space limiting between inner and chloroplast called stroma. A number of organized flattened membranous sacs called thylakoid are present in the stroma. These thylakoid are arranged in stalk-like piles of coin called grana. Singular granum or the integral thylakoid. In addition to there are flat membrane tubular called stroma lamella connecting the thylakoid of different grana. The membrane of the thylakoid includes the space called lumen. The stroma of the chloroplast contains enzyme required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecule and ribosomes. Chlorophyll pigments are present in the thylakoid. The ribosomes of the chloroplast are smaller 70s than cytoplasmic 80s. 8.5.6 Ribosome. Ribosomes are granular structure first observed under the electron microscope in dense particular gel palette in 1953. They are composed of ribonucleic acid, RNA, and protein, are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and small subunit. The two subunit 80s and ribosome 60s and 40s while that of 70s ribosome and are 50s and 30s. The S is Swedberg unit, stands for sedimentation coefficient. It is indirectly measure of density and size. Both 70s and 80s ribosome are composed of two subunits. 8.9 Ribosome. Large subunit, small subunit. These combine to form a ribosome. 8.5.7 Cycloskeleton. An elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilament, and intermediate filament present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell involved in many functions such as mechanical support, motility, maintenance of shape of cells. Section of cilia and flagella showing different part. A. Electron micrograph. B. Diagrammatic representation of internal structure. This is diagrammatic representation of internal structure of cytoskeleton. Here is central microtubule. This is interdoublet bridge. This is peripheral microtubules doublet, plasma membrane. This is radial spoke, and this is central sheath. 8.5.8 Cilia and flagella. Cilia, that is singular cilium, and flagella, that is flagellum, are hair-like outgrowth of cell membrane. Cilia are small structures which work like oars, carrying the movement of either the cell or surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscope studies the cilium and flagella shows that they are covered with plasma membrane. Their core, called exonyme, possesses the number of microtubules running parallel to the long axis. The exonyme usually has nine doublet or of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exonymal microtubules is referred to as 9+2 array. Figure 8.10. Here is 9 plus 2, 1, 2, 3, 4, 5, 6, 7, 8, 9 plus 2. 9 plus 2 array. The central doublet are connected by bridge, is also enclosed by central sheath, which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are nine radial spokes. The peripheral doublet also interconnected by linkers. Both the cilium and flagella emerge from centriole-like structure called basal bodies. 8.5.9 Centrosomes and centriole. Centrosome is the organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphophorus pericentriolic material. Both the centrioles are in the centrosome lie perpendicular to each other, in which each has an organization like cardwheel. They are made up of 9 evenly spaced peripheral fibril tubulin protein. Each of the peripheral fibril triplet, the adjacent triplets are also linked. The central part of the proximal region of centrioles is also proteinous and called hub. which is connected by tubules of the peripheral triplets called radial spoke made of protein. These centrioles form basal body of cilia or flagella and spindle fiber that give rise to spindle apparatus during cell division and animal cell. 8.5.10 Nucleus. Nucleus as the cell organism was first described by Robert Brown as early 1831. Later, the material of nucleus stand by the basic dyes was given by name chromatin by Fleming. The interphase nucleus of the cell, when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one of the more spherical bodies called nuclei, singular nucleolus. Electron microscope has revealed that the nuclear envelope, which consists of two parallel membranes with space between 10 to 15 nanometer, called perinuclear space, form the barrier between the materials present inside the nucleus and that of cytoplasm. The outer membrane is usually remain continuous with the endoplasmic reticulum and also bear ribosomes on it. Structure of nucleus: nucleoplasm, nucleolus, nuclear pore, and nuclear membrane. At a number of places, the nuclear envelope interrupted by minor pores, which are formed by fusion of two membranes. These nuclear pores are passages through which movement of RNA and protein molecules take place in both directions between nucleus and cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect name of organisms that have more than one nucleus per cell? Some mature cells even lack the nucleus. Erythrocytes. Many mammals have sheep tube cell, vascular plants. Would you consider these cells are living? The nuclear matrix or the nucleoplasm containing the nucleolus and the chromatin. The nuclei are spherical structure present in the nucleoplasm. The content of nucleolus continuous with the rest of the nucleoplasm, as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nuclei are present in the cell, actively carrying out protein synthesis. You may recall that interphase nucleus has a large and indistinct network of nucleoprotein fiber called chromatin. But during different stages of cell division, cells show structural chromosome in place of the nucleus. Chromatin contain DNA and some basic protein called histones. Some histones proteins and also RNA. A single human cell has approximately 2 meter long thread DNA distributed among 46 chromosomes. You will study the detailed DNA packaging in the form of chromosomes in class 12. Every chromosome is visible only in dividing cell essentially, and primary constriction or centromere on the side which disc-shaped structure called kinetochores are present. Centromere hold two chromatids of chromosomes. Based on the position of the centromere, the chromosomes can be classified into four types. The metacentric chromosomes have middle centromere forming two equal arms of the chromosomes. The submetacentric chromosome has centromere slightly away from the middle of the chromosomes, resulting to one shorter and one arm longer. In case of acrocentric chromosomes, the centromere is situated close to the end, forming one extremely short and other very long, whereas telocentric chromosome terminal centromere. Type of chromosome based on the position of centromere. Here is metacentric, submetacentric, acrocentric, and this is telocentric. Sometimes a few chromosomes have non-standing secondary constriction at the constant location. This gives appearance of a small fragment called satellite. 8.5.1.11 Microbodies. Many membrane-bound minor vesicles called microbodies that contain various enzymes are present in both plant and animals. Summary: microbodies are membrane bounds and present in both plant and animals. Summary: all organisms are made up of cell and aggregates of cell. Cells vary in their shape, size, and activities. Function based on the presence and absence of membrane-bound nucleus and other organelles. Cells and organisms can be named eukaryotic and prokaryotic. A typically eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates the transport of several molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. All the cell organelles perform different but specific functions. Centrosomes and centrioles form the basal body of cilia and flagella that facilitate locomotion. In animal cell, centriole also forms spindle apparatus during cell division. The nucleus contains nuclei and chromatin network. It not only controls the activities of organelle but also play a major role in heredity. Endoplasmic reticulum contains tubular or cisternae. There are also two types, rough and smooth. Endoplasmic reticulum helps in the transport of substances, synthesis of proteins, lipoproteins, and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The secretion of cells are packed in them and transported from the cell. Lysosomes are single membrane structure containing enzyme for digestion of all type of macromolecules. Ribosomes are involved in protein synthesis. These occur in freely and in cytoplasm are associated with endoplasmic reticulum. Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphates. They are bound by a double membrane, and the outer membrane is smooth and inner membrane into several cristae. Plastids are pigment containing organelle found in plant cell only. Plant cell chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reaction and the stroma dark reaction. The green colored plastid are chloroplast which contains chlorophyll, whereas the other colored plastid are chromoplast which may contain pigment. Like carotene and xanthophyll, the nucleus is enclosed by nuclear membrane, the double membrane structure with nuclear pores. The inner membrane is included with nucleoplasm and the chromatin material. Thus, cells are structural and functional unit of life. So, we have just read cell unit of life. सो बायोमोलिक्यूल्स को कंप्लीट करेंगे उसके पहले 2 मिनट अपन 3 मिनट का थोड़ा ब्रेक लेते हैं भाई लोग, ठीक। 5:09 AM हो रहा, हम लोग रेस्ट लेंगे 5:15 तक का, ओके। रेस्ट फॉर 6 मिनट। हम होंगे कामयाब एक दिन, हम होंगे कामयाब, हम होंगे कामयाब, हम होंगे कामयाब एक दिन। सो गाइस, हम होंगे कामयाब एक दिन, जरूर होंगे भाई लोग कामयाब। तो यार एक है, क्या बोलते हैं, अभी टाइम है ना अपने पास, 2 मिनट है, तो कुछ थोड़ा अच्छी बातें कर लेते हैं। तो बोल रहा था अपने ऊपर भी भरोसा करना सीखना चाहिए, ठीक। ऐसा क्यों बोल रहा हूँ, क्योंकि सहारे हमेशा नहीं रहते। किसी पे डिपेंडेंट रह के अपने जीवन में नहीं जियो, समझे। या किसी पे इतना भरोसा नहीं करो, इसलिए क्योंकि तुम्हारे पास जो भी सहारा है ना, जो भी सहारा लग रहा है कि कुछ नहीं रहने वाला है, लास्ट में तुम ही बचोगे, कोई नहीं आएगा साथ देना। इसीलिए खुद पे भरोसा रखो, संघर्ष करो, और क्या, लगे रहो लाइफ में, स्ट्रगल करो, हसल करो। आज नहीं तो कल हो ही जाओगे कामयाब। स्ट्रगल और हसल करना नहीं छोड़ना मेरा भाई, कुछ भी हो जाए। चलो चैप्टर 9 बायोमॉलिक्यूल्स। इसमें क्या क्या पढ़ेंगे अपन। पहले 9.1 हाउ टू एनालाइज केमिकल कंपोजिशन, 9.2 प्राइमरी एंड सेकेंडरी मेटाबोलाइट, 9.3 बायोमॉलिक्यूल्स, बायोमैक्रोमॉलिक्यूल्स, 9.4 प्रोटीन्स, 9.5 पॉलीसैकेराइड्स, 9.6 न्यूक्लिक एसिड, 9.7 structure of protein, 9.8 nature of bond linking monomers of in polymers, 9.9 dynamic state of body constituents, concept of metabolism, 9.8 metabolic basis for living, 9.11 the living state, 9.12 the enzymes. So let's go. There is a wide diversity of living organisms in our biosphere. Now, a question that arises in our mind is: are all living organisms are made up of same chemicals? That is, elements and compounds. You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on plant tissue, animal tissue, and microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, several others, and their respective content per unit mass of living tissue. If the same analysis is performed on a piece of earth crust, as an example of non-living matter, we obtain a similar list. What are differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon, hydrogen with respect to other elements is higher in any living organism than in Earth's crust. 9.1 How to analyze chemical composition. We can continue asking the same way: what type of organic compounds are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or piece of liver, etc., and grind it in a trichloroacetic acid, Cl3C-C11OH, using a mortar and a pestle. We obtain a thick slurry. If we were to stain this through cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, and other or more technically the acid soluble pool, and second the retentant or acid insoluble fraction. Scientists have found thousands of organic compounds in acid soluble pool. In higher classes, you will learn how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extract of organic compound then subject the extract of various separation technique till one has separated a compound from all other compound. In other words, one can isolate and purify a compound analytically technique. When applied to compound, given us an idea of molecular formula and the probable structure of a compound. All the carbon compound that we get from living tissue can be called biomolecules. However, living organisms have also got inorganic elements and compound in them. How do we know this? A slightly different but destructive experiment had to be done. One weight a small amount of living tissue, say a leaf or liver, and this is called wet dry and dried. All the water evaporates, the remaining material gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gases from CO2 water vapors and removed. What is remaining is called ash. This ash containing inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in acid soluble fraction. Therefore, elemental analysis give elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compound give an idea of the kind of organic and inorganic constituents present in living tissue. From a chemistry point of view, one can identify functional group like aldehyde, ketone, aromatic compound, etc. But from biological point of view, we shall classify them into amino acid, nucleotide, base, fatty acid, etc. Amino acid are inorganic compound containing an amino group and an SD group as substituent on the same carbon, the alpha carbon. Hence, they are called alpha amino acid. They are substituted methane. There are four substituent group occupying the 4 valency position. These are hydrogen, carboxyl group, amino group, and variable group designated as R. Based on the nature of R group, there are many amino acids. However, those which occur in protein are only 20 type. The R group in this proteinous amino acid could be hydrogen. The amino acid called glycine, a methyl group, alanine, hydroxyl, methyl, serine. 3 of 20 are shown in figure. A comparison of elemental elements present in non-living and living matter. Element percentage of weight in Earth's crust and human body. हाइड्रोजन, तो इसमें याद कर लो ऑक्सीजन सबसे ज़्यादा है अर्थ में भी और ह्यूमन बॉडी में भी. ठीक, ह्यूमन बॉडी में सेकेंड नंबर पे आता है कार्बन, बट अर्थ क्रस्ट में सेकेंड नंबर पे आता है सिलिकॉन. ह्यूमन बॉडी नेग्लिजिबल है सिलिकॉन, बट इसमें बहुत सारा लिस्ट ऑफ रिप्रेजेंटेटिव इनऑर्गेनिक कॉन्स्टिट्यूएंट इन लिविंग टिश्यू. तो कंपोनेंट फार्मूला सोडियम, पोटेशियम, कैल्शियम, मैग्नीशियम और वाटर और कंपाउंड NaCl, CaCO3, फॉस्फेट आयन, सल्फेट आयन अपने बॉडी में पाया जा रहा है. ठीक, the chemical and physical property of amino acid are essentially of amino, carboxylic and R functional group. Based on number of amino and carboxyl group, there are 80 glutamic acid, basic lysine and neutral valine in amino acid. Similarly, there are aromatic amino acid, tyrosine, phenyl, alanine, tryptophan. A particular property of amino acid is ionizable nature of NH2 and C-double-bond-OH group. Hence, in solution of different pH, the structure of amino acid changes. B is called Jeter form. Here we can see plus and minus both contain the same compound, so we got zwitterine. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxylic group attached to R group, alkyl group. The alkyl group could be methyl or ethyl or higher number of CH2 group 1 to 19 carbons. For example, palmitic acid has 16 carbon, including carboxylic carbon. Arachidonic acid has 20 carbon atoms, including the carboxylic acid. Fatty acid could be saturated without double bond or unsaturated with one or more double bonds. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acid. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglyceride and triglyceride. These are also called fats. And animal based on melting point oils have lower melting points. Gingerly oil and hence remain oil in winter. Can we identify a fat from the market? Some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissue, especially neural tissue, have lipid with more complex structure. Living organisms have a number of carbon compounds in which heterocyclic ring can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cysteine and nucleoside. Adenylate acid, thymidylate acid, and guanylic acid, uridylic acid, and cystidylic acid are nucleotides. Nucleic acid, like DNA and RNA, consist of nucleotides only. DNA and RNA functions as genetic material. Sugars, carbohydrates, amino acids, sugar, glucose, ribose C5H10O5, ribose, glycine contain hydrogen, alanine contain methyl, and serine contains CH2OH. Palmitic acid, fatty acid, glycerol, triglyceride, R1, R2, R3 are fatty acids. Phospholipid, lecithin, cholesterol, fats and oils and lipids. Diagrammatic representation of a small molecular weight organic compound of living tissue. Nitrogenous base, nucleosides, nucleotides. Nitrogenous base: adenine, purine, pyrimidine, uracil, adenosine, uridine, adenylate acid. 9.2 Primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure, if possible synthesizing them. If one were to make a list biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissue, one notices the presence of all such categories of compounds, such in figure 9.1. These are called primary metabolites. However, when one analyzes plants, fungal and microbial cells, One would see thousands of compound other than this. These called primary metabolites, that is alkaloids, flavonoids, rubber essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. While primary metabolites have identifiable functions and play known roles in normal physiological processes, we do not at the moment understand the role of functions of all the secondary metabolites in the host organism. However, many of these useful in human welfare. Rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance. The later chapter years will learn about this. Some secondary metabolites, pigments, carotenoids, anthocyanins, alkaloid, morphine, codeine, and etc. Terpenoids, monoterpene, diterpene, essential oil, lemongrass oil, etc. Toxin, heparin, ricin, lectins, canacolwelin, A, drugs, vinblastine, curcumin, polymeric substance, rubber gum, and cellulose. 9.3 Biomacromolecules. There are one of feature of common to all those compounds found in acid soluble pool. They have molecular weight ranging from 18 to around 800 Dalton approximately. The acid insoluble fraction has only 4 types of organic compounds, that is protein, nucleic acid, polysaccharide, and lipids. This class of compound, with the exception of lipids, have molecular weight in the range of 10,000 Dalton to above. For this very reason, biomolecules, that is biochemical compounds found in living organisms, are of two types. One of those which have molecular weight less than 1,000 Dalton, and other usually referred to as micromolecules or simply biomolecules, while those which are found in acid insoluble fraction are called macromolecules, biomacromolecules. The molecules in the soluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids whose molecular weight do not exceed 800 Dalton come under acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight. Compounds are present not only as such, but also arranged into structure like cell membrane and other membrane. When we grind tissue, we are disrupting the cell structure. Cell membrane and other membrane are broken into pieces and form vesicles, which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with acid insoluble pool, and hence in macromolecular fraction. Lipids are not strictly macromolecules. The lipid soluble pool represents roughly the cytoplasmic composition. The macromolecules form cytoplasm, and organelles become the acid insoluble fraction. Together represent the entire chemical composition of living tissue of organism. In summary, if we represent the chemical composition of living tissue from abundant point of view and arrange them in class wise, we observe that water is the most abundant chemical in living organisms. 9.4 Average composition of cell component percentage of total cellular mass. Water 70 to 90%. Proteins 10 to 15%. Carbohydrate 3%. Lipid 2%. Nucleic acid 5 to 7%. Ions 1%. 9.4 Proteins. Proteins are polypeptides. They are linear chains of amino acid linked by peptide bond as shown in figure 9.3. Each protein is polymer of amino acid, as there are 20 types of amino acid, that is alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer, repeating a n number times. This information about the This information about amino acid content is important, and later in your nutrition lesson you will learn certain amino acids are essential for our health and have to be supplied through our diet. Hence, dietary protein are the source of essential amino acid. Therefore, amino acid can be essential or non-essential. Therefore, latter are those which our body makes, while we get essential amino acids through our diet. Food. Proteins carry out many functions, are living organisms. Some transport nutrients across the membrane, some fight infection organisms, some hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world. The ribulose, biphosphate, carboxylase, oxygenase, rubisco is the most abundant protein in the whole of the biosphere. Some protein and their functions. Protein functions. Collagen, intercellular ground substance. Trypsin, enzyme. Insulin, hormone. Antibody, fight infection agent. Receptor, sensory reception, smell, taste, hormone, etc. GLUT4 enable glucose transport into cell. (9.5) Polysaccharides. The acid insoluble pellet also has a polysaccharide. Carbohydrate as another class of macromolecules. Polysaccharides are long chain of sugars. They are threads in literally a cotton thread containing a different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharides, that is glucose. Cellulose is a homopolymer. Starch is a variant of this, but present as a storehouse in energy of plant tissue. Animals have another variant called glycogen. Insulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. It has branches shown in the form of cartoon. Starch form helical secondary structure. In fact, starch can hold I2 molecule in the helical portion. The starch I2 is blue color. Cellulose does not contain complex cells and hence cannot hold I2. Diagrammatic representation of glycogen. Plant cell wall is made of cellulose. Paper made up of plant pulp and cotton fiber is cellulose. There are more complex polysaccharides in nature. They have a building blocks, amino acids, amino sugars, and chemically modified sugars. That is glucosamine and galactosamine, etc. Exoskeleton of arthropod, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acid. The other type of macromolecule that we would find in the acid insoluble fraction of living tissue is the nucleic acid. These are polynucleotide together with polysaccharide and polypeptide. These comprise the true macromolecular fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is heterocyclic compound, the other is monosaccharide, and the third is phosphoric acid or phosphate. As you notice the figure 9.1, the heterocyclic compound in a nucleic acid are nitrogenous bases named adenine, guanine, uracil, and cytosine and thymine. Adenine and guanine are substituted purines, while the rest are substituted pyrimidine. The skeletal heterocyclic ring is called purine, the pyrimidine, respectively. The sugar found in polynucleotide is either ribose or monosaccharide pentose or 2' deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that contains ribose is called ribonucleic acid RNA. Structure of protein. Protein means are in heteroform containing string of amino acid. Structure of molecule means different thing in different context. In inorganic chemistry, the structure invariably refer to the molecular formula NaCl, MgCl2, etc. The organic chemists always write the two-dimensional view of biomolecules while representing the structure of molecule, that is benzene, naphthalene, etc. Phase conjure of three-dimensional views of molecular structure, while biologists describe the protein structure at four levels. The sequence of amino acid, the positional information in protein, which is the first amino acid, which is second, and so on, is called primary structure of protein. A protein is imagined as a line. The left end is represented by first amino acid, and the right end represented by last amino acid. The first amino acid is also called an terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein thread does not exist throughout as extended right rod. The thread is folded in the form of helix, similar to revolving staircase, or course. Only some portion of the protein thread are arranged in the form of a helix. In proteins, only right-handed helices are observed. Other regions of the proteins involved in prime folded into other forms in what is called the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to tertiary structure. This gives us three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of protein. Some proteins are an essential or more than one polypeptide or subunits. The manner in which these individual polypeptide or subunits are arranged with respect to each other, that is linear string of spheres arranged one upon each other in the form of a cube or plate, etc., is the architecture of a protein, otherwise called quaternary structure of a protein. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other. Hence, the 2 substituent alpha and 2 substituent beta type together constitute the human hemoglobin HB. Primary, secondary structure: alpha helix, beta helix. Tertiary structure contain hydrogen and disulfide bond. And quaternary structure, various level of structure. Cartoon showing our secondary and tertiary protein. 9.8 Nature of bond linking to monomer of polymer. A polypeptide or protein amino acid are linked by peptide bond, which is formed when carboxylic acid or OH group of amino acid react with amino group of next amino acid with the elimination of water via a process called dehydration. In polysaccharide, the individual monosaccharide are linked by a glycosidic bond. This bond is also formed by dehydration. This bond is formed between two carbon atoms of two adjacent monosaccharides. In a nucleic acid, phosphate moiety linked to 3' carbon of one sugar and one nucleotide to form 5' carbon of sugar of succeeding nucleotide. The bond between phosphate and hydroxyl group of sugar is an ester bond. As there is one such structure, ester bond on either side, it is called phosphodiester bond. Nucleic acid exhibit a wide variety of secondary structure. For example, one of the secondary structure exhibited by DNA is the famous Watson-Crick model. This model says DNA exists as a double helix. The two strands of polynucleotides are antiparallel trunked on opposite direction. The backbone of formed sugar phosphate sugar chain. The nitrogen has a projected more or less perpendicular to this backbone but face inside. A and G of up to one strand compulsorily base pairs with T and C respectively on the other strand. Diagram indicating secondary structure of DNA. Guanine, cytosine, thymine, adenine. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds into G and C. Each strand appears like a helical staircase. Each step ascent is represented by a pair of bases. At each step of ascent, the strand turns 36 degrees. One full turn of the helical strand would involve 10 steps or 10 base pairs. Attempt drawing the line diagram. The pitch would be 34 angstrom. This size per base pair is 3.4 angstrom. This form DNA above mentioned silent feature called B DNA. In higher classes, you would told that more than dozen form of DNA named after English alphabet with unique structure of feature. 9.9 Dynamic state of body constituent concept of metabolism. What we have learned till now is that living organism, be it simple or bacterial cell or protozoans or plant or animal, contain thousands of organic compounds. These compounds or biomolecules are present in certain concentration expressed as moles cells or moles per liter. One of the greatest discoveries ever made by observation, all biomolecules have turnover. This means they have constantly been changed into some other biomolecules and made from some other biomolecules. This breaking and making is through chemical reaction constantly occurring in living organism. Together, all these chemical reactions are called metabolism. Each of these metabolic reactions result in the transformation of biomolecules. A few examples of such metabolic transformation are removal of CO2 from amino acid, making an amino acid into an amine, removal amino acid group in a nucleotide base, hydrolysis of glycosidic bond in disaccharide, etc. We can list 10 and thousands of such examples. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reaction. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathway. These metabolic pathways are similar to automobile traffic in a city. These pathways are either linear or circular. These pathways are crisscross each other, hence traffic jam. Flow of metabolites through metabolic pathway are different at end direction, like automobile traffic. This metabolic flow is called dynamic state. A body constituent. What is important to learn that interlinked metabolic traffic is very smooth and without single reported mishap. Healthy condition. Another feature of these metabolic reaction is that every chemical reaction is catalyzed reaction. There is no uncatalyzed metabolic conversion in the living system. Even CO2 dissolving in water, a physical process, is a catalyzed reaction in living organism. The catalyst which hastens the rate of given metabolic conversion are also proteins. These proteins is catalytic power are named enzymes. Metabolic basis of living for living. Metabolic pathway can lead to more complex structure from simpler structure. For example, acetic acid become cholesterol or lead to simpler structure from complex structure. For example, glucose become lactic acid in our body skeletal system muscle. The former cases are called biosynthetic pathway or anabolic pathway. The latter constitute degradation and hence called anabolic pathway. Anabolic pathway is expected consume energy. Assembly of protein from amino acid require energy input. Out of the other hand, catabolic pathway led to release energy. For example, when glucose is degraded for lactic acid in our skeletal muscle, energy is liberated in this metabolic pathway from glucose to lactic acid, which occur in 10 metabolic steps, is called glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bond. As and when needed, this bond energy is utilized for biosynthetic, osmotic, and mechanical work that we perform. The most important form of energy currency is living system in the body energy in the chemical called adenosine triphosphate ATP. How do living organisms derive their energy? What strategies they have they evolved? How do they store this energy and in what form? How do they convert in this energy into work? You will study understand all this under a sub-discipline called bioenergetics later in higher classes. The living state. At this level, you must understand that tens and thousands of chemical compounds and living organisms, otherwise called metabolites and biomolecules, are present concentration and concentration characteristics of each of them. For example, the blood concentration of glucose in normal human is individual 4.2 millimole per liter to 6.1 millimole per liter, while that hormone would be nanogram per ml. 4.2 ml to the most important fact in biological system is that all living organisms in steady state characterized by concentration of each of these biomolecules. These biomolecules are in metabolic flux. Any chemical or physical process moves spontaneously at equilibrium. The steady state is a non-equilibrium state. One should remember from the physics that system at equilibrium cannot perform work. As living organisms work continuously, they cannot afford to reach equilibrium. Hence, the living state is non-equilibrium steady state to be able to perform work. Living process is of constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolism provides mechanism for the production of energy. Hence, the living state and metabolism synonyms. Without metabolism, there cannot be living state. Enzyme. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These enzymes are called ribozymes. One can depict the enzyme by line diagram. An enzyme, like any protein, has a primary structure that is amino acid sequence of protein. An enzyme, like any protein, has a secondary and tertiary structure. When you look at certain structure, you will notice that the backbone of protein chain folds upon itself. The chain crisscrosses itself, and hence many surfaces or pockets are made. One such pocket is a factor active site. An active site of enzyme is surfaces or pocket into which the substances fit. The enzyme through their active site catalyze reaction at a higher rate. Enzyme catalyst differ from an inorganic catalyst in many ways, but one major difference needed to mention: inorganic catalysts work efficiently at high temperature and high pressure, while enzymes get damaged at high temperature and at 40 degrees Celsius. However, enzymes are isolated from organisms who normally live under extremely high temperature, that is, hot winds and sulfur springs are stable and contain retain their catalytic power even at high temperature, up to 80 to 90 degrees Celsius. Thermal stability is thus important quality for such enzyme isolated from thermophilic organism. 9.1 12.1 Chemical reaction. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compound undergoes two types of changes: a physical change, simply referred to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water, or when water becomes a vapor, there are also physical changes process. However, when bonds are broken and new bonds are formed during transformation, this will be called chemical reaction. For example, BOH का hold to H2SO4, ये बना रहा BSO4. In inorganic chemical reaction, similarly hydrolysis of starch into glucose and organic chemical reaction rate. Physical chemical process amount of product formed per unit time. It is expressed rate equal dP by dt. Rate can also be called a velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature, among other factors. A general rule of thumb is that rate double or decrease by half. For 10 degrees Celsius, change in either direction catalyze the reaction process at rate vastly higher than that of uncatalyzed one. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same uncatalyzed reaction. For example, carbon dioxide H2O, carbonic anhydrous. In the absence of any enzyme, this reaction is very slow, with about 200 molecules H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speed dramatically, with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzyme incredibly indeed. There are 1,000 types of enzyme catalyzing the unique chemical catabolic reaction, a multi-step reaction. Chemical reaction, when each step catalyzed by same enzyme complex of different enzymes, is called metabolic pathway. For example, glucose to pyruvic acid. The actually metabolic pathway is glucose become pyruvic acid through different process catalyzed by metabolic reaction. When you study inspiration in chapter 14, you will study this reaction at this stage. You should know that this very metabolic pathway with one or two additional reactions give rise to a variety of metabolic end product in our skeletal system, muscle system. under anaerobic under anaerobic condition, lactic acid form. Under normal aerobic condition, pyruvic acid formed in yeast. During fermentation, the same pathway led to the production of ethanol. Hence, the different condition, different product are possible. 9.12.2 How do enzymes bring about such high rate of chemical conversion? To understand this, we should study enzyme a little more. We have already understood the idea of an active site. The chemical of or metabolic conversion refer to reaction. The chemical which is converted into product is called substrate. Hence, enzyme protein with three-dimensional structure including active site converts substrate into product. Symbolically, it is now understood that substrate has to bind to enzyme at an active site within the given cleft or pocket. The substrate has diffused toward the active site. There is thus an obligatory formation of an enzyme substrate complex. Enzyme stand for enzyme, E stand for enzyme, and this complex formation is a transient phenomenon. During the state where substrate is found to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into structure of product. Yes, the pathway of this transformation must go through the so-called transition state structure. There could be many more altered structural state between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structure states are unstable. Stability of some is something related to energy status of molecule or structure. Hence, when we look at this pictorial through a graph, it looks like something as in figure 9.6. Concept of activation energy, progress of reaction, potential energy. Here is substrate activation energy with enzyme, activation energy without enzyme, transition state, and product. The y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or states through the transition state. You would notice two things: the energy level difference between S and P. If P is the lower than level than S, then the reaction is exothermic reaction. No one need not to supply energy by heating it in order to form product. However, whether it is exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the gas has to go through much higher energy state or transition state. The difference in average energy content of gas or from that this transition state is called activation energy. Enzyme eventually bring down this energy barrier, making the transition S and P more easy. 9.12.3 Nature of enzymes. Enzyme action. Each enzyme E has a substrate as binding site in its molecule, so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociated into its product and unchanged enzyme with intermediate formation enzyme product complex EP. The formation of enzyme substrate complex is essential for catalysis. The catalytic cycle of an enzyme action can be described in the following steps. First, substrate bind to the active site of enzyme feeding into the active site. Second, the binding of the substrate induces the enzyme to alter its shape, feeding more tightly around the substrate. Third, the active site of the enzyme now is close proximity to the substrate, break the chemical bond and substrate, and the new enzyme product complex is formed. 4. The enzyme release the product and the reaction, and the free enzyme is ready to bind another molecule of the substrate and run through the catalytic cycle once again. 9.12.4 Factors affecting enzyme activity. The activity of enzyme can be affected by changing condition, which can be altered by tertiary structure of protein. These include temperature, pH change in substrate concentration, and binding of a specific chemical that regulate the activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Figure 9.7. Each enzyme shows its highest activity at particular temperature and pH, called optimum temperature and optimum pH. Activity decline below and above the optimum value. Low temperature preserves the enzyme and temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. 9.7 Effect of change in first pH, second temperature, second concentration of substrate enzyme activity. If you increase pH, then at optimum pH it's so maximum activity. If we increase more than optimal pH, then the reactivity decreases, same as temperature. And here is substrate velocity max of reaction. This is km value when velocity of reaction become half of maximum velocity. Concentration of substrate. With increase in substrate concentration, the velocity of enzymatic reaction rises at first. The reaction ultimately reaches the maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecule, and after saturation of these molecules, there are no free enzyme molecule to bind with additional substrate molecule. The activity of enzyme is also sensitive to the presence of species chemicals that bind to the enzyme. When the binding of chemicals shuts off enzyme activity, the process called inhibition, and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in the molecular structure and inhibit the activity of enzyme, it is known as competitive inhibitor. And due to close of structural similarity with the substrate, the inhibitor competes with the substrate for substrate binding site of enzyme. Consequently, the substrate cannot bind, and result the enzyme action decline. That is inhibition of succinic dehydrogenase by malonate, which closely resemble the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogen. 9.12.5 Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses, and named according to 4-digit numbers. First, oxidoreductase or dehydrogenase. So here is trick to learn all these groups. And trick is oath lil, oxidoreductase, transferase, hydrolase, liases, and isomerases, and ligases. So oxidoreductase, enzyme which catalyze oxidoreduction between two substrates S and S Example: S reduce, S oxidize, S oxidize, and S reduce. This perform something like redox reaction, means this oxidize and reduce both in same reaction. Transferase, enzyme catalyzing transfer of group G group other than hydrogen between a pair of substrate S and etc. Hydrolysis, enzyme catalyzing hydrolysis of ether peptide glycosidic carbon halide and PN bond, means phosphorus and nitrogen bond. Lysis, enzyme that catalyze removal of group of substrate by mechanism other than hydrolysis, leaving the double bond. In lyase, double bond leave the leaving carbon double bond. Isomerase include all enzyme catalyzing interconversion, optical, geometrical, and positional isomerism. Ligase, enzyme catalyzing linking together two compounds, that is enzyme which catalyze joining of carbon oxygen, carbon sulfur, carbon nitrogen, and phosphorus oxygen etc. bond. 9.12.6 cofactor, enzymes are composed of one or several polypeptides, and however there are number of cases in which non-protein constituent called cofactor are bound to enzyme to make enzyme catalytically active. In these instances, the protein portion of enzyme is called apoenzyme. Three kinds of cofactor may be identified: prostatic group coenzymes and metal ion. Prostatic groups are organic compounds and are distinguished from other factors in that they are tightly bound to the apoenzyme. For example, peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prostatic group and is part of active site of enzyme. Coenzymes are also organic compounds, but their association with apoenzyme is only transient, usually occurring during course of catalysis. Furthermore, coenzymes serve as cofactor and a protein and a number of different enzyme catalyzed reactions. The essential chemical component of many coenzymes are vitamins, that is coenzyme nicotinamide adenine dinucleotide NAD and NADP contain vitamin niacin. A number of enzymes required for metal ion for their activity, which form a coordination bond with side chain at active site and at the same time form one or more coordination bond with substrate, that is zinc is a cofactor for proteolytic enzyme carboxypeptidase. Catalytic activity lost when cofactor is removed from the enzyme, which testified that play a crucial role in catalytic activity of enzyme. Summary: although there is bewildering diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. The elemental composition of living tissue and non-living matter appears also to be similar when analyzed qualitatively. However, a closer examination revealed that the relative abundance of carbon, hydrogen, and oxygen is higher in living system when compared to inanimate matter. The most abundant chemical in living organism is water. There are thousands of small molecular weight less than 1000 Dalton biomolecules, amino acid, monosaccharides and disaccharides, sugar, fatty acid, glycerol and nucleoside and nitrogen bases are some of the other organic compounds seen in living organisms. There are 20 types of amino acids, 5 types of nucleotides, fats, oils, glycerides, and in which fatty acids are esterified to glycerol. Phospholipid contain addition phosphorylated nitrogenous compounds. Only 3 types of macromolecules, that is protein, nucleic acid, and polysaccharide, are found in living system. Lipid, because of their association with membrane, separate in the macromolecular fraction, Biomolecules are polymers. They are made up of building blocks which are different. Protein are heteropolymers made up of amino acid, nucleic acid, RNA, and DNA, and are composed of nucleotides. Biomacromolecules have a hierarchy of structure: primary, secondary, tertiary, and quaternary. Nucleic acids serve as genetic material. Polysaccharides are components of cell wall and plants. In plants, fungi are also of the exoskeleton and arthropods. They also are a storage form of energy, that is starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes, some antibiotics, some receptors, some hormones, and some others are structural proteins. Collagen are most abundant protein in animal world, ribulose biphosphate carboxylase oxygen ribosco is most abundant protein in whole biosphere. Enzyme is protein which catalyze biochemical reaction, and cell ribosome the nucleic acid with catalytic power. Proteinaceous enzyme exhibits substrate specificity, required optimal temperature and pH for maximal activity. They are denatured at higher temperature. Enzyme lower activation, energy activation. It can enhance greatly rate of the reaction. Nucleic acid carry hereditary information that passes through parental generation progeny. सो भाई अपना कंप्लीट हुआ ये बायोमोलिक्यूल्स में। काफ़ी ठीक ठाक चैप्टर था वाले बायोमोलिक्यूल्स में, मतलब लेंथ ठीक है बायोमोलिक्यूल्स का। अब इसके बाद अपन को करना है क्या, सेल साइकिल एंड सेल डिवीजन। तो उसके लिए थोड़ा 5 मिनट रेस्ट करें क्या भाई। सो करेंगे इसके लिए हम 6:15 हो रहा है अभी, 6:20 इससे 1 सेकेंड 6:15 to 6:20 AM रेस्ट। सॉरी भाई इसके लिए रेस्ट करना हो। इसके बाद स्टार्ट करेंगे चैप्टर 10 सेल साइकिल एंड सेल डिवीजन। सो भाई लोग, टाइम हो गया, अपन आ भी गया। आए हैं तो तबाही मचाएंगे, आए हैं तो गलियों में तबाही मचाएंगे ही। सो लेट्स गो। तो ये सेल साइकिल स्टार्ट करने से पहले कुछ शायरी हो जाए क्या भाई लोग? तो कुछ इस तरह है। हुए बदनाम मगर फिर भी सुधर न पाए हम, फिर वही शराब, फिर वही इश्क, फिर वही तुम। वाह 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 वाह, कैसा लगा भाई लोग मेरा शायरी? फिर से सुनो। हुए बदनाम मगर फिर भी न सुधर पाए हम, वही शराब, वही इश्क, फिर वही तुम। अब इसी बात पे चलो करते हैं अपना सेल साइकिल स्टार्ट। समझ में आया तो अच्छी बात, नहीं आया तो समझ जाओगे टाइम आते आते। चलो चैप्टर 10, मेरे दिल के टुकड़े को। चैप्टर 10, सेल साइकिल एंड सेल डिवीजन। 10.1 सेल साइकिल, 10.2 मेटाफेज phase, 10.3 significance of mitosis and 10.4 meiosis, 10.4 significance of meiosis। चलो भाई स्टार्ट करते हैं, लेट्स गो ब्रो। Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organism। थोड़ा स्पीड बढ़ाते हैं भाई, सॉरी। Growth and reproduction are characteristic of cell, indeed of all living organisms. All cells reproduce by dividing into with each parental cell giving rise to two daughter cell each time they divide. These newly formed daughter cell can themselves grow and divide, giving rise to new cell population that are formed by growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. 10.1 Cell cycle. Cell division is a very important process in living organism. During cell division of cell, DNA replication and cell growth also take place. All these processes, cell division, DNA replication and cell growth, hence have to take place in coordinated way to ensure correct division and formation of progeny. Cells containing intact genome, the sequence of events by which a cell duplicates its genome, synthesize the other constituents of the cell, and eventually divide into two daughter cells, is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is continuous process, DNA synthesis occur only during one specific stage in the cell cycle. The replicated chromosome DNA are taken distributed to daughter nuclei by a complex series or event during cell division. These events are themselves under genetic control. 10.1.1 Phases of cell cycle, or typically eukaryotic cell cycle, is illustrated by human cell in culture. These cells divide once in approximately every 24 hours. However, the duration cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell in only about 90 minutes. This cell cycle is divided into two basic phases. First, interphase, second, M phase or mitosis phase. The M phase represents a phase when actual cell division or mitosis takes place, and the interphase represents a phase between two successive M phases. It is significant to note that 24 hours average duration of cell cycle in human cell. Cell division proper lasts for about an hour. The interphase lasts more than 95% duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is time during which cell is preparing for cell division, undergoing both cell growth and DNA replication in orderly manner. The interphase is divided into 3 phases: G1 gap 1 phase, ES phase synthesis phase, G2 gap 2 phase. Here is diagrammatic view of cell cycle indicating formation of two from one cell. EM phase, these are mitosis EM phase, and these are interphase G1S and G2. G1 phase is synthetic phase, and this is a G0 phase. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase of cell is metabolically active and continuously grow but does not replicate its DNA. ES or synthesis phase marks period during which DNA synthesis or replication take place. During this time, the DNA per cell doubles. If initial amount of DNA denoted as 2C, then it increases to 4C. However, there is no increase in chromosome number. If cell diploid are 2n chromosomes G1 phase, even after S phase, the number of chromosomes remain same 2n. In animal cell during S phase, DNA replication begin in the nucleus and central duplicate in the cytoplasm. During the G2 phase, protein and synthesize the preparation for mitosis while cell growth continues. How do plant and animal continue to grow all their lives? Do all cells in plants divide all the times? Do you think all the cells continue to divide in the all plant and animals? Can you tell the name and location of tissue having cells that divide all their lives? Higher plants, do animals have similar meristematic tissue? You have studied mitosis in onion roots tip cell. It has 16 chromosomes in each cell. You can have tell how many chromosomes some cells in adult animals do not appear exhibit division in that a heart cell, and many others cell divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exist in G1 phase to enter in an active stage called quiescent phase, G0 phase of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of organism. In animals, mitotic cell division is only seen in the diploid somatic cell. However, there are few exceptions of this where haploid cell divide by mitosis. For example, male honeybee. Again, this plant can show mitotic division in the form of diploid, haploid and diploid cell. From your recollection, example alternation of generation in plants, identify plant species and stage at which mitosis is seen in haploid cell. 10.1 EM phase. This is the most dramatic period of cell cycle involving the major reorganization of virtually all component of cell. Since number of chromosomes in parent progeny cell is the same, it is also called equational division. For convenience, mitosis have been divided into 4 nuclear division karyokinesis. It essentially to understand that cell division is a progressive process. Very clear cut line cannot be drawn. Various stages karyokinesis involve 4 stages: prophase, metaphase, anaphase, telophase. 10.1 2.1 prophase. Prophase, which is first stage of karyokinesis of mitosis, follows the S or G2 phases of interphase. In the S phase, G2 phase of new DNA molecules formed are not distinguishable. Intertwined prophase is marked by the initiation condensation of chromosomal materials. The chromosomal material become untangled during the process of chromatin condensation. The centromere, which had undergone duplication during S phase interphase, now begin to move toward the opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic. First, chromosome material condensed to form compact mitotic chromosomes. Chromosomes are seen to compact of two chromatids attached together at centromere. Centromere, which had undergone duplication during interphase, begin to move toward opposite poles of cell. Each centromere radiate out microtubules called ester. The two ester together with spindle fibers form mitotic apparatus. Cell at the end of prophase viewed under microscope do not show Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. 10.2.2 Metaphase. The complete disintegration of nuclear envelope marks the start of second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they cannot be observed clearly under microscope. This stage, which morphology of chromosome is most easily studied. In this stage, metaphase chromosomes is made up of two sister chromatids which held together by centromere. Small disc structure surface of centromere are called kinetochores. These structures serve as the site of attachment of spindle fibers formed from spindle fibers to the chromosomes that moved into position of center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie to the equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole to and its sister chromatid connected by its kinetochore spindle fiber from opposite pole. The plate of alignment of chromosomes at metaphase referred to metaphase plate, and the key feature of metaphase are given below. First, spindle fiber attached to kinetochore of chromosome. Second, chromosome move to spindle fiber equator and get aligned along metaphase plate through spindle fibers of both poles. Diagrammatic view of stage of mitosis. Here, early prophase, late prophase, and transition of metaphase. 10.2.3 Anaphase. At the onset of anaphase, each chromosome are arranged at the metaphase plate and split simultaneously, and two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration toward two opposite poles. As each chromosomes move away from the equatorial position, the centromere of each chromosome remain directed toward the pole and hence at the leading edge. Means centromere of chromosome is a leading edge, with arm of chromosome is trailing behind. Thus, anaphase stage is characterized by the following event. First, centromere is split and chromatids separate. Second, chromatids moves to opposite poles. 10.2.4 Telophase. At the beginning of final stage of kerakokinesis, the telophase of chromosomes that have reached their respective poles recondense and lose their individuality. The individual chromosomes can no longer be seen. Each set of chromatin material tends to collect each of the two poles. This stage, which shows following key events. First, chromosome cluster at opposite spindle poles and their identity is lost at discrete elements. Second, nuclear envelope develop around the chromosomes cluster and each pole forming two daughter nuclei. Third, nucleus, Golgi apparatus and Golgi complex endoplasmic reticulum reform anaphase. Telophase and interphase. Diagnostic view of mitosis. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei karyokinesis, but the cell itself is divided into two daughter cells by separation of cytoplasm called cytokinesis, at the end of which cell divided into completed division get completed. In an animal cell, This achieved by appearance of furrow in the plasma membrane. The furrow gradually deepens and ultimately join in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by its relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism in plant cell. Wall formation start in the cell of, of the cell and growth outward and meet existing lateral walls. The formation of new cell wall begin with the formation of simple precursor called cell plate that represent the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastic get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis as a result of multinucleated condition leading to formation of syncytium, and that is liquid endosperm nucleus. For example, a liquid endosperm nucleus. 10.3 Significance of mitosis. Mitosis or the equational division is usually restricted to diploid cell only. However, in some lower plant and in some social insect, haploid cell also divide by mitosis. It is very essential to understand that the significance of this division is in the life form of organisms. Are you aware of some examples where you study about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and cytoplasm. It therefore becomes essential for cell to divide to restore nucleus-cytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cell of upper layer of the epidermis, cell of lining of gut, and the blood cell are being constantly replaced. Mitotic division in meristematic cell is the apical and lateral cambium, result in continuous growth of plants throughout their life. 10.4 Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This is specialized kind of cell division that reduces the chromosome number by half, result in the production of haploid daughter cell. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexual reproducing organism, where fertilization restores the diploid phase. We come across the meiosis during gametogenesis in plants and animals. This led to the formation of haploid gametes. The key feature of meiosis are as follows. First, meiosis involves two sequential cycles of nuclear and cellular cell division called meiosis first and meiosis second, but only a single cycle of DNA replication occurs. Second, meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatids at S phase. Third, meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids and homologous chromosomes. Four haploid cells are formed at the end of meiosis. Second, meiotic event can be grouped in the following phases: meiosis first, prophase first, metaphase first, anaphase first, telophase first, and meiosis second, prophase second, metaphase second, anaphase second, and telophase second. Let's see meiosis first. 10.4.1 Meiosis first. Prophase first. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior. First, leptotin, zygotin, pachytin, triplotin, diakinesis. Here is the trick: Lalu, Jai, Plain, से दीपिका को देखने. So alpha leptotin, zygotin, pachytin, diplotin, and diakinesis. So first stage, leptotin. Stage of leptotin stage of chromosome become gradually visible under light microscope, and compaction chromosome continue throughout leptotin. This is followed by second stage of prophase, first called zygotin. During this stage, chromosomes start pairing together, and this process association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrograph of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptomal complex. The complex formed by the pair of synapse homologous chromosomes is called bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of process is relatively shorter lived compared to the next stage, that is pachytene. During this stage, the four chromatide of each bivalent chromosomes become distinct, clearly visible, appears as tetrad. This stage is characterized by appearance of recombination nodules, the site at which crossing over occur between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also enzyme-mediated process, and enzyme involved in is called recombinase. Crossing over led to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytene, leaving the chromosome linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptomal complex and the tendency of recombinant homologous chromosome of bivalent separated from each other except at the sister crossover. This X-shaped structure is called chiasmata, in whose site of some vertebrate diplotin can last for months or years. The final stage of meiotic process first is diakinesis. This is marked by the terminalization of chiasmata. During this phase, of chromosomes are fully condensed and meiotic spindles are assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappear and nuclear envelope also break down. Diakinesis represents transition of to metaphase. Metaphase first, the bivalent chromosomes align at the equatorial plane. The microtubule from opposite poles of spindle fiber attached to canonical homologous chromosomes. Stage of meiosis: first prophase, then metaphase, anaphase, telophase. Anaphase, the homologous chromosomes separate while sister chromatids remain associated at centromere. In anaphase first, we can see homologous chromosomes are getting divided, but sister chromatids are remain intact. These going to divide in anaphase second. Okay. Telophase first, the nuclear membrane or nucleolus reappears. Cytokinosis follows, and this is called dyad of cells. Although in many cases of chromosomes do undergo some dispersion, they do not reach extremely extended state of interphase nucleus. The stage between the two meiotic division is called interkinesis. Is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase second, a much simpler prophase than prophase first. 10.4.2 Meiosis second. Prophase second. Meiosis second is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis first, meiosis second resembles normal mitosis. The nuclear resembles disappearance by end of prophase second. The chromosomes again become compact. Metaphase second. At this stage of chromosome align at the equator and microtubules of opposite spindle get attached to the kinetochore of sister chromatids. Anaphase second. It begins simultaneously splitting up centromere each chromosome, which holding sister chromatids together, allowing them to move in opposite poles of the cell by shortening the microtubules of attached to kinetochores. Stages of meiosis first, so prophase first, metaphase second, prophase second, and anaphase second. In anaphase second, this sister chromatids going to be divided. So the 4 cells are formed, and RR, if these cells are diploid 2n, then all these cells are n. So this is also a reductional division. Telophase second, meiosis and will telophase second, in which the two group of chromosomes once again get enclosed by nuclear envelopes. Cytokinesis follow, resulting in formation of tetrad cell, 4 haploid daughter cell. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generations in sexual reproducing organisms. Even though the process per se paradoxically result in reduction of chromosome number by half, it also increase the genetic variability in the population of organism from one generation to another. Variation is very important in process of evolution. So meiosis is process of per se paradoxically. This is French term. You should Google this term and search why we say per se paradoxically to meiosis. Summary. According to the cell theory, cell arises from pre-existing cell. The process by which occur is called cell division. Any sexual reproducing organism start their life cycle from single cell zygote. Cell division does not stop the formation of mature organisms, but continues throughout its life. This stage throughout a cell passes from one division to next is called cell cycle. Cell cycle divided into two phases called first interphase A, period of preparation of cell, and second mitosis, the actual period of division. Interphase is further subdivided in G1 S and G2 S is phase of periodic when cell grow and carry out normal metabolism. Most organelle duplication occur during this phase. S phase marks the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. Chromosomes condensation occur during the prophase. Simultaneously, the centriole move to opposite poles in prophase. Nuclear envelopes and nucleolus disappear and spindle fibers start appearing. These all occur in prophase. Metaphase is marked by alignment of chromosomes at the equatorial plate. During anaphase, the centromere is divided and the chromatids start moving toward the opposite poles. Once the chromatids reach the two poles, the chromosomes elongation start, nucleolus and nuclear membrane reappear. This stage is called telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis thus is the equational division in which chromosomes number of parent is conserved in daughter cell. In contrast to mitosis, meiosis occur in diploid cell, which is destined to form gametes. It is called reduction division since it reduces the chromosomes number by half while making the gametes. In sexual reproduction, when the two gametes fuse and chromosome number restores the value of parent, meiosis is divided into two phases, meiosis first and meiosis second. In the first meiotic division, the homologous chromosome pair formed bivalent undergoes crossing over. Meiosis first undergo long prophase, which divided in further 5 phases. These are leptotin, zygotin, pakitin, diplotin, diakinesis. During metaphase, first bivalent arrange in the equatorial plate, and this is followed by the anaphase first, in which homologous chromosomes move to opposite pole with both their chromatids. Each pole receive half the chromosomes number of the parent cell. The telophase first in nuclear membrane nucleolus reappear. Meiosis second similar to mitosis. During anaphase, second sister chromatids separate. Thus, end of meiosis 4 applied. So end of meiosis 4 applied cells are formed. So we completed this chapter: cell division and cell cycle and cell division. Sorry, what's happened here? So we completed cell cycle and cell division. So we are gonna to cover plant physiology unit 4. तो भाई लोग इसमें क्या करेंगे, यूनिट 4 में अपन क्या करेंगे, चैप्टर 11 नहीं पढ़ूंगा या नहीं करूँगा बिकॉज ये नीट के सिलेबस में नहीं है। मेंटल न्यूट्रिशन नहीं करेंगे बिकॉज नीट के सिलेबस में नहीं है। इसमें करेंगे अपन फोटोसिंथेसिस, रिस्पिरेशन, प्लांट ग्रोथ एंड डेवलपमेंट, और इसके बाद एक कंप्लीट होने के बाद आ जाएगा ह्यूमन फिजियोलॉजी, अपना चैप्टर ख़त्म हो जाएगा। ठीक है भाई, तो फिजियोलॉजी स्टार्ट करेंगे। अभी थोड़ा थोड़ा तुम लोग आराम कर लो, 6:50 AM हो रहा है, चलो 6:55 से AM से स्टार्ट कर देंगे फोटोसिंथेसिस इन हायर प्लांट। तब तक के लिए क्या करें, आराम करें। ये सो गाइस, कैसे हो भाई लोग? बहुत कर लिया आराम, बहुत कर लिया रेस्ट, चलते हैं फिर लगते हैं स्ट्रगल वाली लाइफ में। 6:55 हो गया, आ गया लाइफ। सो भाई, आशा करता हूँ अच्छा। तो भाई, जिसने खोया है वही जान पाया है उसकी कीमत क्या थी। ठीक है, तो यार खोने से पहले ही मैं एक्सपीरियंस से बता रहा हूँ, खोने से पहले महत्व तो पता कर लो किसी भी चीज़ों का, कि खोने के बाद तुमको ज़्यादा अफसोस ना हो। ठीक है, तो कैरियर हो या प्यार हो, कुछ भी हो, उसमें लागू होता है। इसीलिए खोने वाली बहुत दुख होता है भाई। इसीलिए पढ़ाई कर लो, मेहनत कर लो, जो भी टाइम है। वो बोल रहे कि आज सुनाई नहीं दे रहा ब्रो मत बोल में भाई नहीं तेरा, बहुत ट्रेंड में है भाई ये सब गाना। वो बोल रहे कि आज सुनाई नहीं दे रहा ब्रो मत बोल में भाई नहीं तेरा। आ जा भाई, करते हैं प्लांट फिजियोलॉजी का। क्या मेरी जान, आ जाओ। Plant physiology, let's go. The description of structural variation of living organism over a period of time ended up as two apparently irreconcilable perspectives on biology. So, the two perspectives of essentially rested on two levels of organization of life forms and phenomena. The one described as organismic and above level of organization, while the second described at cellular and molecular level of organization. The first resulted in ecology and related to discipline. The second resulted in physiology and biochemistry. Description of physiological process in flowering plant as an example of is what is given in this chapter in this unit. The process of mineral nutrition in plants, photosynthesis, transport, respiration, and ultimately of plant growth and development is described in molecular terms, but in context of cellular activity and even at organismic level. Wherever appropriate, the relation of physiological process to environment is also discussed. So, Melvin Calvin, born in Minnesota in April 1911, he received his PhD chemistry from University of Minnesota. He served as professor of chemistry at University of California, Berkeley. Just after World War II, when the world was under shock after Hiroshima and Nagasaki bombing and seeing the ill effect of radioactivity, Calvin and co-workers put radioactivity a beneficial use. He, along with J. Bassam, studied the reaction of green plants forming sugar and other substances from raw material like carbon dioxide, water, and minerals by labeling the carbon dioxide C14. Calvin proposed that plants changes the light energy into chemical energy by transforming an electron in an organized array of pigment molecules and other substances. The mapping of pathway of carbon assimilation by photosynthesis earned him Nobel Prize in 1961. The principle of photosynthesis, as established by Calvin, are at present being used in studies on renewable resources of energy and material and basic studies in solar energy research. चैप्टर 11, सॉरी भाई, इसको तो करना ही नहीं है। सॉरी 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 अगर तुम साथ हो, तेरी नजरों में है मेरे सपने, मेरे सपनों में मुझे लगता है कि बातें दिल की। चलो भाई, चैप्टर 13, फोटोसिंथेसिस इन हायर प्लांट्स। 13.1 what do we know? 13.2 early experiments. 13.3 where does photosynthesis take place? 13.4 how many pigments are involved in photosynthesis? 13.5 what is light reaction? 13.6 electron transport. 13.7 where are ATP and NAD which we used. 30.8 the C4 pathway. 30.9 photo respiration. 30.10 factor affecting photosynthesis. So let's start. All animals including human beings depend on plants for their food. Have you ever wondered from where plant get their food? Green plant in fact have make or rather synthesize food they need. All other organisms depend on them on for their needs. The green plants make or rather synthesize the food they need through photosynthesis and therefore called autotrophs. You have already learned that the autotrophic nutrition is found in plants, and all other organisms that depend on green plants for food are heterotrophs. Green plants carry out photosynthesis, a physicochemical process by which they use light energy and drive synthesis of organic compound. Ultimately, all living forms on Earth depends on light for energy. The use of energy from sunlight by plant doing photosynthesis is the basis of life on Earth. Photosynthesis is important due to two reasons. First, it is primary source of all food on Earth. It is also responsible for the release of oxygen into atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on structure and photosynthesis machinery that and the various reactions that transform light energy into chemical energy. 30.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done earlier classes have shown that chlorophyll, green pigment of leaf, light and carbon dioxide are required for photosynthesis to occur. You may have carried out experiment to look for stars formation in two leaves, a variegated leaf and or a leaf that was partially covered with a black paper and exposed to light. On testing these leaves for presence of stars, it was clear that photosynthesis occur only in green plants, green parts of leaves in the presence of light. Another experiment you may have carried out where part of leaves included in test tube containing some KOH soaked with cotton absorbed CO2, while the other half exposed to air. The setup is then placed in light for some time. On testing for the presence of stars later in the two part of leaf, you must have found that the exposed part of leaf tested positive for stars, while the portion that are in test tube tested for negative. This showed that CO carbon dioxide were required for photosynthesis. Can you explain how this conclusion could be drawn? 30.2 Earlier experiment. It is interesting to learn about what does simple experiment that led to gradual development of understanding of photosynthesis. Joseph Priestley in 1733 to 1804 in 1970 performed a series of experiments that revealed the essential role of air growth of green plant. Priestley, you may recall, discovery of oxygen in 1970. Priestley observed that candle burning in closed space of bell soon get extinguished. ABC similarly a mouse would soon suffocate a closed space. He concluded that a burning candle or an animal that can breathe the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and candle continued to burn. The Priestley hypothesis as follows: plant restore to the air whatever breathing animal and burning candles remove. Can you imagine how Priestley have conducted the experiment using candle and plant? Remember, he would need to rekindle candle to test whether it burns after a few days. However, how many different ways you can think of light candle without disturbing the setup. Using a similar setup as as the one used by Presley, but placing it once in dark and once in sunlight. Then engine house 1930 to 1979 showed that sunlight is essential for plant process that somehow purified the air followed by burning candle breathing animals. Engine house is an elegant experiment with aquatic plants so that the bright sunlight small bubbles were formed around the green plants while the dark they did not. Later he identified that the green bubbles to be oxygen, hence so that it is only the green part of plant that could release oxygen. It was not until 1854 that Julius Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His later studies show that green plant substances in plants chlorophyll as we know it is located in special bodies called chloroplast within plant cell. He found that green parts of plant is where glucose is made, and that glucose is usually stored as a starch. Now consider an interesting experiment done by T.W. Engelmann in 1843 to 1904. Using a prism, he split light into spectral components and then illuminated a green algae Chlorophylla, placed in suspension of aerobic bacteria. The bacteria were used to detect the site of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of split spectrum. A first exchange spectrum of photosynthesis were thus described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of 19th century, the key feature of plant photosynthesis were known, namely that plants could use light energy to make carbohydrate from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as carbon dioxide, water in this form, light, and in the presence of light form CH2O plus oxygen, where CH2O represents the carbohydrate glucose 6 carbon sugar. A milestone contribution of understanding photosynthesis was that made by microbiologist Cornelius von Linne from 1897 to 1985, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from suitable oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed as follows: 2H2O plus CO2 in light presence form 2F CH2O plus water. In green plant, water is the hydrogen donor and oxidized to O2. Water oxidized and CO2 reduced. Some organisms do not release oxygen during photosynthesis. When H2S instead of hydrogen donor for purple green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not oxygen. Hence, they infer that oxygen evolved by the green plant comes from water, not from carbon dioxide. This was later proved by using radioisotopic technique. The correct equation that could be represent the overall process of photosynthesis refers 6CO2 plus 12H2O in the presence of light 6 C6H12O6 plus 6H2O plus 6O2, where C6H12O6 represents glucose, the oxygen released from water. This was proved using radioisotope techniques. Note that this is not single reaction but describes a multi-step process called photosynthesis. So 13.3, where does photosynthesis take place? You would of course answer in the green leaf chloroplast. Based on what earlier read in chapter 8, you identified photosynthesis does not take place in green leaves, but it doesn't also other green plant parts of plants. You would recollect from the previous unit that mesophyll cells in the leaves have large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of mesophyll cell such that they get optimal quantity of incident light. When do you think chloroplast will align themselves in flat surface of parallel? तो फ्लैट पिकअप करेगा जब लाइट परपेंडिकुलर आ रहा हो. You have studied the structure of chloroplast in chapter 8. Within the chloroplast there is a membranous system consisting of a grana, the stroma lamella, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping light energy and also the synthesis of ATP and NADPH. In stroma, the enzymatic reaction of synthesis of sugar, which turn form starch, the former set of reactions, since they are directly light driven, are called light reaction. Photochemical reaction, the latter are not directly light driven but are dependent on product of light reaction, ATP and NADPH. Hence, the distinguished later they are called by convention dark reaction, carbon reaction. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. Diagrammatic representation of electron micrograph section of chloroplast. Here is outer membrane, inner membrane, stroma lamella, grana, stroma, and ribosomes, starch granule, and lipid droplet. 13.4. How many type of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, when we can look for answer to this question by trying to separate the leaf pigment of any green plants through paper chromatography. A chromatographic separation of leaf pigments shows that the color that we see in leaf is not due to single pigment but due to four pigments: chlorophyll A, brighter blue green in chromatogram; chlorophyll B, yellow green; xanthophylls yellow; and carotenoids yellow to yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at a specific wavelength. Can you guess the abundant plant pigments in the world? Let us study the graph showing the ability of chlorophyll pigment A absorb light different wavelength. Of course, you are familiar with wavelength of visible light spectrum with your. Now look at the figure itna, and wavelength maximum photosynthesis occur in plant. Can you see a wavelength of maximum absorption of chlorophyll A in the blue and red region? Okay, in blue and red region maximum absorption of light occurs. Higher rate of photosynthesis, hence we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. Here is a graph showing absorption spectrum of chlorophyll A and B and carotenoids. So here it is graph of chlorophyll A, chlorophyll B, and this is carotenoids, and this is graph of chlorophyll A. Rate of photosynthesis, तो जहाँ ऑक्सीजन रिलीज, जहाँ फोटोसिंथेसिस ज़्यादा हो रहा है वहाँ ऑक्सीजन रिलीज हो ज़्यादा हो रहा है. एंड लाइट अब्जॉर्प्शन, तो लाइट अब्जॉर्प्शन जहाँ हो रहा है ज़्यादा यहाँ, वहाँ पे फोटोसिंथेसिस भी ज़्यादा हो रहा है. सो यही कुछ ग्राफ है. भाई साहब, this graph together shows that most of photosynthesis take place in blue and red region of spectrum. Some photosynthesis does not take place other wavelength of visual spectrum. Let us see how this happens. Through chlorophyll is the major pigments responsible for trapping light. Other thylakoid pigments of chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments. Also, accessory pigments also absorb light and transfer to energy. Chlorophyll A, indeed, they are not enable wide range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. 13.5. What is light reaction? Light reaction for photochemical phases include light absorption, water splitting, oxygen releasing, and formation of high energy chemical intermediates ATP and ATPase. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complex LSH within the photosystem first, PS first, and photosystem second, PS second. These are named in sequence of their discovery and not in sequence of in which function they perform. The light harvesting complex made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigment except one molecule of chlorophyll A, forming a light harvesting complex, also known as antenna. These pigments help in make photosynthesis more efficient by absorbing different wavelengths of light. Single chlorophyll A molecule form reaction center. The reaction center is different both the photosystem. Photosystem first, the reaction center chlorophyll A has absorption peak at 700 nanometer, hence is called P700, while the P second has absorption maximum P680 nanometer and it's called P680. The light harvesting complex: photon, reaction center, pigment molecule, acceptor. 30.6. Electron transport. In 47 seconds, the reaction center chlorophyll A absorbs 680 nanometer wavelength of light, causing electron become excited and jump into the orbits farther from atomic nucleus. These electrons are picked up by electron receptor, which passes them to electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of oxidation, reduction, or redox potential scale. The electrons are not used up as they pass through the electron transport chain, but the passed on to the pigments of photosystem first. Simultaneously, electrons in the reaction center PS1 are also excited when they receive red light of wavelength 700 nanometer and then transferred to another acceptor molecule that has a greater redox potential. These electrons then move to downhill again, this time molecule of high rich in NADPH. The addition of these electrons reduces NADP plus to NADPH2. This whole scheme of transfer electron starting from pH2 uphill to acceptor down the electron transport chain to PS1, excitation of electron transfer of another acceptor, and finally downhill to NADP, reducing it to NADPH H plus, is called the Z zone. Due to its characteristic shape, this shape is formed when all the carriers are placed in sequence of redox potential scale. Here is Z scheme of light harvesting complex, light reaction. Z scheme light harvesting complex, and from LHL photosystem light harvesting complex, this electron is accepted by photosystem second, and from photosystem second this comes in electron transport system. From here it comes to from here ATP formed and again go to electron transports light harvesting complex, and from here electron भाई 100 बात की एक बात है, यहाँ पे वाटर का ब्रेकडाउन हुआ लाइट हार्वेस्टिंग कॉम्प्लेक्स फर्स्ट में, ठीक। और यहाँ से इलेक्ट्रॉन जो रिलीज होता है वो आता है आपका इसमें, क्या बोलते हैं भाई इसको, हाइपोथेसिस। उसमें तुम्हारा नहीं हाइड्रोजन आ गया और ऑक्सीजन रिलीज हो गया यहाँ से। फिर यहाँ से जो इलेक्ट्रॉन एक्सेप्टर है फिर फोटोसिंथेसिस सेकेंड में प्लास्टोक्विनोन ही है ना, वहाँ से आएगा तो वहाँ से जाता है फोटो सिस्टम सेकेंड में, यहाँ पे NADPH2 बनता है। तो एक लाइट हार्वेस्टिंग कम्प्लेक्स पास फिर क्यों दिखा रहा है, ये इसका मुझे कोई आइडिया नहीं आ रहा, ठीक पढ़ना पड़ेगा मुझे फिर। अब स्प्लिटिंग ऑफ वाटर कैसे हो रहा है, 13.6 you would have then asked how does pH second supply electron continuously. The electron that were moved from photosynthesis second now be replaced. This is achieved by electron available due to splitting of water. The splitting of water associated with pH second, water is split into 2H plus oxygen and electron. This creates oxygen, one of the net product for photosynthesis. The electron needed to replace those removed from photosystem first and provided by photosystem second. वाटर ब्रेक हुआ, 4 H प्लस आयन प्लस O2 प्लस 4 इलेक्ट्रॉन रिलीज हुआ। ये कहाँ गया, ये आपका उस हाइपोथेसिस कीमो हाइपोथेसिस ऑस्मोटिक हाइपोथेसिस में गया, जहाँ पे प्रोटॉन ग्रेडियंट बनाएगा तो क्या होगा, वहाँ पे ATP फॉर्म होगा और ए इलेक्ट्रॉन चला जाता है क्या, NADPH2 को बनाने में चला गया, ऑक्सीजन रिलीज हो गया। वी नीड टू एम्फसाइज हियर दैट वाटर स्प्लिटिंग कॉम्प्लेक्स इज एसोसिएटेड विद pH सेकेंड। देख भाई, वाटर स्प्लिटिंग कॉम्प्लेक्स जो होता है ना pH सेकेंड से जुड़ा होता है, विच इट्स फिजिकली लोकेटेड ऑन इनर साइड ऑफ, और ये कहाँ होता है, इनर साइड में होता है ऑफ मेम्ब्रेन ऑफ थैलेकोइड। देन वेयर आर प्रोटॉन्स O2 formed likely released into lumen of the outer side of membrane. 30.6 cyclic and non-cyclic photophosphorylation. Living organisms have capability to extract energy from oxidizable substrate and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bond. This process through which ATP is synthesized by cells in mitochondria and chloroplast, named phosphorylation. Photophosphorylation is synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in the series, first photosystem second and then photosystem first, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through a electron transport chain and seen earlier in the Z scheme. Both the ATP and NADPH2 are synthesized by this kind of ATP flow. Okay. When PS1 is functional, then electron is circulated within the photosystem and the photophosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is the stroma lamella. While the membrane of or lamella of grana have both PS1 and PS2, the stroma lamella membrane lack PS2 as well as NADP reductase enzyme. The stroma lamella membrane lack PS2 as well as NADP reductase. The excited electron does not pass on to the NADP plus but is cyclic back to PS1 complex through the electron transport chain figured in 30.6. The cyclic flow hence resulted only in the synthesis of ATP but not in NADPH. A plus H plus. Cyclic photophosphorylation also occur when only light of wavelength beyond 680 nanometer are available for excitation. Cyclic photophosphorylation, लाइट आया कहाँ, P700 पे क्या PS1 है और यहाँ से क्या हुआ, इलेक्ट्रॉन एक्सेप्टर के पास गया, फिर से यहाँ इसमें क्या होता है, नॉन साइक्लिक साइक्लिक है ना, और साइक्लिक में तो NADPH2 का फॉर्मेशन नहीं होगा बिकॉज इलेक्ट्रॉन तो साइकिल बैक हो जा रही है ना। 30.6.3 की ऑस्मोटिक हाइपोथेसिस। Let us now try to understand how actually ATP synthesized in chloroplast. The chemoosmotic hypothesis has put further to explain the mechanism. Like in respiration, the photosynthesis to ATP is linked to development of proton gradient across the membrane. This time, these are membranes thylakoid, where is one difference through here. Proton accumulation toward inside the membrane, that is in the lumen respiration, proton accumulate in intermembrane of mitochondria, electron through it is. Let us understand cause of proton gradient across the membrane. We need to consider again the process that takes place during activation of electron from transport defined proton gradient. Since splitting of water molecule takes place by inner side of membrane, the proton of hydrogen ion that produced by splitting water accumulate within the lumen of thylakoids. Okay, here hydrogen is accumulated in membranes of thylakoid that formed by PS second plastogonium cytochrome B. As electrons move through photosystem, protons are transported across membrane. This happens because the primary receptor of electron, which is located toward the outer side of membrane, transfer its electron not to an electron carrier but an hydrogen carrier. Hence, this molecule removes a proton from stroma while transporting an electron, which when this molecule passes to its electron to electron carrier on inner side of membrane, the proton is released into inner side of lumen of membrane. See NADP. NADP reductase enzyme are located in stroma of the membrane along with electron that come from acceptor of PS1. Proton are necessary for reduction of NADP plus to NADPH2. These protons are also removed to stroma. Hence, within chloroplast, proton stroma decreases while luminal accumulation of proton. This creates proton gradients across thylakoid membrane, a measurable decrease in pH. Why are so interested in proton gradient? The gradient is important because it is breakdown of gradient that led to the synthesis of ATP. The gradient is broken down due to movement of proton across membrane, stroma through transmembrane, a channel of CF0 of ATP synthase. The ATP synthase enzyme consists of two parts. One labeled as CF0 is embedded in the thylakoid membrane and form transmembrane channel that carries out facilitated diffusion of proton across membrane. The other portion is called CF1 and protrudes on the outer side of thylakoid membrane on the side that faces the stroma. The breakdown of a gradient provide enough energy to cause conformational change in CF1 particle of ATP synthase, which make the enzyme synthesize several molecules energy packed ATP. Chemoosmosis require a membrane of proton pump, proton gradient, ATP and ATP synthase. Energy is used to pump proton across a membrane to create gradient or high concentration of proton within thylakoid lumen. ATP synthase that has a channel that allows diffusion of proton back to across membrane. This release enough energy to activate ATP synthase enzyme that catalyze the formation of ATP. Along with the NADPH produced by the movement of electron, the ATP will be used immediately in biosynthetic reaction taking place in stroma responsible for fixing CO2 and synthesis of sugar. 13.7 Where are ATP and NADPH used? We learned that product of light reaction and ATP, NADPH and O2 of these O2 diffuse out of chloroplast, while ATP and NADPH are used to derive process leading to synthesis of food, more accurately sugars. This biosynthetic phase of photosynthesis, this process does not directly depend on presence of light but dependent on the product of light reaction, that is ATP and NADPH2. Beside CO2 and water, you may wonder how this would be verified. It is simple. Immediately after light reaction, light become unavailable. The biosynthetic process continues for some time and then stops. If then light is made available, the synthesis start again. Can you hence say that calling the biosynthetic phase dark reaction is misnomer? Discuss this amongst yourself. Discuss misnomer have I? Yes, because it depend on light, indirectly depend on light, Let us now see how the ATP and NADPH2 is in the biosynthetic phase. We saw earlier CO2 combined with water to produce CH2O, means carbohydrate or sugar. It was interesting to scientists to find out how this reaction proceeded, or rather what was the first product formed when carbon dioxide is taken in the reaction or fixed just after World War II. Among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive C14 by him in algal photosynthesis study led to discovery that the first carbon dioxide fixation product was a 3-carbon organic acid. He also contributed to working out complete biosynthetic pathway, hence it was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many कार्बन एटम does it have? तो बताओ PGA का 3 कार्बन होता है ना. Scientists also tried to know whether all plants have PGA as first product of carbon dioxide fixation, or whether any other product was formed in other plants. Experiment conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of carbon dioxide fixation was an organic acid, but on which has 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA, since carbon dioxide assimilation during the photosynthesis was said to be two main types. Those plants in which the first carbon dioxide fixation product C3 PGA, the C3 pathway, and those which first product was C4 acid, oxaloacetic acid, the C4 pathway. These two groups of plants show other associated characteristics that we will discuss later. 13.7.1 Primary acceptor of CO2. Let us now ask yourself a question. What was asked by scientists who were struggling to understand the dark reaction? How many carbon atom or molecules have which after accepting fixing carbon dioxide would have 3 carbon of PGA? These studies very unexpectedly showed that acceptor molecule of 5 carbon ketose sugar ribulose bisphosphate RABP did anything possibility. Do not worry. The scientists also took a long time to conduct many experiments to reach the conclusion. They also believed that since the first product was C3 acid, the primary acceptor was 2-CD, but it became wrong. They spent many years trying to identify 2 carbon compound before discovery of 5 carbon compound RABP. 13.7.2 Calvin cycle. The Calvin and his co-worker worked on the whole pathway so that pathway operated in cyclic manner. The RUBP regenerate us now see how many Calvin pathway operates and where sugar synthesized. Let us let us out let us at the outset understand very clearly that Calvin pathway occur in all photosynthesis phases. It does not matter whether they are C3 or C4 plants pathway. For each understanding, Calvin cycle is described under the three stages of carboxylation, reduction, and regeneration. First, carboxylation. Carboxylation is the fixation of carbon dioxide into stable organic intermediate. Carboxylation is the most crucial step in Calvin cycle where carbon dioxide is utilized for carboxylation of RUBP. This reaction is catalyzed by enzyme RUBP carboxylase, which results in the formation of two molecules of 3-phospholic acid. Since this enzyme also has an oxygenation activity, it would be more correct to call RUBP carboxylase oxygenase RUBisco. Here is sucrose. RUBP ribulose 1,5 bisphosphate combines carbon dioxide and water form this process known carboxylation form 3-PGA phosphoglycerate, and after reduction using 1 ATP and NADPH2 in this form triphosphate. After 3 cycles this form sugar. ATP and used in regeneration, then RUBP again formed. The Calvin cycle proceeds in 3 stages. First, carboxylation, during which carbon dioxide combined with ribulose 1,5 bisphosphate. Second, reduction, during which carbohydrate is formed at the expense of photochemical made ATP and NADPH. Third, regeneration, during which carbon dioxide acceptor ribulose 1,5 bisphosphate is formed again so that cycle continues. Second, reduction. And these are series of reactions that lead to formation of glucose. These steps involve utilization of two molecules of ATP for phosphorylation and 2 NADPH for reduction per carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide and 2 turns of cycle are required for formation of 1 molecule of glucose from this pathway. Third, regeneration. Regeneration of carbon dioxide molecule RUBP crucial if cycle is continued uninterrupted, and regeneration steps require ATP for phosphorylation or to form RUBP. Hence, for every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH2 are required. It is probably to meet the difference in number of ATP and NADPH. In dark reaction, that cyclic phosphorylation takes place. In 6 CO2, 18 ATP and 12 NADPH2, out 1 glucose, 18 ADP, 12 NADP. 13.8 C4 pathway. Plant that adapted to dry tropical region have C4 pathway mentioned earlier. Though these plants have C4 oxaloacetic acid, the first carbon dioxide fixation product, they use a C3 pathway or Calvin cycle as the main biosynthetic pathway. Then, in what way they different from C3 plants? This question have reasonably asked. C4 plants are special. They are special leafy anatomy. They tolerate higher temperature, shows response to higher light intensities. They lack process called photophosphorylation as greater productivity of biomass. Let us understand one by one. Study vertical section of leaf. One C3 plant and other C4 plant. Do you notice difference in क्या दिया, the particular large cells around the vascular bundle C4 plants are called C bundle sheath. The leaves which have such anatomy is said to be Cranch anatomy. Cranch means wreath and is reflection arrangement of cells. The bundle sheath may form several layers around vascular bundle. They are characterized by having large number of chloroplasts, thick wall impervious to gaseous exchange, and no intercellular spaces. You may like to cut a section of leaf, carbon plant match, or sorghum to observe the Cranch anatomy and distribution of mesophyll cell. It would be interesting to know that collect leaves diverse species of plant around you. Cut vertical section of leaf, observe under microscope. Look for bundle sheath around vascular bundle. The presence of bundle sheath would be help in identification of C4 plants. And now to study pathways shown in figure 13.9. The pathway that has been named hash and slag pathway is an cyclic process. Let us study the pathway by listing the steps. The primary carbon dioxide acceptor is 3 carbon molecule phosphoenol pyruvate PEP and the present in mesophyll cells. The enzyme responsible for fixation of phosphoenol pyruvate carboxylase or PEPK. It is important to register that the mesophyll cell lack Rubisco enzyme. The C4 acid or oxaloacetic acid is formed in mesophyll cell. It then forms other 4 carbon compound like malic acid or aspartic acid in the mesophyll cell itself, which are transported to bundle sheath cells. In the bundle sheath cells, C4 acid are broken down to release carbon dioxide and 3 carbon molecules. The carbon molecules is transported back to mesophyll cell, whether it is converted to PEP again, thus completing the cycle. The carbon dioxide released in the bundle sheath cell enter the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in enzyme ribulose, bisphosphate carboxylase, and oxygenase Rubisco, but lack peptidase. Thus, the basic pathway that result in the formation of sugar in Calvin pathway is the common in 2, C3 and C4. Did you note that Calvin pathway occur in all mesophyll cells of C3 plant? In the C4 plant, it does not take place in a mesophyll cell, but does so only in bundle sheath cell. Here diagrammatic representation of hash and slag pathway. Carbon dioxide enters in mesophyll leaves. It reacts to bicarbonate and phosphoenol and bicarbonate. Carbon dioxide dissolves in cytoplasm form bicarbonate and it reacts to phosphoenol pyruvate and form oxaloacetic acid. After this fixation, this OA C4 carbon transported to bundle sheath, and in bundle sheath decarboxylation of OAA occur and carbon dioxide releases. This fixed by in Calvin cycle. After that, 3 carbon transported again to this mesophyll cells. 30.9 Photo respiration. Let us understand one more process that creates the important differences between C3 and C4 plant photorespiration. To understand photorespiration, we have to know little bit more about first step of Calvin's pathway. The first carbon dioxide fixation step, which is this is reaction where RUBP combined with carbon dioxide to form 2 molecules of 3-PGA. That is catalyzed by Rubisco. RUBP plus carbon dioxide in presence of Rubisco form 2 phosphoglyceric acid. Rubisco that is most abundant enzyme in the world. Do you wonder why? Characterized by the fact that active site can bind to both carbon dioxide and oxygen, hence the name. Can you think how this could be happen? How this could be possible? Rubisco has a much greater affinity for carbon dioxide when the carbon dioxide oxygen is nearly equal. Imagine that. Imagine what would happen if this were not so. This binding is competitive. It is relative concentration of carbon dioxide and oxygen and carbon dioxide that determine which two will bind to enzyme. In C3 plants, some oxygen does bind to Rubisco and hence carbon dioxide fixation decreases. Here, RUBP instead of being converted to 2 molecules of phosphoglyceric acid bind to oxygen to form 1 molecule of phosphoglycerate and phosphoglycolate to carbon. In a pathway called photorespiration, pathway, there is neither synthesis of sugar nor ATP. Rather, it results in release of carbon dioxide, which is utilized for ATP. In the photorespiration pathway, there are no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. The C4 plant photorespiration does not occur. This is because they have mechanism that increase concentration of CO2 at the enzyme site. This takes place when C4 acid from mesophyll is broken down in the bundle sheath cell to release carbon dioxide. This result in increasing the intercellular concentration of carbon dioxide. In turn, this ensures that the Rubisco functioning as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plant lacks photorespiration, you probably understand why productivity and yield are better of this plant. In addition to these plants show tolerance of higher temperature based on above discussion. You compare showing C3 plant and C4 plant pathway. Use table format to fill the information. So fill this information. करें क्या भाई? करें पूरा या छोड़ें इसको, स्किप करें? चलो स्किप करके आते हैं 13.10 पे, क्योंकि उसको करने में भी टाइम लगेगा ना. 13.10 factor affecting photosynthesis is understanding of factor that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield that plant, including crop plant. Photosynthesis is under influence of several factors, both internal plant and external. The plant factor includes the number, size, age and orientation of leaves, mesophyll cell and chloroplast, internal carbon dioxide and amount of chlorophyll. The plant and internal factor dependent on genetic predisposition of and growth of plant. The factor responsible would be included in availability of sunlight, temperature, carbon dioxide concentration and water. As a photosynthesis, all these factors will simultaneously affect its rate. Hence, through several factors interact with simultaneously affect photosynthesis or carbon dioxide fixation. Usually, one factor is major cause of one that limit the rate. Hence, any point of rate will determined by factor available at suboptimal level. When several factors that is affected any biochemical process, Blackman 905 law of limiting factor comes into effect. This states that if the chemical process is affected by more than one factor, then its rate will be determined by factor which is nearest to minimal value. It is a factor which directly affects the process by if its quantity is changed. For example, the despite of presence of green leaf and optimal light and carbon dioxide condition, the plant may not photosynthesize if the temperature is very low. This leaf is given the optimal temperature will start photosynthesizing. Here is 13.10.1 light. The need to distinguish between light quality and light intensity and duration of exposure of light while discussing the light as a factor that affect the photosynthesis. There is a linear relationship between the incident light and carbon dioxide fixed rate at low light intensity. At higher light intensities, gradually the rate does not so further increase. As other factor become limiting, what is interesting to note is that the light saturation occur at 10% of full sunlight. Hence, except the plant in shades or in dense forest, light are a rarely limiting factor in nature. Increase in incident beyond point causes breakdown of chlorophyll, a decrease of photosynthesis. Here is graph of light intensity on rate of photosynthesis, light intensity and rate of photosynthesis. Here is directly proportional, and after that this become constant, and after that some limiting factor rate of photosynthesis decreases due to photolysis. 10.13.10.2 Carbon dioxide concentration. Carbon dioxide is a major limiting factor of photosynthesis. The concentration of carbon dioxide is very low in atmosphere between 0.03 to 0.04%. Increase in concentration up to 0.05% can cause an increase in carbon dioxide fixation rate. Beyond this level can become damaging over longer period. Carbon dioxide C3 and C4 plant respond differently to carbon dioxide concentration. At low light condition, neither group respond to high CO2 condition. At high intensity, both carbon dioxide, both C3 and C4 plants show increase in rate of photosynthesis. What is important to note is that C4 plants show saturation about 330 microliter, while a C4 plant respond increased carbon dioxide concentration beyond 450 microliter. Thus, current availability of carbon dioxide levels is limiting to C3 plants. The fact that C3 plant respond to higher carbon dioxide concentration by showing increase in rate of photosynthesis leading to higher productivity has been used to greenhouse crop such as tomato, bell pepper, and allow growth of carbon dioxide in the atmosphere led to higher yields. 13.10.3 Temperature. The dark reaction being enzymatically are temperature controlled through the light reaction are also temperature sensitive. They are affected to much lesser extent. The C4 plant respond to higher temperature and show higher rate of photosynthesis, while C3 plants have much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat, and they are adapted to. Tropical plants have a higher temperature optimum to plant adapted temperate climates. 13.10.4 Water. Even though water is one of the reactant in the light reaction, the effect of water as a factor more through its effect on plants rather than directly on photosynthesis. Water stresses causes stomata to close, hence reducing the carbon dioxide availability. Besides, the water stress also make leaves wilt, thus reducing the surface area of leaves and their metabolic activity as well as सो भाई अपना फोटोसिंथेसिस कंप्लीट हुआ, इसका समरी कर देखते हैं. Summary. Green plants make their own foods, photosynthesize. During the process, carbon dioxide from the atmosphere is taken by leaves through stomata and used for making carbohydrate. Principally, glucose and starch photosynthesis take place only in green plants for green parts of plants, mainly by the leaves. Within the leaves, the mesophyll cell have a large number of chloroplasts that are responsible for carbon dioxide fixation. Within the chloroplast, the membranes are sites for light reaction within chemosynthetic pathway occurring in stroma. Photosynthesis has two stages: the light reaction, carbon dioxide fixation. In the light reaction, the light energy is absorbed by pigment present in antenna and funneled to special chlorophyll A molecule called reaction center of chlorophyll. There are two photosystem: photosystem first and photosystem second. Photosystem first has 700 nanometer absorbing chlorophyll A P700 molecule and its reaction center, while the PS second has P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through PS second to PS first and finally NAD forming NADH. During this process, a proton gradient is created across the membrane of thylakoid and break down a proton gradient due to movement through the F0 part of ATPase enzyme. Release energy through synthesis of ATP. Splitting of water molecule is associated with PS second, resulting in the release of oxygen. Proton transfer electron to PS second. In the carbon fixation cycle, carbon dioxide is either added to enzyme Rubisco to 5 carbon compound RUBP that is converted to 2 molecules enzyme 3 carbon PGA. This is then converted to sugar by Calvin cycle and RUBP regenerated. During this process, ATP and ADPH synthesized in light reaction and utilized. RUBP also catalyzes wasteful oxygenation and reaction C3 plant photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In this plant, first synthesis of carbon dioxide that takes place in mesophyll is 4 carbon compound. In the bundle of synth, Calvin pathway is carried out synthesis of carbohydrate. So अपना फोटोसिंथेसिस कंप्लीट हुआ. ओ भाई साहब, इसका तो हैंग मारने लगा भाई. मेरे दिमाग के साथ मेरा टैब भी हैंग मार रहा है. सॉरी गाइस. चलो भाई, चैप्टर 14 रेस्पिरेशन प्लांट. इसके लिए अपन लेते हैं 7:59 to 8:04 AM रेस्ट. सुनो, जुबानी सुनो, मुझे प्यार हुआ था, इकरार हुआ था। मुझे प्यार हुआ था। प्यार हुआ था। तो गाइज कैसे हो भाई लोग? तुम लोग को भी प्यार हुआ है क्या कभी? अब बोलोगे नहीं भैया नहीं हुआ। क्या बात करते हो? हुआ नहीं है भाई, नीट से हुआ है, अपने लक्ष्य से हुआ है, अपने कैरियर से हुआ है, प्यार तो हुआ ही है। अब देखो भाई, तुम लोग को एक और शायरी सुनाते हैं अभी। जो किस्मत में नहीं था, जिंदगी उसी से टकरा गई। हाँ, बहुत दुख की बात है। नीट ड्रॉपर का ये शायरी। और क्या कोई अच्छा शायरी दिखे तो बोलते हैं। एक और शायरी मिला। बस इतना बता दो, इंतजार करूं या बदल जाऊं तुम्हारी तरह। वाह। ये है बिल्लू शायर फ्रॉम बांग्लादेश। एक और शायरी देखते हैं। किसी शायर ने मौत पर क्या खूब लिखा है। क्या लिखा है? लिखते लिखते मर गया, कहानी बाहर आया नहीं। क्या ही बताएं। देखो एक और बात है भाई, हमेशा किस्मत खराब नहीं होती, कभी कभी हम फैसले भी गलत ले लेते हैं, है कि नहीं? किस्मत खराब नहीं है भाई, तुम गलत फैसला ले लिए, गलत फील्ड चुन लिए, गलत लोगों को चुन लिए। इट्स नॉट योर मिस्टेक, इट्स योर मिस्टेक। तुमने चुना, तुम्हारा मिस्टेक है, और क्या। कहानी सुनो, जुबानी सुनो, मुझे प्यार हुआ था। वन मिनट टू गो ब्रो, 1 मिनट रुक जाओ, करते हैं अपन स्टार्ट। रेस्पिरेशन इन प्लांट। रुकेगा नहीं, झुकेगा नहीं, पुष्पराज आज पड़ेगा रेस्पिरेशन इन प्लांट। वाह शेर, तुम तो शेर निकले। चलो भाई, लेट्स गो ब्रो, स्टार्ट करते हैं ब्रो। रेस्पिरेशन प्लांट 14.1, डू प्लांट्स ब्रीथ। 14.2, ग्लाइकोलिसिस। 14.3, fermentation. 14.4, aerobic respiration. 14.5, respiratory valency. 14.6, amphibolic pathway. 14.7, respiratory quotient. So let's go bro. All of us breathe to live, but why breathing is so essential for life? What happens when we breathe? क्या होता है ब्रीथ करते हैं तो? Also do all living organisms including plant and microbes breathe? If so, how? कैसे? All living organisms need energy for carrying out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. ब्रीथिंग के लिए भी भाई एनर्जी चाहिए ब्रो। Where does all this energy come from? We know we eat food, हमको पता है खाना खाते हैं, but how is this energy taken from food? How is this energy utilized? Do all food give the same amount energy? Do plants eat where do plants get their energy from? And microorganisms, for their energy requirement, do they eat food? Do You may wonder at the several questions raised above. They may seem to be very disconnected, but is in reality the process of breathing is very much connected to process of energy from food. Let us try to understand how this happens. All the energy required for life process are obtained from by oxidation of some macromolecules that we call food. Only green plants and cyanobacteria can prepare their own food by the process of photosynthesis. They trap light energy and convert it into chemical energy that is stored in the bonds of carbohydrate like glucose, sucrose, and starch. We must remember that in green plants too, not all cells, tissue, and organs photosynthesize. Only cells containing chloroplast that are most often located in the superficial layer carry out photosynthesis. Hence, even in green plants, all other organs, tissue, and cells that are non-green need for food for oxidation. Hence, food has to be translocated to all non-green green plants parts. Animals are heterotrophic, that is, they obtain food from plants. Directly herbivore or indirectly carnivores. Saprophytes like fungi are dependent on dead and decaying matter. What is important to recognize is that ultimately all the food that is required for life process comes from photosynthesis. This chapter deals with the cellular respiration or the mechanism of breakdown of food material within cell to release energy and the trapping of this energy for synthesis of ATP. Photosynthesis, of course, takes place within chloroplast in the eukaryotes, where the breakdown of complex molecules its energy takes place in cytoplasm and the mitochondria, also only in eukaryotes. The breaking of carbon-carbon bonds of complex compounds through oxidation within the cells leading to the release of considerable amount of energy is called respiration. The compound that is oxidized during this process is known as respiratory substrates. Usually, carbohydrates are oxidized to release energy, but proteins, fats, even organic acid can be used in respiratory substance in some plants under certain conditions. During oxidation within a cell, all energy obtained in this respiratory substrate is not released free into the cell or in single step. It is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped in chemical energy in the form of ATP. Hence, it is important to understand that energy released in oxidation is respiration is not or cannot be used directly but is used to synthesize ATP, which is broken down whenever energy need to be utilized. Hence, ATP acts as energy currency of the cell. This energy trapped in ATP utilized in various energy requiring process for organism to and carbon skeleton produced during respiration is used as precursor of biosynthesis and other molecule in the cell. 14.1 Do plants breathe? अरे भाई साहब, मैं तो ब्रीथ नहीं कर पा रहा हूँ यार. रेस्पिरेशन करते करते मैं रेस्पिरेशन कैसे करना है मैं भूल गया. Do all plants breathe? Well, the answer to this question is not quite so direct. Yes, plants require O2 for respiration to occur, and they also give out carbon dioxide. Hence, plants have systems to replace that ensure availability of oxygen. Plants, unlike animals, have no specialized organ for gaseous exchange, but they have stomata and lengthy cells for this purpose. There are several reasons why plants can get along without respiratory organ. First, Each plant part take care of their own gas exchange needs. There is very little transport of gases from one plant part to another. Second, plant do not present great demand for gas exchange. Root, stem and leaves respire at a rate of far lower than animals do. Only during photosynthesis are large volume of gases exchanged, and each leaf is well adapted to take care of own needs during these periods. When cell photosynthesize, availability of oxygen is, is not a problem in these cells since oxygen is released within the cell. Third, distance that gases must diffuse even in large bulky plant is not great. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about thick woody stem and roots? The stem in stem, the living cells are organized in thin layer present thin layer inside and beneath the bark. They also have opening called lenticels. These cells in the interior are dead and provide only mechanical support. Thus, the most cells of plants have at least a part of their surface in contact with air. This is also facilitated by the loose packing parenchyma cell in leaves. Stem from roots which provide interconnected network of air space. The complete combustion of glucose which produce carbon dioxide and water as end product yield energy, most of which given out as heat. C6H12O6 glucose plus oxygen, 6 carbon dioxide plus 6 water molecule plus energy. If this energy is useful to the cell, it should be able to utilize it to synthesize other molecules that the cell requires. The strategy that the plant cell uses to catabolize the glucose molecule in such a way that not all the liberated energy goes out as heat. The key is to oxidize in glucose not in one step but in several small steps, enabling some steps to be just large enough such that energy released can be coupled to ATP synthesis. How this is done? Essentially, the story of respiration. During the process of respiration, oxygen utilized and carbon dioxide, water and energy is released as product. The combustion reaction requires oxygen, but some cells live where oxygen may or may not be available. Can you think such situation an organism where oxygen is not available? There are sufficient reasons to believe that the first cell on this planet lived in an atmosphere that lacked oxygen. Even among present day living organisms, we know several that are adapted to anaerobic conditions. Some of these organisms are facultative anaerobes, while in other the requirement of anaerobic condition is obligate. In any case, all living organisms retain enzymatic machinery to partially oxidize glucose without help of oxygen. This breakdown of glucose to pyruvic acid is called glycolysis. 14.2 Glycolysis. The term glycolysis has originated from Greek word glykos means sugar and lysin is flating. The scheme glycolysis was given gusto emden automero of J. Parnassus and is often referred to as the EMP pathway. In anaerobic organism, it is only process in respiration. Glycolysis occurs in cytoplasm of cell and is present in all living organisms. In this process, the glucose undergo partial oxidation to form two molecules of pyruvic acid, and in plant this glucose is derived from sucrose, which is end product of photosynthesis or from storage carbohydrate. Sucrose is converted into glucose and fructose by enzyme invertase, and these two monosaccharides readily enter in glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. The phosphorylated form of glucose then isomerized to form fructose 6-phosphate. Subsequently, step of metabolism, glucose and fructose are same. The various steps of glycolysis is depicted in figure 14.1. In glycolysis, a chain of 10 reactions under the control of different enzymes take place to produce pyruvate from glucose. While studying the steps glycolysis, please note that steps are utilization in synthesis of ATP. In this case, NADH2 take place. ATP utilized in first two steps in the conversion of glucose into glucose 6-phosphate, and second step conversion of fructose into fructose 6-phosphate to fructose 1,6-phosphate. The fructose 1,6-bisphosphate is split into dihydroxy acetone phosphate and 3-phosphoglyceraldehyde phosphate PGAL. We find that there is one step where NADH2 is formed from NAD+. This is when 3-phosphoglyceraldehyde PGAL is converted to 1,3-bisphosphoglycerate BPGA. Two redox equivalents are removed in the form of two hydrogen bonds from PGAL to and transferred to a molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid PGA is also energy yielding process. This energy trapped by the formation of ATP. Another ATP is synthesized during conversion of PEP to pyruvic acid. Can you calculate how many ATP molecule directly synthesized from one glucose molecule? So directly synthesized ATP there are 2. But if you calculate net ATP, then we need to calculate नेट सिंथेसाइज एटीपी की बात कर रहे हैं ना, तो 2 कंज्यूम हो गया और एक ग्लूकोज से 4 एटीपी बन रहा है ग्लाइकोलाइसिस में. सिर्फ एटीपी का बात कर रहे हैं, NADPH2 नहीं इंक्लूड कर रहे हैं, तो यहाँ पे 2 ही एटीपी गेन हो रहा है. ठीक, अगर NADPH2 को ला लोगे तो वहाँ पे 8 हो जाएगा. Pyruvic acid then key product of glycolysis. What is metabolic fate of pyruvate? This depends on cellular needs. अब pyruvate का क्या यूज है आगे पढ़ेंगे, तब तक एक चार्ट देखते हैं. ग्लूकोज 6 कार्बन 1 ATP यूज हुआ तो ग्लूकोज 6 फॉस्फेट बना है 6 कार्बन का. फिर ये क्या हुआ, ये कन्वर्ट हुआ फ्रक्टोज 6 फॉस्फेट में. अभी फिर एक ATP का यूज हुआ तो फ्रक्टोज 1,6 बिसफॉस्फेट 6 कार्बन का. ये डिवाइड हो गया 2-3 कार्बन के प्रोडक्ट में. ट्राइगोज फॉस्फेट डाइहाइड्रोक्सी एस्टोन फॉस्फेट 3 कार्बन का और ट्राइगोज फॉस्फेट ग्लिसरॉल्डेहाइड 3 फॉस्फेट 3 कार्बन का. अब देखो यहाँ का जो नेक्स्ट स्टेप है, यहाँ पे NADH बन रहा है. इससे क्या बनता है, 3 ATP अपने को मिलता है, किसमें इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में अपने को मिलेगा. 2 ट्राइगोज फॉस्फेट इससे बन रहा है. इससे क्या हो रहा है, ATP. इसका ट्रिक मैं बना के अपलोड की जाऊँ भाई, वो मैं देसी ट्रिक में ही बनाया हूँ, उसको देख सकते हो अगर याद करना हो तो. 2x ट्राइगोज फॉस्फेट 3 फॉस्फोग्लिसरिक एसिड बन रहा है, इससे 2 फॉस्फोग्लिसरेट बन रहा है. फॉस्फोइनोल पायरुवेट बन रहा है, जिससे फॉस्फोपायरुविक एसिड बन रहा है. यहाँ देखने वाली बात है, यहाँ क्या निकल रहा है, पानी निकल रहा है 2 फॉस्फोग्लिसरेट के जगह पे. ठीक, और देखो there are 3 major ways in which different cell handle pyruvic acid produced by glycolysis. These are lactic acid fermentation, alcoholic fermentation and aerobic respiration. Okay, fermentation takes place under the anaerobic condition in many prokaryotes and unicellular eukaryotes for complete oxidation of glucose, CO2 and H2O. However, organisms adopt Krebs cycle, which also called aerobic respiration, required for oxygen supply. Of 14.3 fermentation. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under anaerobic condition by set of reactions where pyruvic acid converted to carbon dioxide and ethanol. The enzymes pyruvic acid decarboxylize and alcohol dehydrogenate catalyze these reaction. Other organisms like some bacteria produce lactic acid from pyruvic acid. The steps involved in shown in figure 14.2. In animal cells also like muscles, during exercise when oxygen is inadequate for cellular respiration, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent NADH2, which reoxidizes to NAD plus, and both process. In both lactic acid and alcohol fermentation, not much energy is released. Less than 7% energy in glucose is released, and not all the trapped in high energy bond of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is the net ATP that is synthesized? Calculate how many ATP are synthesized and deducted from a number of ATP utilized during glycolysis. When one molecule of glucose fermented to alcohol or lactic acid, yeast poison themselves. The concentration of alcohol reached 30%. What is then would be maximum concentration of alcohol beverage naturally fermented? 13%. How do you think alcoholic beverage alcohol content greater than this concentration obtained by what? क्या प्रोसेस बोलते हैं ब्रो? डिस्टिलेशन बोलते हैं क्या इस प्रोसेस को कंसंट्रेट करने का? What did the process by which organism can carry out complete oxidation of glucose, extract energy to store to the synthesis to larger number of ATP molecule needed for cellular metabolism. In eukaryotes, these steps take place in mitochondria and require oxygen. Aerobic respiration is the process that led to the complete oxidation of organic substance in presence of oxygen and release of carbon dioxide. Water and large amount of energy present in substrate. This type of respiration take most common in higher organism. We will look at this process in next section. So यहाँ देखेंगे, पहले डायग्राम देखते हैं. मेजर पाथवे ऑफ एनारोबिक रेस्पिरेशन. ठीक है, तो ग्लूकोज ग्लिसरॉल डाई 3 फॉस्फेट में चेंज हुआ. ग्लिसरॉल डाई 3 फॉस्फेट NADPH2 यहाँ रिलीज होता है. ठीक, 3 फॉस्फोग्लिसरिक एसिड में चेंज हुआ. This changes into phosphoenol and this changes into pyruvic acid. After pyruvic acid, in absence of oxygen, pyruvic acid may be converted into lactic acid or it may be converted into ethanol. जो ये फॉर्म गया ना NADH2, इसका यूज हो जाता है यहाँ पे और यहाँ पे भी, इसीलिए 2 ही ATP इसमें मिलता है. ओके, तो इथेनॉल प्लस कार्बन डाइऑक्साइड बन रहा है. पायरुविक एसिड इन अब्सेंस ऑफ ऑक्सीजन. और लैक्टिक एसिड फर्मेंटेशन कहाँ होता है? हम लोग को मसल में होता है, हैवीली एक्सरसाइज करा जाता है. ठीक है. 40.4 एरोबिक रेस्पिरेशन. For aerobic respiration to take place within mitochondria, the final product of glycolysis, pyruvate, is transported to cytoplasm into the mitochondria. The crucial event of aerobic respiration: first, the complete oxidation of pyruvate by stepwise removal of all hydrogen atom, leaving 3 molecules of carbon dioxide. Second, the passing on the electron removed as a part of hydrogen atom to molecular oxygen is simultaneously synthesized of ATP. Okay. What is interesting to note is that first process takes place in the matrix of mitochondria, while the second steps take place in a mitochondrial space. Pyruvate, which is formed by glycolytic catabolism of carbohydrate in cytosol after it enters the mitochondrial matrix, undergo oxidative decarboxylation by complex set of reactions catalyzed the pyruvic dehydrogenase. The reaction catalyzed by pyruvic dehydrogenase require a participation of several coenzymes including NAD plus and coenzyme A. Pyruvic acid plus coenzyme A in presence of NAD plus, magnesium catalyst and pyruvate dehydrogenase के presence में acetyl coenzyme plus कार्बन डाइऑक्साइड प्लस NADH2 फॉर्म होता है. इसको क्या बोलते हैं? लिंक रिएक्शन. लिंक रिएक्शन इसी को बोलते हैं ब्रो. During this process, two molecules NADH is produced from the metabolism of 2 molecules of pyruvic acid produced from 1 molecule of glucose during glycolysis. The acetyl enzyme enters cyclic pathway tricarboxylic acid, more commonly called Krebs cycle. Scientists given by Krebs first listed. So this acetyl enzyme enters in Krebs cycle or tricarboxylic acid cycle. We are going to study that. 14.4.1 tricarboxylic acid cycle. The tricarboxylic acid cycle starts with the condensation of acetyl coenzyme with oxaloacetic acid OAA. The water to yield citric acid. The reaction is catalyzed by enzyme citrate synthase and the molecule coenzyme A is released. Citrate is then isomerized to isocitrate. This followed by successive steps of decarboxylation leading to formation of अल्फा कीटोग्लूट्रिक एसिड. सो पायरुवेट यहाँ पे आया, लिंक रिएक्शन में एस्टिल कोएंजाइम बनाया. अब ए एंटर किया, 2 कार्बन का था, ये एंटर कर गया अपना क्रेब साइकिल में. अब क्रेब साइकिल में इसका एक यहाँ NADH2 बन रहा है, यहाँ कार्बन डाइऑक्साइड रिलीज हुआ, NADH2 यहाँ बना, NADH2 यहाँ पे GTP बन रहा है, FADH2 बन रहा है. तो देखो इसको पढ़ने के लिए पूरा डिटेल करना पड़ेगा. हम लोग NCERT रीडिंग कर रहे हैं तो समझ में आ नहीं सकता इतना. NCERT रीडिंग करना है. बस ये देखो यहाँ पे एक NADH2 रिलीज हुआ इसी स्टेप पे. यहाँ पे तो कंप्लीट स्टेप भी नहीं दिया हुआ है ना, उसको याद करने के लिए ट्रिक है, गूगल पे सर्च कर लेना है, मैं बना के डाल दूंगा. एंड सक्सेनिक कोएंजाइम द रिमेनिंग स्टेप्स द सिट्रिक acid cycle succinic coenzyme is oxidized to oxalic OAA oxaloacetic acid, allowing the cycle to continue. During the conversion of succinyl coenzyme, the succinic acid molecule of GTP synthesized. This is substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with simultaneously synthesis of ATP from ADP. Also, there is 3 point up cycle where NAD plus is reduced to NADH2 and 1 point where FADH is reduced to FADH2. The continued oxidation of acetyl coenzyme, by the way, tricarboxylic acid cycle required continued replenishment of oxaloacetic acid, the first member of cycle. In addition, it also required regeneration of NAD plus and FAD plus from NADH and FADH2 respectively. The summary of equation of phase respiration is given in the following. देखो अब ये जो पूरा इक्वेशन है इसका समरी यहाँ पे दिया है. NADH का यूज कितना 4, FADH 1 लगा, 2 वाटर मॉलिक्यूल लगे, ADP लगा, इनऑर्गेनिक फॉस्फेट, माइटोकॉन्ड्रियल मैट्रिक्स में 3 कार्बन डाइऑक्साइड रिलीज हुआ. तो देखो ये लिंक रिएक्शन प्लस क्रेब साइकिल दोनों को ऐड करके किया है, तो इसको भी याद लर्न करना इम्पोर्टेंट है ब्रो. We have till now seen that glucose has been broken down to release carbon dioxide and 8 molecules of NADH2. 2 of FADH2 have been synthesized besides just 2 molecules of ATP and tricarboxylic acid cycle. You may have wondering why we have been discussing respiration at all. Neither oxygen has come into the picture nor promised large number of ATP has yet been synthesized. Also, what is the role of NADH2, the FADH2 that synthesized late? Let's now understand the role of oxygen respiration and how the synthesis occur. तो देख भाई यहाँ पे जो NADH2 FADH2 यूज हो रहा है, ऑक्सीजन का यूज आ रहा है, तो ऑक्सीजन ना हम लोग यूज नहीं देखे. इन सब का यूज अपन कहाँ करते हैं? इन सब का यूज आता है ETS में, और ETS में बनाते हैं क्या ATP. तो जितना भी हम लोग का देखो ग्लाइकोलिसिस में भी NADH2 बना था, यहाँ भी बन रहा है. इसका सबका यूज किस में करेंगे? ETS में, इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम को कैसे करेंगे देखते हैं. 14.4.2 इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम ETS एंड ऑक्सीडेटिव फॉस्फोराइलेशन. लेट्स ग्रो ब्रो. The following step of respiratory process to release and utilize the energy stored in NADH2 and FADH2 This is accomplished when they are oxidized throughout the electron transport system and electrons are passed on to oxygen, resulting in the formation of water. The metabolic pathway through which electron passes from one carrier to another is called electron transport system, ETS, and it is present in inner mitochondrial membrane. Electron from NADH produced in mitochondrial matrix during citric acid cycle oxidized by NADH dehydrogenase complex first, and electron are then transferred to ubiquinone located within the inner membrane. Ubiquinone also received reducing equivalent, by the way, FADH2 complex second, that is generated during oxidation of succinate in citric acid cycle. The reduced ubiquinol is then oxidized with the transfer of electron to cytochrome C by the way of cytochrome BC1 complex third. Cytochrome C is also a small protein attached to the outer surface of inner membrane and acts as a mobile carrier for transfer of electron between complex third and 4. Complex 4 referred to as cytochrome C oxidase complex containing cytochrome A and A3 and 2 copper centers. When the electrons are passed from one carrier to another, by the way, complex 4 to 4, the electron transporters they are coupled to ATP synthase complex 5 for the production of ATP from ADP and inorganic phosphate. The number of ATP molecule synthesized depend on the nature of electron donor. Oxidation of one molecule of NADH give rise to 3 molecules of ATP, while that of one molecule of FADH to produce 2 molecules of ATP. Although aerobic process respiration take place through only in the presence of oxygen, the role of oxygen is limited to terminal stage of the process. Yet the presence of oxygen is vital since it drives whole process by removing hydrogen from the system. Oxygen acts as final hydrogen acceptor. So final hydrogen एक्सेप्टर कौन है? ऑक्सीजन है. ठीक. Unlike phosphorylation, where it is light energy that utilized for the production of proton gradient required for phosphorylation, in respiration it is energy oxidation and reduction utilized for same process. It is for this reason the process is called oxidative phosphorylation. You have already studied about the mechanism of membrane linked ATP synthesis explained in chemiosmotic hypothesis. ये जो पढ़े तो photosynthesis में in earlier chapter. As mentioned earlier, energy released during electron transport system utilized in synthesis of ATP with a help of ATP synthase complex 5. This complex consists of two major components, F1 and F0. The F1 is headpiece. This is headpiece peripheral membrane protein complex containing the site for synthesis of ATP. Here ATP formed from ADP, the inorganic phosphate. F0 is an integral membrane protein complex that form the channel through which proton cross the inner membrane. The passage of proton through the channels is coupled to catalytic site of F1 component and production of ATP. So इधर से दो हाइड्रोजन गया और यहाँ पे एक ATP फॉर्म हो रहा है. For each ATP 2H+ pass through F0 from the intermembrane space of matrix down the electrochemical gradient. तो देखो एक ATP के लिए दो प्रोटॉन पास हो रहा है. इसको देख लेना भाई, क्योंकि ये बाहर से भी देख लेना इसलिए क्योंकि यहाँ पे प्रोटॉन का जो है ना, इसमें कई NCERT में क्या क्या दिया हुआ है, इसको चेक कर लेना एक बार. आउटर साइड इनर माइटोकॉन्ड्रियल मेम्ब्रेन यहाँ पे मैट्रिक्स में. अब देखो यहाँ पे इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम ETS. तो ये रहा मैट्रिक्स. ये हियर इज इनर माइटोकॉन्ड्रियल मेम्ब्रेन, इधर हियर इज इंटर मेम्ब्रेन स्पेस. तो देखो पहला कॉम्प्लेक्स का नाम क्या है? NADH डिहाइड्रोजनेस. सेकेंड कॉम्प्लेक्स देखो, ये मिस्टेक नहीं करना. सेकेंड ई फर्स्ट हुआ, ए होता है थर्ड कॉम्प्लेक्स, ये है सेकेंड कॉम्प्लेक्स, ये और ये है फोर्थ कॉम्प्लेक्स, एंड लास्ट में आता है ए. मतलब क्या करता है? ATP जो फॉर्म कर रहा है इसको क्या बोलते हैं ब्रो? याद नहीं आ रहा ब्रो मेरे को. मतलब एटीपी सिंथेज यहाँ पे लास्ट में आ रहा है। तो फर्स्ट जो कंपोनेंट है इसको क्या बोलेंगे, NADH डिहाइड्रोजनेज। यहाँ पे NADH2 आ रहा है, NAD यहाँ पे हाइड्रोजन देंगे तो ये इसीलिए NADH डिहाइड्रोजन बोलेंगे। और कम्प्लेक्स थर्ड यहाँ पे बोलते हैं, कम्प्लेक्स थर्ड को साइटोक्रोम PC1, और कम्प्लेक्स सेकेंड को बोलते हैं सक्सिनेट डिहाइड्रोजिनेज, यहाँ पे सक्सिनेट फ्यूमरेट में कन्वर्ट हो रहा है। और देखो कॉम्प्लेक्स 4 को साइटोक्रोम C ऑक्सीडेज बोलते हैं। और देखो एक और है मोबाइल कैरियर बोलता है, इलेक्ट्रॉन का कहाँ से कहाँ तक कैरी करा रहा है, तो मोबाइल कैरियर साइटोक्रोम C है। यहाँ पे एक कॉम्प्लेक्स थर्ड से इलेक्ट्रॉन को लेकर कहाँ जा रहा है ब्रो, कॉम्प्लेक्स 4 में। मोबाइल कैरियर कौन है, साइटोक्रोम C, ये भी आ सकता है। तो भाई 2025 इस डायग्राम से क्वेश्चन नहीं आया इस डायग्राम से, तो NTA आएगा ब्रो, छोड़ो। 14.5 दी रेस्पिरेटरी बैलेंस शीट। It is possible to make calculation of net gain of ATP for every glucose molecule oxidized, but in reality this can remain only in theoretical exercise. These calculations can be made only on certain assumption that first, there is a sequential orderly pathway functioning with one substrate forming the next, and with glycolysis, tricarboxylic acid cycle and electron transport system pathway following one after another. Second, the NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. Third, non-intermediate pathway are utilized in synthesis of any other compound. Four, only glucose is being respired, no other alternative substrate are entering pathway at any other intermediate stage. But this kind of assumption are not really valid. Living system, all pathway works simultaneously and not take place one after another. Substrate enter the pathway and drop from it as and when necessary. ATP is utilized as when needed. Enzymatic rate are controlled by multiple means. Yet it is useful to do this exercise to appreciate the beauty of efficiency of living system and extraction of storing energy. Hence, there can be net gain of 38 ATP molecules during aerobic respiration in one glucose molecules. Now let us compare the fermentation and aerobic respiration. Fermentation first. Fermentation accounts for the only partial breakdown of glucose, whereas the aerobic respiration is completely degraded of carbon dioxide and water. Second, fermentation there is net gain of ATP, 2 molecules of ATP for each molecule of glucose degraded to pyruvic acid, whereas many more molecules of ATP generated in aerobic. Okay. NADH is oxidized to NAD plus rather slowly fermentation. However, the reaction is very vigorous in case of aerobic. So NAD में कन्वर्ट तो हो रहा है दोनों, बट क्या है कि एरोबिक में क्या हो रहा है, बहुत विग्रस हो रहा है. 40.6 एम्फीबोलिक पाथवे. Glucose is favored substrate for respiration. All carbohydrates are usually first converted into glucose before they are used for respiration. Other substrates can also be inspired as they've been mentioned earlier, but then they do not enter the respiratory pathway at the first step. See figure 14.6. See the entry of different substrate in the respiratory pathway. Fat would need broken down into glycerol fatty acid first. If fatty acid were respired, they would first degrade to acetyl coenzyme entering the pathway. Glycerol would enter the pathway and after being converted to PGAL, the proteins would be degraded by protease and individual amino acid after deamination, depending on their structure, would enter pathway at some state with Krebs cycle or even pyruvate or acetyl coenzyme. Since respiration involves breakdown of substrate, the respiratory process has traditionally been considered a catabolic process, and the respiratory pathway is catabolic pathway. But is the understanding correct? We have discussed above at which point in respiratory pathway different substrate would enter if they were to respired and used to derive energy. What is important to recognize is that these very compounds that would be drawn from respiratory pathway for the synthesis of said substrate, hence fatty acid would be broken down to acetyl coenzyme before entering the respiratory pathway when it is used as substrate. But when organism need to synthesize fatty acid, acetyl coenzyme would withdraw respiratory pathway for it. Hence, this respiratory pathway come into the picture both during air breakdown and synthesis of fatty acid. Similarly, during the breakdown and synthesis of protein, two respiratory and intermediate form link. Breaking down the processes within the living organism, catabolism, the synthesis is anabolism. Because of respiratory pathway involving both anabolism and catabolism, it would be better consider respiratory pathway amphibolic rather than its catabolic one. Here is fat, carbohydrate, proteins. Protein form amino acid and amino acid form pyruvic acid, and in this converted into acetyl coenzyme, and fat also converted into fatty acid or glycerol, and the fat or glycerol converted into dihydroxyacetone phosphate, and this isomerized to glyceraldehyde 3-phosphate, and these finally converted into acetyl coenzyme. After all, they go in Krebs cycle and form water and carbon dioxide. Okay, interrelation of metabolic pathway. So in respiration mediated process different organic molecule carbon dioxide and water. (40.7) Respiratory quotient. Let's now look at another aspect of respiration. As you know, during aerobic respiration, oxygen is consumed and carbon dioxide is released. The ratio of volume carbon dioxide evolved and volume of oxygen consumed in respiration is called respiratory quotient, RQ, respiratory ratio. RQ is equal to volume of carbon dioxide evolved and oxygen by volume of carbon dioxide oxygen consumed. Sorry, bro. The respiratory quotient depends upon the type of respiratory substrate used during respiration. When carbohydrates are used substrate and completely oxidized, RQ value is 1 because equal amount of carbon dioxide and oxygen evolved consumed respectively in the given equation. Like ग्लूकोज में जितना ऑक्सीजन यूज होगा उतना ये रिलीज हो रहा है, CO2 रिलीज बाई ऑक्सीजन कंज्यूम 6 तो RQ 1 हो जा रहा है. ऐसे ही क्या करना है, व्हेन फैटी एसिड इंस्पायरेशन RQ लेस देन 1, फैटी एसिड को ले रहे हैं तो RQ लेस देन क्या हो जा रहा है 1, क्योंकि 102 CO2 रिलीज हुआ और 145 O2 लगा, मतलब O2 ज़्यादा है तो इस RQ लेस देन 7 हो गया. व्हेन प्रोटीन इज रेस्पिरेटरी सबस्टेंस रेशियो वुड बी 0.9. अब देखो प्रोटीन अगर रेस्पिरेटरी सबस्टेंस में रह रहा है तो RQ क्या है 0.9, इससे थोड़ा ज़्यादा है बट कम है ग्लूकोज से. व्हाट इम्पोर्टेंट टू रिकॉग्नाइज ऑर्गेनिज्म रेस्पिरेटरी सबस्टेंस ऑफन मोर देन 1, प्योर प्रोटीन और फैट नेवर यूज रेस्पिरेटरी सबस्टेंस. व्हाट इम्पोर्टेंट टू रिकॉग्नाइज दे आर लिविंग ऑर्गेनिज्म रेस्पिरेटरी सबस्टेंस आर ऑफन मोर देन 1. देखो बहुत लिविंग ऑर्गेनिज्म है जिसका रेस्पिरेटरी कॉन्स्टेंट क्या होता है मोर देन 1 होता है. जैसे कि मान लो क्या होता है एस्टिक एसिड वगैरह का लगभग, इसका देखना पड़ेगा, इसका कहीं एस्टिक एसिड वगैरह था ना, कमेंट में बता देना भाई. समरी. Plant unlike animal have no special system of breathing and or gaseous exchange. Stomata and lenticel allow gaseous exchange by diffusion. Almost all living cells in plants have their surface exposed to air. The breaking of carbon bond complex organic molecule by oxidation cells leading to release of lot of energy is called cellular respiration. Glucose is the favored substrate for respiration. Fat and protein can be broken down into yield energy. The initial stage of cellular respiration takes place in the cytoplasm. Is glucose molecule is broken through a series of enzyme catalyzed reaction into two molecules pyruvic acid. This process is called glycolysis. The fate of the pyruvate depends on the availability of oxygen to organism. Under anaerobic condition, either lactic acid fermentation or alcohol fermentation occur. Fermentation take place under the anaerobic condition. Many prokaryotes, unicellular eukaryotes, and germinating seeds. In eukaryotic organism, an aerobic respiration occur in the presence of oxygen. Pyruvic acid is transported into the mitochondria, where it converted to acetyl coenzyme when released to with release of carbon dioxide. Acetyl coenzyme A, when entered to tricarboxylic acid pathway or Krebs cycle operating in the matrix of mitochondria, NADH plus H plus FADH2 are generated in the Krebs cycle. The energy of these molecules are well as that NADH2 is synthesized during glycolysis used to synthesize ATP. This accomplished through a system of electron carriers called electron transport system or ATP ETS located on the inner membrane of mitochondria. The electron, as they are moved through the system, release enough energy that trapped synthesize ATP. This is called oxidative phosphorylation. In this process, oxygen is ultimately accepted electrons and set of reduced water. The respiratory pathway is an amphibolic pathway as it involves both anabolism and catabolism. Respiratory quotient depends upon the type of respiratory substrate used in respiration. तो भाई अपना respiration भी complete हुआ. नेक्स्ट चैप्टर चैप्टर 15 प्लांट ग्रोथ एंड डेवलपमेंट. इसको अपन करेंगे 8:47 से ब्रेक ले रहा है टू 8:52 AM स्टार्ट करेंगे. तब तक के लिए भाई आराम करते हैं, थोड़ा सांस ले लें भाई. रिस्पायरेशन पढ़ के आए तो थोड़ा सांस ले लें. ब्रो तो भाई लोग ब्रेक लें, 2 मिनट का और अभी ब्रेक है. तो ब्रेक में कुछ शायरी हो जाए क्या ब्रो? तो एक तो है मोटिवेशन बोलो या डीमोटिवेशन. आज वही कल है जिस कल का फिक्र हमें कल थी, है कि नहीं? हम लोग का यही कल है ना. कल हम लोग का फिक्र था और जो आने वाला कल है उसका आज फिक्र है. तो आज ही सुधारते हैं, कल तो सुधर जाएगा ही ना ब्रो. एक शायरी बोलते हैं, रुको बताता हूँ. एक अच्छा शायरी दिखा. एक उम्र निकाल दी बिना मिले तुमसे. सुनो ध्यान से. एक उम्र निकाल दी बिना मिले तुमसे और लोग हमें इश्क करना सिखा रहे हैं. वाह से वाह. ये पता है कौन इस किस पे जमता है ब्रो? ये शायरी जमता है रेखा पे, अमिताभ के लिए. या सलमान का ऐश्वर्या के लिए, वाह क्या शेर। और कोई अच्छा ऐसे दिखे 2 मिनट है अपने पास। हम्म। तो एक और शायरी है भाई। आज थोड़ी बिगड़ी है, कल फिर संवार लेंगे। ठीक, आज थोड़ी बिगड़ी है, कल फिर संवार लेंगे। जिंदगी है, जो भी होगा संभाल लेंगे ब्रो। क्या भाई, थोड़ा सा बिगड़ा है, कल संवार लेंगे कि नहीं? थोड़ा सा बिगड़ा है, कल संवार लेंगे। जो भी होगा ज़िंदगी में आगे संभाल ही लेंगे भाई। पेरेंट हमको इतना संभाल ही रहे हैं, माता पिता, तो ब्रो हम लोग नहीं संभालेंगे यार? आगे क्या है ब्रो, आगे शायरी। अभी अपने पास 1 मिनट टाइम है। दूसरों को समझने से बेहतर है अपनी बारे में पूरी समझ रखना, समझे? इसीलिए दूसरों को नहीं समझो ब्रो, अपने आप को समझो और आगे बढ़ो, मूव ऑन। डोंट वेस्ट योर टाइम इन अंडरस्टैंडिंग एनीवन। थैंक यू शुभम ब्रो, नाइस वर्क। अभी ब्रो 52 से स्टार्ट करा जाएगा अपना प्लांट ग्रोथ एंड डेवलपमेंट। अभी ब्रेक है भाई, कोई शायरी हो तो बता दो यार तुम लोग भी। तन्हाई में क्या ही करें, शायरी याद आ रहा, नहीं तो एनसीईआरटी। लेट्स गो ब्रो, टाइम हुआ 52। लेट्स गो। प्लांट ग्रोथ एंड डेवलपमेंट 15.1 ग्रोथ, 15.2 डिफरेंशिएशन, डी डिफरेंशिएशन एंड रीडिफरेंशिएशन, 15.3 डेवलपमेंट, 15.4 प्लांट ग्रोथ एंड रेगुलेटर्स, 15.5 फोटो पीरियोडिज्म एंड 15.6 वनलाइजेशन। नहीं पढ़ूंगा भाई ये, क्योंकि इस पर नीट में आ नहीं रहा है, तो क्या ही मतलब है। सो लेट्स गो। You have already studied the organization of flowering plant, chapter 5. Have you ever thought about where and how the structure like roots, stems, leaves, flowers and fruits and seeds arise, and what to in an orderly sequence? You are now aware of the term of seedling plantlets, mature plants. You have also seen that tree continue to increase in height, girth over period of time. However, the the leaves, flowers, and fruit of the same tree not only have limited dimension but also appear and fall periodically and sometimes repeatedly. Why does vegetative phase precede flowering in plants? All plants' organs are made up of variety of tissue. In is any relationship between structure of cell, tissue, and organs and function they perform? Can the structure function in these can be altered? All cells of plant descendant from zygote. This question is then why and how do they have different structure and function attributes? Development is the sum of two process, growth and differentiation. To begin, देखो development, growth and plus differentiation. To begin with the essential sufficient to know that the development and mature plant from zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces root, leaves, branches, flowers, fruits, and seeds. Eventually, they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable condition of growth exist in environment. In absence of such favorable conditions, the seed do not germinate and goes into period of suspended growth or rest. Once favorable condition return, the seed resume metabolic activity and growth take place. In this chapter, you shall also study some factors uh, which govern and control the development of process. These factors are intrinsic internal or extrinsic external to the plants. Germination of seedling in beans, soil line, cotyledon, hypocotyle, seed coat, cotyledon, epicotyle hook, epicotyle, hypocotyle, and these are hypocotyle. दी जो कोटाइलडन से नीचे उसको हाइपोकोटाइल, जो कोटाइलडन से ऊपर है उसको एपिकोटाइल. बस सिंपल. 15.1 growth is regarded as one of the most fundamental and conspicuous characteristics of living beings. What is growth? Growth can be defined as an irreversible permanent growth in size of organ and its part or even individual cell. Generally, growth is accompanied by metabolic process, both anabolic and catabolic, that occur in the expense of energy. Therefore, for example, expansion of leaf is a growth. How do you describe the swelling piece of wood when placed in water? This is imbibition. Sorry, bro. 15.1.1 Plant growth generally is indeterminate. Plant growth is unique because plant retain the capacity of unlimited growth throughout their life. The ability of plant is due to the presence of meristematic at certain location in their body. The cells of such meristem have capacity to divide and perpetuate. The product, however, soon loses the capacity to divide, and such cell make up plant body. This form the growth wherein new cells are always being added to the plant body by activity of meristem, called open form growth. What would happen if the meristem ceases to divide? Does this ever happen? In chapter 6, you have a study about root apical meristem and shoot apical meristem. You know what is responsible for the primary growth of plant, principally contributed to elongation of plant along their axis. You also know that it's in dicotyledonous plant gymnosperm, the lateral meristem, vascular cambium, and core cambium appear later in life. These are meristems that causes the increase in girth of organ in which they active. This known as secondary growth of plant. Here is diagrammatic representation of location of root apical meristem, shoot apical meristem, and vascular cambium. Arrows exhibit the direction of growth in cellular organ, root and shoot. Here is shoot apical meristem, and here is root apical meristem, and here is vascular cambium and vascular cambium. 15.1.2 Growth is measurable. Growth at the cellular level is principally a consequence of increase in the amount of the protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, in length, area, volume, and cell number. You may find it amazing to know that one single mesh root apical meristem can give rise to more than 70,500 new cells per hour, whereas cell in a watermelon may increase in size up to 350,000 times. In the former, growth is expressed in the increase in cell number. The latter expresses growth as increase in size of the cell, while the growth in pollen tube is measured in terms of length, as increase in the surface area denoted the growth of dorsiventral leaf. 15.1.3 Phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation. Figure 15.3. Let us understand this by looking at the root tip. The constantly dividing cell, both the root apex and shoot apex, represent a meristematic phase of growth, The cell of this region is rich in protoplasm, possess large conspicuous nucleus. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connection. This cell proximal to just next away from the tip to meristematic zone represents the phase of elongation. Increasing vacuolation, cell enlargement, and new cell wall deposition are the characteristic of cell in this phase. Further away from the apex that is more proximal to the phase of elongation lies the portion axis which is undergoing phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modification. Some of tissue and type you have studied in chapter 6 represent this phase. Detection of zone of elongation by parallel technique method. Zones A, B, C, D immediately behind the apex have elongated most. Okay. 15.1.4 growth rate. The increase in growth per unit time is termed as growth rate. Thus, the rate of growth can be expressed mathematically as an organism or plant of organism organism can produce more cells in variety of ways. Here is arithmetic growth. One cell divided into two cells, and these are divided in multiple of 1 to 2. In geometrical, these are divided in geometric fashion. Zygote divide, and from here to here is geometric phase, all cell divide, and here is arithmetic phase of growth. Diagrammatic representation: A arithmetic, B geometric growth, C stage during embryo development showing geometric and arithmetic phase. Growth rate so increase that may be arithmetic or geometric. In arithmetic growth following mitotic cell division, only one daughter cell continues to divide while the other differentiate and mature. The simplest expression of arithmetic growth is exemplified by root elongating at a constant rate. Look at figure 15.5. On plotting the length of the organ against the time, a linear curve obtained. Mathematically, it expresses LT is equal to L naught plus RT. LT is equal to length at time t, L naught length at time 0, R is equal to growth rate elongation per unit time. Here is constant linear plot against L, length L against time t. It is initial length L naught, and after time t this follow this equation. Linear equation. Let us now see what happens to geometrical growth. In most systems, the initial growth is slow lag phase, and it increases rapidly thereafter at exponential rate, log or exponential phase. Here, both the progeny cells following mitotic cell division retain the ability to divide, continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typically sigmoid S curve. A sigmoid curve is characteristic of cell organisms growing in a natural environment. It is typically of all cell, tissue, and organs of plants. Can you think of more similar examples? That what kind of curve can you expect a tree showing seasonal activity? The exponential growth can be expressed as W1 is equal to W0 e to power RT, where W1 is final size, W0 is initial size beginning period, R is growth rate, T is time of growth, E is base of natural logarithm. Okay, this is sigmoid curve, and here is size and weight. Here it is known as lag phase, and this is exponential phase. After this we get stationary phase. Here R is the relative growth rate and is also measured of the ability of plant and to produce new plant material, referred to as efficiency index. Hence the final size W1 depends on the initial sign W naught. 15.5 diagrammatic comparison of absolute and relative growth rate. Both leaves A and B have increased their area by 5 cm square by time A and B. Quantitative comparison of between growth and living system can be also be made two way. Measurement and comparison of total growth per unit time called absolute growth. The growth of given system per unit time expressed as common basic. For example, unit initial parameter of given relative growth rate. The figure 15.2 growth rate A and B drawn at different size but show absolute increase in area in given time to give leaves A1 and B1. However, one of them show much higher relative growth. which one of the following. So if you देखो रिलेटिव ग्रोथ देखेंगे ब्रो तो देखो यहाँ पे 10 सेंटीमीटर स्क्वायर पहले 5 था तो इसका 10 सेंटीमीटर स्क्वायर हो गया, पहले क्या था 5 था तो रिलेटिव ग्रोथ इसका क्या हुआ ज़्यादा हुआ, डबल हो रहा है। और यहाँ पे देखो 5 इसका बढ़ रहा है, सिर्फ 55 ऊपर है नीचे 50, तो इसका रिलेटिव ग्रोथ क्या उतना नहीं हो रहा है। ठीक, एब्सोल्यूट ग्रोथ इक्वल है दोनों का, दोनों 5 है 5 सेंटीमीटर स्क्वायर, बट रिलेटिव ग्रोथ क्या है इसका ज़्यादा है इस लीफ का। ओके आगे दो 50.1.5 कंडीशन फॉर ग्रोथ। why do not try to write down what you think are necessary condition for growth. This list may have water, oxygen and nutrient as very essential element for growth. The plant cell grow in size by cell enlargement, which in turn require water. Turgidity of cells help in extension of growth. Thus plant growth and further development is intimately linked to the water status of plant. Water also provides the medium of enzymatic activities needed for plant. Oxygen help in releasing metabolic energy essential for growth activities. Nutrients, macromolecules are required by plants for synthesis of protoplasm and act as source of energy. In addition to every plant organism, optimum temperature range best suited for growth. Any deviation from range could be detrimental to survival. Environmental signals such as light and gravity also affect the certain stages of growth. 50.2 differentiation, de-differentiation and redifferentiation. नोट कर लो भाई, 25 में क्वेश्चन इससे आएगा। नहीं आया तो क्या ही करोगे। The cell derived from root apical, shoot apical meristem and cambium differentiate and mature to perform a specific function. This act leading to maturation is termed as differentiation. During differentiation, cell undergoes few to major structural changes, both in their cell walls and protoplasm. For example, to form a tertiary element, the cell would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell wall to carry water to the distance even under extreme tension. Try to correlate the various anatomically features you encounter in plant to function they perform. Plants show another interesting phenomena. The living differentiate cell that by now have lost the capacity to divide and regain capacity to division under certain conditions. This phenomenon is termed as de-differentiation. For example, formation of meristem, interfascicular cambium and core cambium from fully differentiated parenchyma is example of de-differentiation. While doing so, such meristem tissue are able to differentiate and produce cells that once again lost the capacity to divide but mature to perform a specific function and get redifferentiated. List of some of this tissue would be dicotyledonous plant product redifferentiated. How would you describe tumor? What you call the parenchyma? Parenchyma cell that made to divide under control laboratory condition during plant tissue culture. Recall the section 15.1.1. We have mentioned that the growth in plant is open, that is, it can be indeterminate or determinate. Now we may say that even differentiation in plant is open because cell tissue are arised out the cell meristem have different structure at maturity. The final structure at maturity of a cell tissue is also determined by location of cell within. For example, cell position away from root apical meristem differentiate as root cap cell, while those pushed to the periphery matured as epidermis. Can you add a few more examples of open differentiation correlating the position of a cell position is in an organ? 15.3 डेवलपमेंट. तो यार आवाज़ नहीं जा रहा है, उसके लिए क्या करूँ मैं? रूम में हूँ, यहाँ पे ज़्यादा तेज़ बोल भी नहीं सकता हूँ, सॉरी उसके लिए मेरा. Development is a term that includes all changes that an organism does through during its life cycle, from germination of seed to senescence. Diagrammatic representation of sequence of processes which constitute the development of cell higher plant is given in figure 15.8. It is also applicable to tissue and organs. Sequence of developmental process in plant cell: meristematic cell, plasmatic growth, differentiation, and in simultaneously यहाँ पे एक्सपेंशन भी हो रहा है, एलोंगेशन हो रहा है, एंड मैचुरेशन. यहाँ इसका डायग्राम से डायरेक्ट क्वेश्चन आता है भाई, तो इसको देख लेना एक बार. मैच्योर सेल सीनेसेंस, उसके बाद डेथ हो गया, फिर से प्लाज्मेटिक ग्रोथ. Plant follow different pathway to respond environment or phases of life form different kind of structure. This ability called plasticity. That is heterophyly in cotton, coriander, and larkspur. In such plant, the leaves of juvenile plant are different in shape from those in mature plant. On the other hand, differences in shapes and leaves produces in air, and those produced in water or buttercup also represent the heterophilous development due to environment. This phenomenon is known as heterophyly. So here heterophyly का example larkspur और buttercup क्या है, environmental का example, और environmental और age के साथ ही क्या हो रहा है, juvenile to adult होने के साथ larkspur इसका example हो गया. इससे भी क्वेश्चन आएगा ब्रो, याद कर लो. कॉटन, कोरिएंडर, larkspur, C2L ड्यू टू एज के साथ हो रहा है इसमें चेंजिंग, और बटर कप ड्यू टू इन्वायरमेंटल. Thus, growth, differentiation, and development are very closely related to event in the life of a plant. Broadly, development is considered as the sum growth and differentiation. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. The former include both intracellular genetic and intracellular factor, chemicals such as plant growth regulator, while the latter include light, temperature, water, oxygen, and nutrition, etc. 15.4 Plant growth regulators. 15.4 Characteristics. The plant growth regulator PGR are small simple molecule diverse chemical composition